So, shall we start? So, good morning, everyone. It's our great pleasure to see you and to welcome you today. I would like to thank all the on-site participants and all the online participants. So on-site we should be in between around 60 people and online around 100 people. And so welcome and thank you. And on behalf of Marielle and me, it's a great pleasure to see you. So I would like to thank the EGP, the AZID and the RN-RITA for making this event possible. Of course, the speakers, of course, the attendees also. I would like also to thank our two sponsors, PikeBio and BioNanogenomics. And we hope that this event will be a success, that you will enjoy it. For many of you, please do not forget to fill the evaluation form at the end to get your credit points, and for the EGP members to get reimbursed. I leave the place to Marielle to say some words and enjoy. And this is a workshop. We hope you to ask questions. We hope that it will be extremely interactive. So do not hesitate to ask any question. There is no stupid question. It's just, I'll be the first one to ask stupid questions. So go ahead and try to interact with everyone. Thank you. Marielle?
So I'm Marielle van Gijn, the chair of the Molecular Testing Working Party from ER and Rita. And I would like actually to thank Anne for the opportunity for us together host this event at the Institut Imagine, which is a perfect place, I think, in Paris. We can enjoy Paris as well in the evenings. So for the on-site people, welcome. Also the online people, welcome. And all the EGP granted people, very welcome. And please, like Anne already said, do interact, do ask questions. And we are looking forward to your case presentations of this afternoon and tomorrow afternoon. And I think that's all I would like to say. And I would like to leave the floor to Isabelle, our first speaker.
More minutes, because I don't want Isabelle to start and having a number of people not attending.
Okay, so we can wait. Then I can say that can I talk longer, no problem. So please, speakers, keep to your time. We keep you more or less to your time. Because we have a lot of presentations, a lot of short, informative presentations. And to give the other presenters a chance to speak as well. I see there's someone with a question.
Yes, there is. Is there a Wi-Fi access?
Yes. So I wanted to tell it. So on Wi-Fi, you have to load onto the Imagine guest. And on the bottom, you ask for code. You need a name. You have to fill the form, and they will give you the code. Keep it for the 2 days. It's voilà.
Yeah, you have to register more or less with your name and email. Yeah. Any more questions? No. Okay. Yeah.
So it's my great pleasure to invite Isabelle for the first presentation. And Isabelle is going to talk about next generation sequencing of inborn errors of immunity. And Isabelle, so everyone knows Isabelle, so I think I don't have to introduce her. And in addition to all this, is the president of the ESIN. So Isabelle, welcome, and looking forward to your talk. Thank you.
Good morning, Anne. Good morning, Marielle. And thank you very much for inviting me. And I'm really proud that this happened at the end, hopefully, of the pandemic, and that we get to be together and discuss genetics and learn more. So I would describe myself as an intelligent user of next generation sequencing, and not really the expert, but I'm going to tell you about what I learned about next generation sequencing and how I discovered many pitfalls. So also, let's start with actually the scene. As we are involved in the field of inborn errors of immunity, we just are confronted with an amazing and still ever increasing number of inborn errors of immunity that get discovered with different gene defects. And actually, as we are speaking, the next update of the International Union of Immunological Societies is in press. And we are now at more than 480 defects, almost 500, and many more to be described undoubtedly. And it's true that different mutations in a single gene can cause different phenotypes, different modes of inheritance can be in play, and there can be a different effects of the genetic variant or the mutation. So all resulting in different pathogenesis and phenotypes. So in fact, with a few examples, it has become impossible to have a diagnostic test just using Sanger sequencing or very straightforward methods. So we almost always rely on next generation sequencing for a diagnosis in inborn errors of immunity. And you can also see here the gap when next generation came into play, actually around 2011. You can see that there has been, so is that my mistake or what is it? Well, you could have. Let me check. Does this do something? Because here I'm definitely in, yes, I'm not inventing my slides.
So how come the slides?
Because I've had this before, and it can be sometimes that we have two screens or two windows, but I don't have my IT wizard with me today, but.
We have one.
Désolé. So I have my slides here, but they don't project.
Okay.
That's why they took me as the first speaker.
Because
I'm not stressable.
that's good. That's cool.
Which one?
This one. Okay, thank you. And you can see actually here in 2011, that's when next generation sequencing came really into research and became more affordable, that you see that there has been a steep further increase in our unraveling of inborn errors. So this is actually a slide taken from the upcoming presentation, and you can see that we are now really heading towards the 500 gene defects. And actually, when we were working on it, we discovered a few more. So it's become really a big task to follow up with all these new discoveries. And I would also like to stress, so in recent years, in the recent update, there is going to be a stress on new defects in innate immunity, and also not coincidentally combined with autoinflammation. And what is also quite interesting is that 16 of the 55 novel gene defects have actually been unraveled by the study of single patients. And I think that's really something that is, that is really interesting, and that also confronts us with the fact that each patient, in fact, is a research project potentially. So at the very least, we can say that every patient with a primary immunodeficiency is really a diagnostic challenge. And so if we have a patient with a suspected inborn error of immunity, of course, we can rely on different tools to work out the diagnostics, but whatever happens, we will have to do a strong validation to be able to decipher either a novel phenotype for a known gene defect or a novel gene defect to start with. So of course, the ideal phenotype of the patient that we are confronted with would result, let's say, in a small set of genes to be sequenced. And then we could refer to traditional sequencing methods, as you're all aware of. What we sometimes still do for confirming genetic diagnosis of NGS is a Sanger sequencing, which has a very low error rate, but which is really low throughput because of the mechanism and the technique itself. I think it's still useful, and we all know that, for sequencing a small set of candidate genes, but it's definitely not the first choice anymore in the most diagnostic situations. So the perfect example would, of course, be the B- patient, if it's a boy, a gamma globulinemic patient, where you would go for BTK sequencing, of course. But all the other patients would say probably fall into this group, where we either go for a targeted panel, which could be the exome, or, oops, it's moving all the time, or could be a set of known PID genes, which is usually used in clinical settings with panels that are adapted each year. So the big difference, in fact, between genome sequencing and whole exome sequencing here, or between panel sequencing, would be the capture step, where we have to enrich the set of genes that we are interested in by a capture and hybridization step, which almost always involves a PCR step, which is in the same time the Achilles heel of the procedure, because that introduces polymerase errors, sampling errors, and also, let's say, template errors, etc. So we have to realize that when we sequence the exon, we sequence much more than just the exons of the protein coding genes. We also sequence a number of microRNAs, small nucleolar RNAs, and also some intergenic non-coding RNA genes. And so this is, in fact, the flow schedule of what happens when you do whole exome sequencing. You have the genomic DNA, which is, in fact, deconstructed into a DNA library, which usually also involves a PCR step if you don't have a lot of genomic DNA to start with. Then you have to repair the ends of these cut DNA fragments, which are then hybridized for the capture step. And then you go either for whole genome sequencing and whole exome sequencing. And of course, this means that in some parts of the protein where there's variants or pathogenic variants, you will miss these depending on the technique that you use. So for instance, in whole genome sequencing, you will reliably call also the variants that are intronic, whereas this is, of course, not always the case for whole exome sequencing. Now, of course, many of the pitfalls of next-generation sequencing, and especially whole exome sequencing, have been progressively improved with improving of the capture kits. But I still think it's important to realize that there's many pitfalls. So if, for instance, what you need to know about exome sequencing is first the coverage. And usually the read depth is what is meant with coverage, is the number of reads that you have per nucleotide. And the more numbers of reads you have, the more increased the price will become. And so that's why it's almost always more simple to sequence a targeted region than to go for the entire exome in a very high depth. I think it's also interesting to realize that even with the newest whole exome sequencing target kits, that despite a good optimization, you still need a higher mean read to have a good coverage of your target exons. So I think that's also really important. If you get, for instance, one variant called in a panel, you really have to go back to that variant, to the nucleotide position, and really look at what, so the slides are moving on their own, I'm sorry. So you really have to go back to that nucleotide position and really make sure that it's well covered. So another big pitfall is, I think, really important. Besides the introns, exome sequencing is really bad at giving you information on structural variations. So structural variations in your gene, which are, by the way, very frequent and also mostly associated with where the positions of interest are, because these are highly mutated regions often. So we don't get a lot of info by performing whole exome sequencing. And in fact, That's what the next speaker will be talking about. More than 15% of our genome is in fact inaccessible to routine NGS because of the repeat content or because of high GC content. And this involves, so for instance, also regions of segmental duplications where many genes of interest for inborn errors of immunity are in fact located. And I think it's also important to realize that pseudogenes make interpretation difficult and that several genes are still not well covered by any capture or by any target panel. And I think NCF1 is definitely one of the more difficult ones, and ELANA as well. So there are several genes that are really important that we are not, well, that are difficult to sequence with NGS. I think probably one of the next speakers will also discuss this. Whole genome sequencing is still more powerful than whole exome sequencing for detecting potential disease-causing variants. And I think this is a nice slide where some patients were in a straightforward way compared, and the variants that were called by whole genome sequencing versus whole exome sequencing were compared. And in fact, what was interesting is that the variants that were called only by whole genome sequencing, cannot even touch the slide, so whole genome sequencing, these were actually mostly true and correct variants, whereas the ones called by the exome sequencing were mostly wrong. So they are mostly false. So I think that is really gives us a lot of insight that indeed whole genome sequencing can be more routinely or better used for CNV detection and detection of structural variations. And also interesting, the mean read depth needed for whole genome sequencing to have the same coverage is much lower for whole genome than for whole exome sequencing. So that gives us an idea of how good we can cover gene fragments with whole genome sequencing. So whatever you do, you still end up with a huge number of variants at the end of your pipeline, and that is in fact the real trick once you have those variants, to decipher the one that is really the one of interest for your patients. And so for whole exome sequencing, by and large, you get 20,000 variants. And then you really need to start thinking what you need to find and what is important for your patients. So usually for research matters, and also, by the way, for clinical diagnostics, there's usually a cutoff for high-frequency alleles where it's supposed to be, or thought that these are benign. And that is where the big difference comes into play with a clinical geneticist or a clinician who knows let's say, the pitfalls, is that you don't exclude all common variants. Because, for instance, CFTR variants can be very common, and you would just exclude them by filtering with a simple mean allele frequency of less than 1%. And I think we've also recently learned some nice recent publications also on IFNAR1 deficiency, where you see that actually a common variant can actually be disease-causing. So you cannot just blindly filter out the common variants. You have to do it in an intelligent way. Of course, the functional effect of the variant, there are some prediction scores that you all know, the CAT score, the polyphen, the SIFT, etc., many more. There's also some intronic variant prediction scores, but mostly all the variant scores are focused on missense variants rather than all the structural or intronic variants. You can also look at the conserved, how conserved a given nucleotide position is by the GERB score, or there's also other scores that you can use. And I think that gives you really some idea about how important this nucleotide is for the protein structure and for the ultimate function, because it's really very strongly related to the genomic evolutionary profile of that nucleotide. And then some nice tools that you can use if you want to see it visually is PopVis, which I will show you, and also the blacklist of variants. And in fact, these two tools are quite handy in reducing what we would say the non-pathogenic variants in your group of variants that you find, because these are in fact variants that are rare in common population databases, but that are common in your own set of data patients or of data exomes, or if you're in your own, let's say, country or population even. And so you can see that most of these blacklist variants, they can be just excluded, and they are usually in exactly these areas that are difficult to read, and they're in fact wrong, right? They're in fact mostly not relevant. And this is a nice tool, I think. So here is my favorite gene, ADA2, with one of the most frequent mutations in the northern population, R169Q, and you can in fact display here the CAT score, so the deleteriousness score with the mean allele frequency, and you can see where your gene or where your variant pops up. And in fact, the higher they are here and the more to the left, the more interesting for research or whatever you want to do with it. Of course, it also, looking at a variant and a gene also means that you look for the protein function. Of course, we don't know all the protein functions yet, and sometimes it becomes a real endeavor and trajectory to find the gene or the function of the protein. And I think useful tools here that we often use are just the protein function analysis or human gene connectome, where you can find if there is a link with a gene that is already known to be involved in primary immunodeficiency, because it can help you in deciphering the pathophysiology. And of course, you need to dive into literature and see what is really important. And then, of course, if it's relevant, if the expression profile is relevant, of course, it's also very important, because you can't end up with a gene or a protein that has a highly pathogenic or suspected I don't know what is happening here with my slides, but so it could be irrelevant to pathology if it's not expressed, for instance, in leukocytes or whatever. So you have to be quite careful in looking at what is really going on. And then I want to just call in this. Also, the gene level approach is quite interesting if you know. I don't know what to do with this. What is happening? My slides are passing all the time, moving. So, okay. So I just want to stress another thing, the gene damage index and residual variation intolerance score gives you an idea of how good a gene can cope with variants. And that is also interesting, because if you have a highly variable region or a highly variable gene, it's quite unlikely that one given variant in that gene will be the one that you're looking for. It can be the one, but I mean, the odds are against you at that point. So I think it's always good if you have all these tools in your annotation file, so that you can have a good overview of all these tools. And then something that has been recently reported is in fact the scones or the cone score, which gives you an idea of how strongly negative selection has worked on a particular gene. And in fact, negative selection is more strongly acting in an autosomal dominant inheritance fashion versus the autosomal recessive ones, and also has more negative selection involved in haploinsufficiency mechanism than dominant negative. And also, if you look at loss of function versus gain, there's higher negative selection on the positions that result in a loss of function. And so you can use this score in another way by looking into how negative, for instance, the score is, can give you an idea of how important this gene is, or this position, in fact, can be for our functioning and for the functioning of the cell in itself, because there would be a very strong negative selection on that position or on mutations. So, of course, the combined approach is quite interesting, and this is in fact, if you combine CAT score with the mutation significance index, then you have an idea of how well a gene tolerates a given change in the genome. So, and that's mostly what we use in the lab to dig into exomes. And then the question of unbiased, because in the beginning of next generation sequencing, there was the idea that this is really unbiased, looking for gene defects, but in fact, if you go unbiased, you can end up with really with bad outcomes. So you have to do it in an intelligent way and look into different modes of inheritance. Don't exclude autosomal dominant or de novo, of course, in a consanguineous population, for instance. Look at the clinical penetrance, because it's not straightforward, as you know. There is a lot of incomplete penetrance, especially in the autosomal dominant setting. with loss of function or dominant negative, etc. And there's a lot of genetic heterogeneity. And so what we know from literature from single patients can be a really different phenotype than the one you are looking at. So it can be really misleading also to be informed. So you have to go blind and also go in an intelligent way through your data. And then of course the ideal setting would be the multiple affected individuals in one kindred, but of course we all know that we usually have the single patient where we have to look and to look and to look and to look further. So, sorry, I went back. So this is the ideal world, but we all know that this is just a pragmatic scheme that can help you a lot, but then it still comes down to filtering, to reading into literature, and to dig further to find the gene that is of interest, or the position that is mutated and that causes the disease. So here I give some overview about, I think, panel diagnostics versus exome, and in fact what we used to call a gene chip diagnosis for inborn errors of immunity versus whole genome sequencing, which is here. And of course one thing we haven't touched upon, because it's also not really the topic of today, is the risk of incidental findings that goes up, of course, from panel sequencing all the way to whole genome sequencing. And I think it's important, we still don't have a good way to manage these incidental findings, where we would find, for instance, a tumor-related gene or a gene predictive of, I don't know what, Huntington disease, etc., if you're just looking for an inborn error of immunity. And there's some good guidelines out there, but they haven't been implemented on a wide scale. So I think that's also something to think about. And I think especially in research, there's still not so much interest for this important aspect of genome sequencing and exome sequencing. One thing that I probably won't not touch on too much, because I think Alexander is going to go quite deep into it, is the defect of copy number variation. So if you're looking into inborn errors of immunity, you need to realize that several genes are really carrying many deletions and duplications, and that these can be the cause of the disease. And that in fact with whole exome sequencing it's very difficult to find these unless you get a very big database in-house and go then with these tools through them. I think one of the ones that we use most is actually Exome Depth, but there's many more that are being described. And it becomes intriguing, it becomes increasingly more easy to do it, but still the filtering of small deletions is still not very easy. So that is where microarrays and array CGH and MLPA come into play, where we can use these to either detect CNVs or to confirm them and to really look for instance, DOC8 is one of those genes, GATA2 is one of those genes where it's really important to dive deeper and to look into structural variations. So I think this is a nice slide that gives you an overview of the size differences of the different techniques, how much you can cover with what technique, and how big the stretch is from one base pair to 100 megabases is really important. And you see the sizes of the standard genes and exons that you can sequence. So you see that there's still an entire gap here to be able to sequence well. And so I'm reaching the end of my slide set. I think one of the questions that came into play next is, because of all the variants that you end up with, and in the ideal situation that never happens and only is in the textbooks and in the nice articles, how to go deeper and to confirm what you have. And sometimes, if you don't have a good answer, do you use other NGS tools, for instance, RNA-seq, or do you go from whole exome to whole genome sequencing? And there's been a few papers that have been studying specifically if there's an additional value in going from WES to RNA-seq. And in fact, usually they can add a few diagnoses, but it's mostly in the range of 7 to 10% that you get extra. So it's not really so impressive in the papers that have been published. And it also depends, of course, on the phenotype that you're looking at. So if you look into very severe phenotypes, or for instance, in neurodevelopmental phenotypes, you have much more chance of having additional findings than when you look into a broad view of patients with inborn error of immunity. And then one big step, and I want to highlight this paper, and I want also to highlight the crucial final step, is that a published variant does not equal pathogenic mutation. I've really stepped into this in the past a few times, finding some variant, for instance, in STAT1, you think, okay, it's going to be a STAT1 gain of function mutation, but in fact, if you, and Anne has gone through this journey with me, that we actually were able to show that this variant that had been published to be pathogenic was not pathogenic. And I think it's really important to validate well, because you're going to deliver a diagnosis to a patient. It's really important. And so you have to go through these, for the individual patient, individual variant, you have to go through these steps, ideally. Rescue of the phenotype is a huge work. It's not easy. It's not done in one day, week, month, usually. So, but it's important, and especially if it's going to be published, I think it's important to do the best you can to have the data right. And then I would say, diagnosis in practice. If we are honest, depending on the severity of the phenotypes we include in our NGS, we end up, in the best papers I found, it was up to 60%. But these were, like, for instance, really severe combined immunodeficiency phenotypes. But if you go down, for instance, to antibody deficiency, you go down to 20–25% at most. Some people say no, some people say yes. But I think that's what I found in literature. And so you can push a little bit further with whole genome or RNA-seq, but it's still very difficult. And also I would like to highlight this, multigenic mechanisms. Reviewers these days, if we go to research, they like to ask, but are there not other genes involved? Well, probably, you know. I think that's a fair question and a fair statement, and something that we cannot refute, or that we cannot really say anything against, because that's how our body works. There's epigenetics, undoubtedly. There's so much we don't look for. Probably there's going to be more somatic mutations discovered that underlie inborn errors of immunity, and that's why a good sequencing depth is so important, because otherwise you would just miss it. And I'm more and more wondering if we, for diagnosis, we can design really pathway-driven analysis. But honestly, it's going to be really difficult to have one-size-fits-all diagnostic strategy for PID. And also, I would say, how can you just at one point say that one variant is not important? Because you would have to start validating the non-pathogenicity of a variant, because some variants really look pathogenic. So how far do you go in diagnostics? And I want to highlight again this, published does not equal pathogenic. So go into the paper, don't stop in the abstract, but see how well it was done, and be serious about delivering a diagnosis. And I don't have to say this for clinical geneticists, of course, but I think sometimes we are too eager to give a diagnosis when we don't have the data yet. And I think that's the end of my introduction. And I think, yeah, we need coffee breaks, because I do think this is a field where a lot of collaboration is needed. Sometimes to our own, let's say, I don't know how to say that. I think collaborating is very important, even if we lose our own benefits. Voila, the patient first. That's it.
Thank you very much, Isabel. Very nice and inspirational talk. Really something to start with, because lots of subjects you mentioned will come back during the 2 days. I especially like the fact that you mentioned published does not mean real pathogenic, because that's something we have to take into account, really. Are there any questions from the audience?
Okay, thank you. Nice presentation, Isabel. My question is, how do you deal with the reanalysis of NGS data? Because, as you said, the performance of whole exome sequencing on other technologies do not reach 100%, obviously. And in your experience, do you have any specific protocol to do a periodic reanalysis, or or some kind of automatic reanalysis that will help to identify new published variants, or?
No, that's the sad thing about it, because nowadays most of the clinical diagnostics lab do perform exome sequencing and use that technology or platform, and then do only a panel analysis on it. So they would be able to go back every, I don't know, 5 years seems like a reasonable term, or maybe sooner for some severe conditions. But mostly what we have done is we have taken the exomes into the clinic after consenting the patient into the research lab, and we've gone through it ourselves. And then it's of course important to really keep track of the captures, of the sets that have been used, and so on. But yeah, I think we have been able to find some diagnosis in a reanalysis of what was delivered. In our own research exomes, we haven't been successful in getting diagnostics after reanalysis. But taking them from the clinical diagnostics to the research lab, yes, quite often actually.
Any other questions?
Yeah, if I may. Yeah, that was wonderful and a very nice introduction. There's 2 points that are a little bit, at least in first sight, a little bit conflicting. On the one side, you say that for the latest ACID database, there's a significant amount of new genes identified based on a single case. On the other hand, you say, clearly, and I fully agree, that there's sometimes bits and pieces in literature that cannot be replicated. Obviously, we all have, so I'm not criticizing the single patient studies. If there's proper functional evidence for it, you mentioned it clearly, that read the paper carefully, how well it's done. But I think a little warning is important, that an N is 1. I mean, we tend to believe what we see and we will want to see. So even with some functional evidence, an N is 1 remains a little bit risky. So yeah, I think we as a community need to step up to.
We have to be open-minded
exactly.
about it. And
exactly. I'm fully on your side. This was not a critique, but it's just the flaw in the system that there's too much data out there. We all know Hans-Hilger Ropers, who once published 50 ID genes, and I think by now 30 of those are refuted. So, I mean, it is dangerous if there's only one finding a single case. So I think we really, as a community, have to step up to find more of these rare cases.
And by the way, it also asks for an open mind to look at the phenotype, because it's not because one case or one patient has this phenotype that the next patient you find with this mutation or gene mutated is going to have the same phenotype. Absolutely. And by the way, it's not always easy to get that published, by the way.
Anyway, absolutely that's right. Another topic. I think there's probably for all of the genes that we're currently working, there will be allelic series. So you will have loss of function, gain of function, and a variety of domain-specific mutational effects. I mean, I think we don't have enough numbers for all our rare diseases. No, not enough genes for all the rare diseases. So
we're not there yet. I think that's the conclusion.
Yeah, that's a good point. We won't get bored anytime soon.
Okay, there's time for one quick question still. And
So it's very basic. So Isabel, in the IOIS table, so do we see an increase of discovery of gene with gain of function mutation, and or genes involved in autoinflammatory, autoimmune disorders?
So actually, we see now many more genes that are in the autoinflammatory region and innate immunity, and I think that has to do with COVID and gene discovery there. But it seems like the link between those two areas is really booming, and I think that's because the more severe phenotypes, I think, I'm not saying we have them all, but I think, yeah, we are getting there. It seems like now we are going deeper into the quote-unquote less severe phenotypes, because I wouldn't call them less severe, but it's probably what's coming next. But it can also be the trend just now. Maybe, I don't know what's going to happen next. I think there's also still many SCID genes, by the way, to be unraveled. We don't find our diagnosis for SCID babies in, I don't know what the percentage is, but I think in a recent review, I thought we were only at 30% of finding SCID phenotypes, but you have to pick up the patient soon enough to get all the samples that you need. Yeah.
Thank you.
And what I forgot is but they will be, they can get questions next time. For all the online speakers, they can put questions in the chat. And I did not put any attention to that. So, but first we go on to the next speaker. So for the next speaker, I will give some time for the chat as well. I apologize. The next speaker is Alexander. Alexander is the first. Yeah.
Oh, I thought I was the third one.
So.
I thought there's a genome sequencing topic.
You know.
Yeah.
You changed it. Okay, yeah, perfect.
Okay, so the next. So
I thought you wanted to build up from exomes to genomes to long-read genomes.
Yeah.
That's what I thought.
We changed a bit, yeah.
No
anyway, it will give me a time for some good introduction. Alexander Horschen, lots of us already know him. He's an expert from, a genome expert from Nijmegen, from the Radboud University. And he's very experienced in various novel genome technologies. He's really has profound knowledge in that part, and he's also involved in SolveRD projects, where he's one of the leaders, the PIs in solving the rare disease patients, not only immunodeficiency, but with all kinds of rare disease patients from there. You can have the floor.
Thank you so much for the kind words, and thanks for the kind invitation. It's wonderful to be in Paris, even more wonderful to be at Imagine, very important to everybody who's into rare diseases, obviously. And indeed, I'll try to share some of our experience of particular the last 2 to 3 years maybe, generally speaking about long-read technologies. And Isabel just gave a wonderful introduction to some of the caveats that we still face. And I can't promise you yet that we solve all of those issues, but at least certain aspects, in particular the CNVs and the structure variants, I believe we can solve, at least to some extent, by these latest technologies, and I'll try to take you along on that. So for full disclosure, if I may sound excited about the technologies of BioNano and PacBio, I hope you believe me at the end that this is not only because they paid some of our reagents. I should also declare that I'm not a medical doctor, nor am I an expert on immunology, and certainly not on rare immune diseases like inborn errors of immunity or PIDs. I tried to get there, but you are all much more expert there. But I hope that with my genomics insights, I can help you a little bit. And I won't talk really about the immune diseases particularly, because these latest technologies we've mainly now tried and experienced on other diseases, but hopefully some of them are still good illustrations. Just to have one slide on how we tackle some of the immune diseases. Generally in Nijmegen, we collaborate closely with our colleagues in Maastricht, and together we sequence a lot of exomes for a lot of rare diseases. So up until now, I think we've sequenced more than 70,000 exomes for rare diseases. That covers a variety of 25 heterogeneous or so disorders. And last year alone, it was roughly 12,000 exomes done for these variety of rare diseases. And amongst those, there's usually a few hundred rare immune patients presenting with what we suppose to be inherited immune diseases, inborn errors of immunity, PIDs. And in total, we now reached close to 1,400 patients since we started this, and all of that is done diagnostically. And I just wanted to show you how we usually tackle that. We have a monthly exome sign-out meeting with all the experts around the table, luckily with a lot of immunologists. This is really spearheaded and organized by my dear colleague Annette Simons, who's really an expert on diagnostics and a diagnostic staff member of our department. What we usually do, and that refers to what Isabel also just explained, we usually perform a 3-step approach, and our patients have the right, after being well informed, to opt in and opt out to these steps. So usually we always sequence the exome and then analyze in silico the panel of genes that we know more about, and in our case that's obviously then a PID panel. At the bottom there is a link if you're interested. That panel is updated once per quarter usually and then shared. With the latest panel, it was close to 420 genes that we analyze. If we don't find anything in there, in the patient consents, we do an exome-wide analysis, properly in the diagnostic setting, so really analyzed by my colleagues and then discussed in this meeting. But for those patients where we get even broader consent, it's also my research group who jumps in and then looks again at the exome, so very similar to the Leuven setup. And in the Dutch system, this is called academic medicine. And I think it's a wonderful thing because our patients really benefit from that. It's really on the borderline between proper clinic and diagnostics and research, and we really all inspire each other with our insights, and that at the end will be good to the patients. for very specific patients, we also have something special that we call the rapid clinical exomes. So for very urgent cases, and occasionally that really helps out, in particular for these kind of diseases, we can offer the test and give back results. So after retrieving bloods, we give results within 7 days. If you're interested in more details, especially of our diagnostic PID exomes, my former PhD student wrote an overview of the first 250 of those cases. My recent PhD student wrote a little bit of a review recently where we explained quite nicely the challenges that Isabella also clearly placed out here. And just for full honesty, I think our current field is similarly, let's say, disappointing, especially if you come from other rare diseases. You sometimes feel, oh, we should be somewhat better here. And we're not yet. So it's roughly speaking 20–25%. And if you then look at the data again and research, at best we probably have 10% or so where we have very good candidates that are worth following up. Maybe we sometimes follow up some other ones, but that is roughly what we're working with here. And occasionally, even in single cases or in 1 or 2 families, we're lucky that everything falls together and you just, that just exome sequencing itself works. A hypothesis is correct, and then you find very few cases with deficiencies, as we've done for the TLR7 deficiency in the first wave of COVID. But, as already mentioned, we may miss a lot in exomes. And unfortunately, we also still seem to miss quite a lot in the standard short-read genomes. And there's an entire list of things, especially as a geneticist. I think we should consider that some diseases may not be genetic at all at the end. We may tend to forget about that. And the more we've already studied in genetics in a certain patient, the more likely it may be that it's not a genetic disease. And I'm still convinced that a lot of the diseases we are currently studying are genetic, but we'll only see in a few years from now. But my personal bet is that there's a lot of structure variants and CNVs and other types of structure variants that we missed so far. So in essence, the question that I try to address today is, why is it actually that after 12 years of NGS, we still need to do cytogenetics and use cytogenetics tools, even karyotyping, FISH, microarrays? Why is that? Why isn't NGS better at that? And in detail, and that will be the first part of my talk, I think here's the 3 top reasons why I think this hasn't happened. A lot of CNVs and SVs happen in very difficult regions, usually mediated by repeats, such as segmental duplications. I think the analysis of genome data for structure variants is still very complex and very sequence-centered, usually run in bioinformatics labs that may not have real proper cytogenetics background. So cytogenetics really have difficulties in understanding genome data. And then finally, maybe, especially in the acquired structure variants, I think coverage is very important. And we can't barely afford a genome of 30x coverage, and ideally we would go much deeper. And then the second question is, how much of the structure variation in our genome did we really miss so far? And I'll try to answer that with a real long-read sequencing that I'll show you in the second half of my talk. So can we be more comprehensive if we do long-read sequencing? And can we ideally even find not only the structure variants, but even also the smaller ones all in one go? I think that's the ultimate test that we all in human genetics already wait for. And finally, the question is, of course, can this help rare disease research at all? So before diving into that, a little genomics quiz. And the winner will get 1 extra glass of red wine during the dinner. How many structure variants does each of us carry if you compare our genome to the reference? Shout. 200. Okay. Who says more than 200? 100,000. 100,000. All right. Who says more or less than 100,000? A few thousand. Yeah, that's a bit vague. I'm not sure whether you're not exactly off, but I'm not sure whether you deserve a red wine for that. I think some of the data I'll show you and that many others have produced, my guess best at the moment, if you define it as being larger than 20 base pairs, is somewhere in the range of 50,000. And then maybe even the more interesting question, if you compare that to the footprint that substitutions have, so SNPs in the genome, that's roughly 3 to 4 million. So 3 to 4 million base pairs are affected by single nucleotide variants. How many megabases may be affected by structural variants? Actually, there my count is also off, so I don't even know so much. It certainly is factor 3 or 4 more than the single nucleotide variant, so it's certainly more than 10 megabases. I guess once we're more complete, it may be more in the range of 15 megabases or so of sequence that are affected. So that intrinsically would already tell you, maybe we shouldn't forget about those, and maybe we underestimate the importance of structural variants in the genome quite dramatically. So it's actually quite a surprise that already by mainly looking at point mutations, we already solved so many cases. So and to really tackle that, I'll give you brief vignettes of two technologies. One is what is now known as optical genome mapping. I usually refer to that as next generation cytogenetics. It has nothing to do with sequencing. And I'll give you little examples of that, and then I'll finish with real sequencing, the long-read sequencing, and only one of the technologies that is on the market we've a lot of experience with, that is PEG-Bio hi-fi sequencing. So many, many years ago, I was originally trained in a cytogenetics laboratory, but I always had real issues recognizing chromosomes. I really was not good at it, because it is a proper art to recognize chromosomes and even recognize that this one band of chromosome 1 may be deleted. So you really need terrific skills to do so. But now we can do that. Even I can now do cytogenetics because we use BioNano. And just imagine for a second, you could zoom into this chromosome, and where you usually have one band in GTG-banded chromosomes, you now have thousands of bands. And you have all those bands, not 500 genome wide, but 500,000 genome wide. And you can measure the distance much more reliably between these bands, or labels as they are now. That would deliver you something that is karyotyping with 10,000-fold increased resolution. And still it keeps a lot of what karyotyping can do for you. It still is a genome-wide analysis. It still gives you positional information. And in essence, you're still looking at one chromosome at a time, or at least one long DNA molecule at a time. So there is a chance for single molecule resolution. How does this technology now really work? We unfortunately can't use DNA that we have readily in the lab, simply because DNA that we use for any kind of sequencing is too short. So we need to go back and need cells, or at least nuclei, and isolate DNA extremely carefully to maintain very intact DNA molecules. Then we label the DNA, and BioNano will have a little workshop later and give you more details, but we label the DNA on a 6-mer DNA motif just with a fluorescent label. We then take these long individual DNA molecules up to 2 megabases in size. Current average size is 270 kilobases, so very, very long. We put them into so-called nanochannels and take pictures. So each picture pictures one long DNA stretch with all these labels on it. And if you do that over and over again, usually with 100-fold or a few hundred-fold coverage of the human genome, we can perform a de novo assembly. Without knowing the sequence, just based on these labeled molecules that are digitalized, we can do a de novo assembly. And we can then use this de novo assembly and compare it to the reference genome. So the reference is shown here in green. You can't see my mouse, let's see. Here is the reference, and this is our sample. And if the reference, the expected label pattern in the reference is different from what you see in your sample, you may have a structural variant. And the good news is we cannot only look at the very obvious deletions and duplications or gains and losses, but we can even look at more complex forms of structural variants. But also, importantly, what arrays never could deliver, also all the balanced events are visible, and I think they're very important. Translocations, inversions, for example. I'll do on the next slide as a first BioNano result. And here you see a classical result from BioNano. So we have here a nice ring of ideograms, so that's chromosome 1 till the X and the Y chromosome. And then in the middle pattern, you see in this case, all the rare structure variants that are called in a genome. So in different colors, we have deletions, insertions, inversions, and duplications. We then, more or less for free, have a coverage-based track that also gives us the CNVs. And then in the middle, you would see translocations. As we only explain here the rare events, there is no translocation in this case. But if you look carefully and check the copy number, this was a man with azoospermia and had a Klinefelter karyotype, so 47XXY. So that is very easy to spot also in this technology. But more intriguingly, we said, okay, this is really enabling cytogenetics at a much better scale. So we teamed up with several groups here in France, and this consortium was led by the wonderful Leila El-Khattabi here from Paris. She works at Cochin. And what we just did is we just went back to samples that were positive in the cytogenetics laboratory. So I think it's fair to say this is a routine workflow or routine set of samples that come to a cytogenetics laboratory that included full aneuploidies, small and big deletions, duplications, translocations, inversions, all of that, several very complex aberrations. And I won't bore you with all the details. The good news is all the known clinically relevant aberrations were found in all samples. And just to give you one example, here was a translocation from karyotyping. It was known that there's a likely de novo balanced event. Then we do Bionano, and we can clearly see that we have the de novo assembly map of our patient that in part maps completely to chromosome 9, but the second half maps very nicely and very consistently to chromosome 17. And here the two purple lines distinguish the breakpoint. So we don't get breakpoint resolution in base pairs by sequencing, but we are getting very close, a few kb closer to the breakpoint. And the beauty is we can then go back to the original molecules, click on this translocation map, and we can see how many molecules support an event, and therefore we know whether it's more likely a true event if there's enough molecules, whether it's full-blown in germline, or whether it may be a mosaic event. If you're interested in the details, we published that last summer, and I think it really is a tool that may enable cytogenetics at a completely different level than we did so far. When we did this, we also thought, oh wow, if this works for constitutional cytogenetic aberrations, maybe it's even more useful for hematological malignancies with acquired structure variants, or leukemias. So there we just in our center took 52 samples that had clinically important relevant findings, looked, oh, I have the same issue now. Yeah. All those were studied in our diagnostic laboratory, usually by a combination of karyotyping, FISH, and CNV microarrays. And also here we see 100% concordance for all the clinically relevant findings once they're at 10% variant allele fraction or higher. And again, also just a nice small example. Here we had a small microdeletion of the TET2 gene originally found by a CNV microarray. And then if you look at the Bionano data, this is even an event called by both algorithms that are provided, the CNV tool, but also the de novo assembly-based tool that is what they call the structure variant caller, usually much more accurate, and both tools directly give us the exact breakpoint of this deletion, really deleting the entire TET2 gene. If you talk about leukemia, translocations are obviously even more important. In this series, there were 3 cases of CMLs, all had the Philadelphia chromosome, and very easily we spotted the Philadelphia chromosome, so the chromosome 922 translocation that is so important, creating the BCA-ABL fusion gene. And sometimes in leukemia we find very, very complex cases. If you wish, it's ethically not exactly correct, but they're genomic explosions sometimes. So really catastrophic events. Here's just one example where already the array, this is figure panel C, already showed that there's a very complex pattern on chromosome 8. If you now look at the circus plot, you see a lot of intrachromosomal rearrangements. And if you zoom in, this is the bottom plot, we now have something that is very similar to the CNV profile of the microarray. But the little blue maps, you don't need to see all the details, are fusion molecules that happen. So at least in theory, we are now able to reconstruct the entire reshattered chromosome to the new chromosome. So we can really start tracing that. With that in mind, we had 100% concordance, as I said. I would dare to say this technology delivers a low false positive rate, and very often we are much closer to the true underlying biology of the genome rearrangements. There's 5 or so other studies that have come out since then and all show very similar data. And I predict that especially for hematological malignancies, we will find a lot of new actionable and important fusion genes and translocations that were missed by the classical means. Even my colleagues in diagnostic are so enthusiastic that they now start a prospective clinical utility study to push it into clinic. In Leuven, they are one step ahead. They already use it diagnostically in the clinic, so that is really wonderful. And just finishing this part and then going to the last few minutes of PAC-Bio sequencing, just something on rare diseases where this technology may help. So together with my former colleague Roland Kuiper, we studied a family that presented with a really severe, very early onset childhood disease, atypical teratoid-rhabdoid tumors. So Roland and Marjolein and others studied a family, a Dutch family, that already had lost 2 children right after pregnancy or late in pregnancy due to this very severe brain tumor. And usually the cause for these brain tumors is known. 2/3 are pure somatic mutations in the brain tissue, but in 1/3 of cases a germline mutation is strongly predisposing and only the second allele mutates somatically in the brain. In this family, because there were 2 affected already, it was suspected to have a germline mutation. So all the workup was done in the clinic, in diagnostics, Sanger sequencing, MLPA, even whole exome sequencing, no mutation found. Then Roland went on, did genome sequencing. Wow, who's calling there? Sorry for that. Genome sequencing was on, again, mutation not found. Luckily, even while the children were already deceased, Roland saved an EBV cell line, sent us a cell palette, and we did optical genome mapping. What we standard find in filtering is roughly 7,000 structure variants. So not all the 50,000 that should be present in each and every genome, but 7,000 that are larger than 500 bases. And with very simple filtering, we filter just for the rare events. Genome-wide, for such a rare disease case, it's usually in the range of 30 or 40, 35 in this particular case. And maybe not a surprise, one of the structure variants was at the SMARCB1 locus. Here you would usually expect a wild-type marker distance in the reference of closely to 5 kb. But in our patient, there was 3 kb or 2.8 kb of extra material inserted. So very likely, this is the underlying germline mutation that we were hunting for by all the other technologies, but that remained unsolved so far. With that, just summarizing the optical genome mapping part and then a brief part on PEG-Bio. So I think for optical genome mapping really may offer a great tool to replace all of cytogenetics in the future. It also, we did some tests to use it for repeat expansion disorders. Also there it's a very accurate but indirect test. And this last example hopefully shows you that we can find structure variants that escape all the standard tools so far simply because the genome may be too complex and too repeat rich at some of those regions. So I think it really starts next generation cytogenetics, and I'm quite excited about that. And there's additional improvements to the technique that should really allow many more users to use this. But now, for the last few minutes, I'll move into long-read sequencing that we do on instruments provided by PEG-Bio. So we have 5 or 7 of those sequences now in our facility. And what we do there is so-called, or what PEG-Bio now markets as so-called hi-fi sequencing. They will give you also a little workshop tomorrow, so I won't go into all details. What you have to remember is those are single molecule sequences of very long molecules, 20 kb in size, and they have a quality that by now is already better than Illumina sequences. So it's basically sequencing very accurately, but now with 20 kb reads. So we can start using that for a lot of cool things, I think, and I'll show you more details on 8 trios that we've sequenced. What we do quite often is using severe intellectual disability as a model disease, and that we don't sequence the patient only, but patient-parent trios. We also did several individual research cases. A bigger project that Marielle kindly mentioned is Sol4D, where we now go for 500 unsolved rare disease cases that we sequence with a little bit lower coverage, mainly to find structure variants in their genomes. But then in parallel in-house, a lot of other uses come up. So for example, long amplicon sequencing for some of the genes that Isabel highlighted are so difficult by the standard means, and I have one slide on that. I think this potentially could completely kick out Sanger sequencing, which still exists in some centers, also in ours. For RNA sequencing, I think there's some cool applications. And we also get a lot of requests in our facility to use it for completely different technologies or for different species where there is no reference genome at all. So this is just one slide on the long amplicon sequencing. 2021, we alone sequenced close to 4,000 samples for long-read amplicons spanning 27 different genes. You may not know all of them, but you certainly know the HLA genes, very complicated to sequence. It's 5 different PCRs spanning the 5 most important HLA genes. It can span the entire mitochondrial DNA, but there's also IGH genes, or what is, for example, known as some of the opsin genes, very complicated, very homologous genes. And there's a few others that you may know. There is PMS2, very important in Lynch syndrome. And that nasty PMS2 gene is 16 kb in size, but 8 kb are completely homologous to another part, a pseudogene in the reference genome. So therefore, we can now span up to 16 kb is in our experience, where long-range PCR still is a good method to enrich. And the nice thing is we now see all the variants in that 60 kb fragment. We can clearly distinguish pseudogenes and other homologous sequences. And I think ultimately we can go quite deep and quite accurate with this technology. So also for mosaicism, if you're sure about a certain locus, I think long-range PCR, just for your one locus, may be very elegant. And we discussed it prior to the talk. Isabel said sometimes the doctors are just right. And we all know those cases. The doctor insists since 10 years, it must be this one gene. It's clearly this disease. It can't be anything else. I think there, just make a long-range PCR, and it's very affordable. For 150 euros or so, you have the entire locus sequenced by now. I think that's really incredible. Now going to the more genome sequencing that you're waiting for. So we use intellectual disability as a model disease because we can learn a lot from that, I think. And we know by now that de novo, so new mutations that only occur in father or mother, that are not present in mother or father, but only occur in this one oocyte or the one sperm cell that is used, or just very briefly after fertilization postzygotically. Those are important for severe disorders in general. But if you work with new technologies, the nasty bit is there's very few in a genome. And even if you only do a few mistakes in your new technology, there may be very often many more mistakes than true de novo mutations. So that's one reason why we like starting to sequence trios whenever we start with a new technology. That's what we did here. Could only afford 8 trios, but that's what we did. I think it's also important to keep in mind, because in some fields of human genetics, I think it's not so commonly done to do trio sequencing. I think it's really great to reduce complexity of the data. At least if your hypothesis that a de novo mutation is causing the disease is correct. It may not always be. You really need to look at the pedigree, at the disease to understand that. But if it does, if it's a de novo mutation, the wealth of information is much easier to digest because there is so few in a genome. And I think de novo mutations, and in particular de novo structure variants, will help us as a human genetics community to understand the regulatory genome and the known coding space much better than point mutations and inherited variants will do. So even if you're not studying intellectual disability, especially for severe and sporadic diseases, I think studying de novo mutations very systematically by trio sequencing is a very elegant approach. And even for the rare immune diseases, we now start doing that. So we've now done the first 120 trios systematically, where we only had one sporadic, severely affected child. And this is just out as a preprint if you want. So going back to our general cohort, if we do exome genome sequencing, everything we do, indeed, this is one of the papers where we also dare to cite originally 60% yield. I think in real practice, we are sometimes, even for intellectual disability, a little bit lower. But even for those where we really highly select cases do up to genome sequencing, there's still 40% that we don't solve. And 8 of those cases where everything else was done, yeah, perfect. We now did hi-fi genome sequencing with a main focus, 8 patient-parent trios to understand all de novo mutations. And on the next slide, you'll just see the results of all 8. It's very simple. There's a lot of variation in a genome. And I think this is much closer to the true underlying variation in a personal genome. So we find close to a little bit more than 4 million point mutations, SNVs. We find close to 1 million insertions, deletions. We find as being 1 to 20 bases in size. And we find a little bit more than 50,000 structure variants if we start counting at 20 bases. And now the important trick, if we have the parents, we can reduce the amount dramatically. And for point mutations, we end up with 84. And that number matches quite well what we know already from Illumina genome sequencing and other studies. So I think that's quite a realistic number. We roughly find 10 indels that are de novo candidates and 7 structure variants. But those are candidates, and validations are still in part ongoing. It's not so trivial to follow all of them up. And I think especially the last one may be quite a strong overestimation. I guess it will not be more than 2 in a single individual genome. And just to show you the data here, this is a de novo point mutation in one child. If you compare now the PEG-bio data, you very nicely see that this is clearly a heterozygous call, nicely in cis with the homozygous inherited variant. But if you look back at the same patient and the same patient's data in Illumina genomes, yeah, there's some reads also supporting the variant, but a lot of these white reads that just not map very well, very ambiguously mapping reads. So it's a complex region with more repeats. Hence, Illumina cannot find that variant. And if you do that systematically, I won't bore you with all details, but what this comes down to, if you now compare 8 patient-parent trios systematically, short versus long reads, we see a huge concordance for the long read data. So even for point mutation, long reads are now already better, is the essence of that. Because if you go for some of the long read only de novo point mutations, a lot of them are true. And if you go for those point mutations that long reads can't find, but short reads find, usually the vast majority of those are not true variants. So I think it's fair to say that, and that's quite a surprise, that long-read hi-fi sequencing is already better now than Illumina sequencing, even for the small events. One other advantage is that we can phase variants, so we even have higher sensitivity for somatic events. Here, for example, we have an 11% variant allele fraction, clearly showing that a third allele is popping up in the child, because we can now phase the alleles, so that's much more powerful. Let me see. Oh, yeah, and this is just the structure variant, 68 base pair de novo duplication, which is also called and validated, but additional validations are ongoing. Again, we can phase, so that's really nice and gives us extra insights. So just to sum up the 8 trios here, I think it's just fair, as I said, in essence, they are better than Illumina genome data, and they're much more complete on all variant levels, point mutations, indels, and also structural variants. The disappointing news, however, is in none of the 8 we found a disease-causing mutation. So we spent an awful lot of money, a lot of analysis time, and still it's a little bit disappointing that we don't find anything that is meaningful in the clinic. There's also a similar paper by Evan Eichler just out, and we were marginally involved in that. He shows exactly the same. De novo mutations can be now better called with these long-read technologies than short reads could ever do. And then finally, I'll just come back to this one case because I left you a little bit behind with this MARCB1 insertion. And the truth of the matter is, Roland was excited about this, tried several PCR techniques to find what is inserted, and that didn't succeed. So at the end, we also added PEG biosequencing, because there's no PCR involved, much easier, and just one single smart cell, just 8-fold coverage of the genome, helped us to understand what happened here. So the insertion for this one case is actually an SVAE retrotransposon that is inserted in intron 2 of this one case. We now know that that causes a defect on RNA level, unfortunately, and therefore the gene is disrupted. And I think these kind of events we really miss a lot, and there's more and more examples coming up in the literature showing that this is an important event in some of the rare diseases. This is published, and luckily we even helped the family to offer prenatal testing after all they've been through, and now they have a healthy baby born just a month ago. So this is really the end, final slide of my talk. I think OGM really opens the space for what I call next generation cytogenetics. I think these new, especially high coverage genomes, hi-fi genomes, are the most comprehensive genomes I've ever seen. And I think for some of the research cases, the combination of the two, or maybe OGM and short-read sequences, may already be a very good add-on to find some of the hidden variants. So in summary, the long-read revolution only started. We won't get bored anytime soon. I think it's still all so exciting. And I'm very grateful to be here, grateful to everyone involved, our French collaborators on Nano, a lot of people in our group, and my own group. So thank you so much for your attention, and I'm very happy to discuss now and later, of course. Thank you so much.
Well, thank you very much, Alex. Because of time reasons, there's time for one more question, and then we move on to the next speaker.
Thank you very much for your wonderful presentation. I have 3 questions, but that's too much, isn't it? Okay. So I will only ask the one about the PEG biosystem. Is it possible to sequence mRNA and study transcriptome? And wouldn't it be useful, especially in PID cases?
Absolutely, yes. Sorry, close my presentation too early. I'll just show you one slide. So yes, indeed, you can do mRNA sequencing. They call that isoform sequencing, because indeed, especially for shorter isoforms and transcripts, you can sequence the entire transcript from start to end. And we just started to do that. So I think it still is quite expensive. It doesn't give you great coverage. A single smart cell usually gives you, there we have one slide on that. So here we just used, here we used PBMCs of healthy donors and stimulated them in vitro with various stimuli. In this case, you see Staph aureus. And these isoforms expressed only after certain stimulations that are not seen in normal PBMCs. And while standard mRNA sequencing can find that, you need a lot of bioinformatic tools to find it. And here you just read the entire transcript, so you're much more reliable in finding new isoforms or not so well annotated isoforms. And the ground truth is, not only for blood cells and immunodeficiencies, almost every tissue that has now been sequenced with IsoSeq shows that the isoform landscape is much more complex than we ever thought. So I think the genome browsers we all use, they're only showing us a tiny fraction of reality. Probably there's much more complexity also on that level. And indeed, long reads, this one and also ONT will certainly help us there dramatically.
Okay, thank you. I think tomorrow there's a PacBio talk, so you can ask all your questions there, and he's also around. So, and do discuss during the lunch times.
Thank you so much. It was fantastic. So our next speaker is Aurélie Cobat. Aurélie is a researcher from the lab, and she's going to talk from WES, so from whole exome sequencing to whole genome sequencing. So Aurélie, the floor is yours.
Yes, thank you very much, Anne, for this introduction. So as I said, I'm a researcher in the lab, but I'm a researcher in statistical and computational genetics. And the way I understood the title of my presentation was more from whole exome sequencing to whole genome enrichment. And the aim of my talk is to give you some introduction on rare variant association analysis. So I try to put it full screen, sorry. Okay, so the outline of my talk is first a brief introduction from the patient-based approach, which you have seen, I think, from the beginning of the morning, to the cohort-based approach, which is more the approach in which I have some expertise. Then the main method that we use to search for rare variant enrichment, and the underlying hypothesis. And I will finish with a new tool we have recently developed in the lab in order to test some alternative genetic hypotheses. So perhaps you have seen the aim of the patient-based approach is to identify in one or few cases with homogeneous phenotypes from one of a few family a reasonable number of promising candidate mutation with high impact that could underlie the phenotype under study. And in order to reduce the search space from all the variants that can be identified in a typical exome to a reasonable number of candidate mutations that can be further functionally validated, we will apply an optimal filtering strategy that is based on many criteria, both at the variant level and at the gene level, such as the mode of inheritance, the frequency of the variant in public databases, its predicted functional impact, the use, we can make use of deleteriousness scores. It will also make use of some gene filtering, focusing on functionally relevant or biologically relevant genes, and also makes use of some selection measure on the genes, and also it can make use of additional information such as linkage information if you had informative families. So it's not exhaustive. I'm pretty sure many other criteria that can be used. It has been very successful in the field of Mendelian and clinical genetics, and in particular in the field of inborn errors of immunity, leading to the identification of a large number of variants and genes underlying several phenotypes. However, there are still a number of challenges that remain, as you have seen this morning. And since the number of patients with homogeneous phenotypes that have now been collected and that have now been sequenced also, there is room to apply some alternative approaches, in particular cohort-based approaches, which can help to identify new disease-causing genes or new disease-causing variants, in particular, like new genes that would be like biologically unsuspected, or to account for incomplete penetrance, which can be difficult in the patient-based approach. So the unit of the cohort-based approach is pretty different. Then we need a cohort of cases, so typically at least several dozens of cases, ideally the large number of them, so the larger the better, and we will need a cohort of controls. Ideally also it will be an in-house cohort with homogeneous sequencing data and sequencing and processing of the sequencing data as the cases, but we can also adapt the methods to handle of external cohorts, in particular to make use of publicly available control cohorts. And here we will use statistical evidence to prioritize the genes and the variants that could be interesting candidates, that's showing an association with the phenotype, which is measured typically by an odds ratio, for quantified by an odds ratio and a p-value. Okay, so there are several levels at which we can perform those cohort-based analyses. We can look for enrichment at several levels. The first one, which is the simplest one, is at the allelic level. It's what we did in genome-wide association studies. We were just comparing the allelic frequency between cases and controls for all individual variants. However, this approach implies that there is substantial allelic homogeneity. That is, the same mutation underlie a substantial proportion of the cases. Otherwise, we have no power to detect any association. So in the context of rare variants, alternative methods have been proposed in order to overcome this issue, and we will, what has been proposed is to collapse, to aggregate all multiple rare variants into one genetic unit. And the first genetic unit, the most interesting one, is the gene, but it can be extended to any genetic unit of interest. And then what the underlying hypothesis is that there is a substantial number of patients that can carry different mutations, but at least those mutations are located in the same genetic unit, so in the same gene. So I think the most used, because it's easiest to implement, to understand, to interpret, is what we call the burden test, and in particular with binary collapsing. The idea is pretty simple. We will look at all the rare variants in the genetic unit. So you have here an example with 6 individuals, 3 cases and 3 controls, and a gene in which we have 4 rare variants and 3 different exons. With a number of variant alleles for each individual that is indicated in this matrix. So 1, meaning the patient here is heterozygous for the variant 1. 2 is homozygous for the variant allele at the second variant. 0, meaning that it's homozygous wild type, or homozygous reference. And so we will, for each individual, recreate a single variable that will be an indicator, a binary indicator, to say if the individual does carry at least one variant allele in the gene at any rare variants or not. So it's very simple. So for the first case, it does carry 1. Yes, it's heterozygous for the first one. The second one, it also carries at least 1. It's homozygous for this one and heterozygous for this one. In this method, the number of variants doesn't matter. It's really a yes or no. And you see, like for the controls, we have 2 controls that do not carry any variant alleles. When we have this, we are back to very simple case. So we have the phenotype, status, or case control, and a binary variable. I do carry at least 1 rare variant in the gene. I don't carry any rare variant in the gene. So we are back to the simple case when we can just draw a simple 2x2 contingency table, and we can test for association with very simple method. So it can be a chi-square test. Better is to use logistic regression, which will allow to account for covariates that can be, so it can either be important covariates that we know may modulate the risk of the disease, and taking them into account may increase the power of the analysis, such as like age or sex. But also importantly, using logistic regression is very useful to account for confounding factors, the main one in genetic analysis being population stratifications when cases and controls are ethnically diverse, and then we will simply put some principal components of the first principal component of a principal component analysis in the logistic regression model. Okay, so there are several factors that can affect the power of this analysis. The stronger the effect size, that is, the higher the penetrance for a given mutation and gene, the higher will be the power. Also, if you increase the sample size, you will increase the power of the study. There is also the level of genetic homogeneity. That is, the higher will be the proportion of cases carrying a disease-causing variant in the gene studied, the higher will be the power. And the last important factor is the proportion of non-disease-causing variants in the genetic unit that is in the gene that is analyzed will negatively affect the power. And you can imagine that if you add like among the rare variants, you have many synonymous variants that have absolutely no effect. They will be found with an equal probability, both in cases and in controls. So at the end, when we will collapse all this information, well, the proportion of cases and controls carrying at least one rare variance will be very similar, and so it dilutes completely the association signal. So it's why we need to apply some a priori variant selection algorithm, very similarly to what we did in patient-based approach. So based on several criteria, as before, the impact of the variant, the frequency, the inheritance model, also some deleteriousness scores. The idea is to try to maximize the proportion of disease-causing variants in the unit that is analyzed to increase the power of the study. But what you have to keep in mind is that when you do so, there is always a risk if you are too stringent to remove some disease-causing variants. And this will artificially decrease the level of genetic homogeneity and so can decrease the power of the analysis. So it's always a balance between the two, and it's why it's very useful to use several variant sets and so to repeat the analysis using different criteria for the variants, because the ability of the selection algorithm, I would say, will depend on the genetic architecture of the trait, which can change from one gene to another and which is unknown. Okay, so just one example of this gene burden test. So we applied it to the study of life-threatening COVID-19. So as you know, there is large inter-individual variability upon infection with SARS-CoV-2, with most of the individuals remaining asymptomatic or post-symptomatic, and a small proportion will developing life-threatening pneumonia and eventually die, around 1% before the vaccine era. And we know there are epidemiological and clinical risk factors that increase the risk of severe manifestation, like age being the most important, but also male are at increased risk. However, they do not explain the full variability. And at the beginning of the pandemic, we hypothesized that inborn errors of immunity could underlie critical COVID-19. So in order to test this hypothesis, our lab launched an international consortium, the COVID-Human Genetic Effort, and we collected large number of patients infected with SARS-CoV-2 with various manifestations of the disease and coming from all over the world. You have here the principal component analysis, and you can see you have here the Europeans, Africans, East Asian, and we have patients from pretty much all the continents with life-threatening COVID-19, but also patients with mild or asymptomatic diseases that are used as controls in the analysis. And we first hypothesized that maybe the fact that males are at increased risk of critical COVID-19 could be due to X-linked genetic factors. And so to test this hypothesis, we perform a gene burden test on all the genes on the X chromosome, comparing the proportion for each gene, the proportion of patients, of individuals carrying at least one predicted loss of function or missense with a frequency below 10⁻⁴ in the Genome ID database between more than 1,000 critical cases, male cases, and about 300 male controls. And the analysis was adjusted on the first 5 principal components to account for the ethnic heterogeneity. And you have here the result of the association. That is a typical representation with evidence of association expressed as minus log 10 of the p-value for each gene according to its genomic location on the X chromosome. And this bar represents the chromosome-wide significance threshold, accounting for the number of genes that were tested. And we did identify one significant gene, TLR7, with 21 carriers in cases versus 0 carrier in controls, and the p-value at 3.5 10⁻⁵. And further functional validation of the variants led us to the discovery that about 1% of males with critical COVID-19 had TLR7 deficiency. So this approach was very, I think, very successful, very useful, led to the gene burden analysis, led to the identification of TLR7, and it was consistently found associated in several other studies. Nevertheless, if we want to identify inborn errors of immunity that account for a lower proportion of cases, it may be challenging with those gene burden analysis, or it may require an increasing a very large sample. We may need to increase the number of cases, but also large is the number of controls. So this is why we need other tools to test for alternative hypotheses. And one very interesting is the hypothesis of physiological homogeneity in the context of genetic heterogeneity. What I mean is that we know that the genetic landscape of several diseases is characterized by genetic heterogeneity. So it can be at the variant, at the mutation level, but for this gene burden tests are pretty well designed, but also at the gene level. That is, in different patients, even with the same phenotype, it's a different mutation in the different gene that underlies the disease. However, what we do know also is that for many of them, this genetic heterogeneity underlines some kind of physiological homogeneity. That is, the genes belong to the same pathway. The products interact with each other. They have a common function. And so it may be much more powerful if we were able to test this hypothesis of physiological homogeneity. It may account for a higher number of cases, and so we may have much more power to do so. The easiest model the easiest method is just to extend the burden test, the unit of the burden test, from the gene to an a priori defined gene set or a pathway. And then we will collapse, aggregate all the rare variants, not at the gene level, but at the pathway level or at the gene set level. This can be very useful. However, it also depends strongly on the current knowledge and current annotation of the genes and current appropriate definition of the pathway or the gene set. And if we take the example, for example, of the cake pathway or reactome pathway, many of them are pretty large, and so there is a high probability, very high likelihood that most of the genes in the pathway are in fact non-disease causing. And so mutation in this will be neutral. And if we analyze those big pathways as a single unit, we will completely dilute the signal of association, and we won't be able to see anything, even if all the pathway regroup all the genes that are disease causing, because they are too large. And so it's why we, in the lab, develop a new tool that we call network-based heterogeneity clustering, with the idea to not using a priori defined pathway, but to be able to identify new cluster of genes that are highly correlated to each other and that are disease-specific. So this is the typical summary of the principle of the method. So it starts from a protein-protein interaction network and from a cohort of cases. So in this cohort of cases, we will apply variant filtration as before to identify the list of candidate mutation in candidate genes. So for each patient, we'll have a list of candidate genes that may harbor a disease-causing mutation. On this list of genes per cases, we will apply a gene clustering algorithm that I will describe a little bit after, based on the protein-protein interaction network. The idea is to identify in the data from the gene list of the patients all the cluster of genes that are connected and that harbor a candidate mutation in our cohort of cases. However, those clusters, they may be some may be disease-specific, so its mutation are really as what it cause the disease, but they can also, some clusters may be a cluster of genes that are just highly damaged in the general population. That they will, some of them, like the mucin genes, they will tend to cluster. They will be biologically connected, but we know they are highly damaged. So you may have a high number of cases with candidate mutations in mucin genes, and you will end up with those large clusters of genes. So in order to differentiate those highly damaged genes in the general population to disease-specific genes, we then perform a cluster-level enrichment analysis. So we make use of a control cohort, and we will compare for each cluster the proportion of cases carrying at least one candidate mutation in at least one of the genes of the cluster to in between cases, and controls, as we did before at the gene level. Then, on the significantly enriched gene clusters, we will apply some overrepresentation analysis in order to better biologically characterize those clusters, to see what the biological function of those. And so we will look for KEGG and reactome pathway and also Go terms that are enriched in the gene list characterizing each cluster. And we'll keep only clusters with a significant pathway or Go term enriched, which are the more like biologically relevant and also the clusters that the easiest to interpret. And of course, so then we will have candidate mutation in candidate genes that will need to be further functionally validated. So here is how it works, the gene clustering algorithm. So we'll start, so we have a list of candidate genes in several patients, and we will start from the first gene of the first patient. We will locate this gene in the network. For each, so the network is characterized by what we call nodes, that are the different genes, and edges in between the genes. And the edges, they have, the thickness of the edge is proportional to the confidence we have in the interaction. And so for the first gene, we will construct a subnetwork selecting only genes with an edge that is higher than the given cutoff to be sure we have a subnetwork of highly connected genes with a high confidence. So when we have located the first gene in the first patient, we will search in the remaining patients for the nearest neighbor in the subnetwork. And so here it's again the gene A in the patient 3. And then again, we will search in the remaining patients, so patient 2, 4, and 5, what is the nearest gene in the subnetwork to gene A. And it's gene B in patient 2, and so on. And the algorithm stops when either all cases have been visited, or in the remaining cases that have not been visited, there are no genes belonging to the subnetwork with a high confidence. And we will iteratively resume this algorithm, starting from each gene from each patient to identify all the possible cluster of genes. So this is what we have done so we have applied this algorithm to the study of herpes simplex encephalitis. So it's a very rare manifestation of following the infection to a very endemic virus like HSV, and there are already known inborn errors of immunity, in particular of type 1 interferon immunity, that are known to underlie HSC. So we applied this algorithm to about 100 cases and about 500 controls, all of European origins, focusing on predicted loss of function and missense variants with a frequency below time -3 and a CAS-score higher than 10. And so we identified 225 gene clusters after applying the gene cluster algorithm. We merged those that were highly overlapping, so it was 143 clusters. Then, when we compare the proportion of individuals carrying at least one rare variant in between cases and controls, we end up with 8 significant clusters, and finally 5 had a Go term or a pathway significantly overrepresented in the cluster. And here are the list of clusters and what. So this is the typical output of the method, and what was very interesting is that the first cluster we identified, so it was a cluster of 32 genes in which 49 patients did carry a candidate mutation, and the top pathway that was overrepresented was the KEGG pathway, toll-like receptor signaling pathway. And the network is represented here, and what was very striking is that we were able to find all in this first cluster the known disease-causing mutation in known disease-causing genes, and also to identify new disease-causing mutation in the already known disease-causing genes in some additional patients, and also very important, interesting candidate mutation in new candidate genes that are currently under study. So to conclude my talk, just briefly, so we can test for enrichment at the variant level, assuming allelic heterogeneity. However, it's very underpowered, and so that's why we developed aggregation methods. The power of those methods highly depends on the level of genetic homogeneity and the proportion of non-disease causing variants, and it's why we need accurate tools to predict the potential, the disease-causing effect of the variants. There are multiple refinements of the method. I presented the simplest one. There are multiple refinements, but not that many examples of successful application. And maybe more interesting are the developments that allow to test for alternative hypotheses, such as, so as I presented, physiological homogeneity in the presence of genetic heterogeneity. And there is still room for improvement of the method we have developed. And also an interesting hypothesis that can be tested with those cohort-based approach is the digenic inheritance. So when two mutations in two different genes are necessary to cause the disease in a single patient. I thank you for your attention. I will be happy to take questions.
There's one question online, isn't there?
Yeah. So thank you, Aurélie. So there's a question online arriving. So first one for Aurélie, is it possible to perform a burden test with a very small set of cases, around 20? And if so, which is the best method?
It's always possible, but the expected power would not be very high. But then it will also depend on the number of controls that are available. As if you compare 20 cases to 20 controls, it will be very difficult to find anything. But if you have thousands of controls, well, you may have a good surprise. It's always so it's better to have a high number of cases, but you can apply the method to, I would say, any number of cases.
Okay. Is there a question in the room? Yes. Sorry, because I have two questions.
Hi, thank you for the nice presentation. My question is about this last algorithm that you showed about physiological homogeneity. My question is, it would be useful also to detect, how to say it, like a signal from a multigenic inheritance. So, I mean, for some immunodeficiencies in which maybe you don't have the causal mutation, if you have an enrichment using this method, maybe in this pathway you will not find a true pathogenic mutation, but the accumulation of variants may give you a signal for a oligogenic or multigenic inheritance. Do you think it's useful to detect this?
Yes, absolutely. For now, we do have statistical methods to test for digenic inheritance. But to statistically assess the hypothesis of oligogenic inheritance, it will need further methodological development. It's a bit more difficult with the tool we have, actually.
Okay, thanks.
And the application of this algorithm in for rarer variants, not less rare variants, not rare variants. So, like if you would like to focus the less rare variant, to see the impact of variants not so rare, and so that
this is absolutely
could imply a predisposition to some to a dysfunction like TIC2, for example. No?
Yeah.
Yes. You don't have a rare variant, but this variant could impact the predisposition to a.
Absolutely, and in the example of TIC2, in fact, so we can do what we used to do in genome-wide association study. So instead of we don't aggregate the variants in the gene, we test each variant individually. And if the variant like TIC2 underline a sufficient number of patients, then we will be able to see a significant association, which was the case for TIC2, I think, when we combine like TB and MSMD cohort. And I don't remember the numbers of cases and controls, but we did have a genome-wide significant signal, and the odds ratio in the recessive model was far the highest odds ratio of all the variants tested in the genome. So yes, absolutely, and it's why. So these are the typical methods we use in GWAS that can be applied, and it's interesting to do so. Absolutely.
So a last question for Alexander before the coffee break. Everyone needed. So when you filter removing parental variants, don't you risk removing relevant variants that can contribute to phenotype in association with a de novo mutation?
Like this. Okay, perfect. This is absolutely correct. So, of course, I did not want to give the impression that de novo mutations explain everything. It is just one hypothesis that sometimes a highly penetrant mutation arises de novo, and then it's good to systematically assess it. But indeed, sometimes you may filter out inherited variants, and of course it gets very tricky once you talk about post-zygotic mutations that may be in a Gonadal Mosaicism in the parents. So there's specific tools where we can also filter for those. But yeah, it's only one, and I think a quite good hypothesis, especially if it's severe sporadic disease, that you start with a trio approach, because it just helps you a lot. But it's certainly not the holy grail to everything, by far not, unfortunately.
So thank you. Thank you for the speakers, and let's have a coffee break and we come back, we resume at 10:45. Thank you.
For coming back. Our next session will be as lovely as the one before, I hope. I'm sure, of course. And the first one to introduce is, I would like to introduce. So, do you hear me? No. Oh. Now you hear me, okay. So thank you very much for coming back. We continue with our next session, and the first one I would like to introduce is Mrna Mensa. She's from the Clinic of Barcelona, and she's from the lab which works for many years in mosaicism, specifically for autoinflammatory disorders. And she will explain us all about the importance of mosaicism in immune deficiencies.
Yeah. Good morning, everyone, and thank you very much to Marielle and Aine for inviting me to participate in this workshop. It's a pleasure to be here. And I will talk about mosaicism. Here are the topics I will talk about. So mosaicism is present in our lives, is visible. You can see, we can find many organisms that are present with mosaicism, like plants, animals, and also human, such as the lines of Blasco or the heterochromia of the iris. But mosaicism goes beyond what is visible, and our genome is really unstable, especially during the cell divisions. Some post-zygotic mutation can arise, and also we are our environment. There are many physical and chemical factors that can induce these post-zygotic mutations that can provoke DNA damage. Fortunately, all the organisms have mechanisms of DNA repair, and they can restore normally, but there are some variants that keep it in the cell, these post-zygotic variants, and most of them are benign, completely benign, so all of us, we are mosaicism. But some of the post-zygotic mutation can stay in the cell, and the cell cannot adapt. And if the mutation locates in a gene that has a very important function, then it can lead to disease. So throughout the life of an individual, there are multiple cell divisions that I have told you, and some of these mutations that arise post-zygotically, they are called as somatic variants or post-zygotic variants, and they are different from those that are called germline variants. And I would like to explain a little bit the difference between them. Germline variants are those variants that are present in the first zygote in the conception because they can be inherited from one parent, or they can arise in a new individual and can provoke a de novo variant. No? And this is because maybe there are some variants that arise during the gametogenesis, for example. So they are present in the first zygote. And that means that the individual all the cells of the individual will carry the germline variant in all the cells until all the life. On the contrary, the post-zygotic variants are strictly de novo always, and the variant is not present in the first zygote. It appears either during embryonic development or in postnatal life in the similar manner that happened with cancer. The body distribution and the frequency of the mutated allele will depend on the precise time of the mutational event and which type of cell occur this mutation. So if the post-zygotic mutation arises early in the development, probably many cells will carry this post-zygotic mutation and will lead to extended mosaicism. In the contrary, if the post-zygotic mutation arises later in the life, in adulthood, for example, it tends to be a restricted mosaicism that is located in one specific cell. So the presence of post-zygotic variants lead to mosaicism, and the definition is that describes an individual who has developed from a single zygote and has two or more population of cells with distinct genotypes. There are different types of mosaicism, and they have different clinical consequences. We can find the gonadal mosaicism. The post-zygotic mutation is restricted in gonadal tissues, and in this case, the individual that carries gonadal mosaicism will be completely healthy, but has a risk of vertical transmission of this post-zygotic mutation and to the offspring, and if the offspring have this post-zygotic mutation, it will be a germline mutation. Somatic mosaicism, it describes an individual that the post-zygotic mutation is restricted in the somatic tissues. This type of mosaicism, the individual can be healthy or affected depending on the gene that carry this mosaicism. If the gene has a very important function, then it will cause disease. But it's important to know that if it's a pure somatic mosaicism, there is no risk of vertical transmission of the disease to the offspring. And we have the most complex mosaicism that it's called gonosomal. It means that the post-zygotic mutation is present in somatic tissues and in the gonadal tissues. So we can find the individual that can be healthy or affected, and because it's located also in the gonadal tissues, there is a risk, moderate to high risk, to transmit the mutation to the offspring, and if it happens, then it will be as a germline mutation. The fact that the individual is healthy or affected, it depends as well of the percentage of the mutated allele and which gene is affected. So how can we detect? It's really a challenge to look for mosaicism, you know that. We have to keep in mind which type of tissue we want to study for mosaicism and which method we will select for that. In primary immunodeficiencies, whole blood, studying peripheral blood, it's very useful, I think, because it contains all the cells of the immune system. But it's true that sometimes if the mosaic is restricted to a specific cell, then it could be better to select first to isolate the population of interest and then study the mosaicism. But this is really difficult because at the beginning, we don't know really what is the cell type. But for example, there are some, for example, in autoinflammatory disease, the main actors of pathogenesis is the innate immunity, so we can isolate myeloid cells and then see if there is a mosaicism there. We can, of course, look for mosaicism in other tissues. In the lab, we always, in our hands, we start with peripheral blood, but then if we find some mosaicism, then we extend to other tissues to see what kind of mosaicism it is, to look for a body distribution. And the method, it's very important always because it has to be very high sensitive. You know that we employ Sanger sequencing for detecting germline mutations, that it's around 50%. But for mosaicisms, the Sanger sequencing, it's limiting. You can see here an example of 15% of mutated allele. You can see this small peak of adenine here, and it's very small peak. This is confusing with the background noise of the technique. So we need to employ for very low grade of mosaicism the NGS-based methods. Next generation sequencing is the best method to detect and quantify the mutated allele. In our hands, we employed Amplicon-Based deep sequencing. It's a technique that offers a high coverage, many reads, up to 10,000 or 1,000 reads, covering a position. And it can help a lot to look for mosaicism. We have all the other techniques like whole exome sequencing or genome, but the coverage that we obtain, it's normally very low, so it's very difficult to detect, and we have to employ a lot of time to detect some variants that are somatic. But in the literature, there are examples that they have found mosaicism by exome sequencing, for example. Some cases are this case of mosaicism in NLRP3. It was detected on 17%, and they have a coverage of 127 reads. So they could finally found a mosaicism that has been missed by Sanger sequencing. It is another example of thinkonomid syndrome caused by a somatic NLRP3. The authors, they used whole exome sequencing, and they found mosaicism in more or less 17% of mosaicism, and they had also 148 variants. And the Sanger chromatograms didn't reveal this mutation. And this is a recent case that I was very surprised when I read, because it describes a woman that have an autoinflammatory disease, a late onset. They made the whole exome sequencing, but they could find anything. But then with the reanalysis, because they suspect a lot about autoinflammatory disease, so they make a new filters analysis, and they could find a mosaicism in that woman ratio of 2.6%. So they only have 115 reads. So it was like very challenging in this case, but they could validate the variant and make pathogenicity. So what we know about mosaicism in primary immunodeficiencies? The first cases were in the late 80s and 90s, and they were somatic reversions. In patients, they don't have the techniques, but they observe the phenotype of the patient that improves the clinical, and it was a somatic reversion in deficiency of ADA1 or X-linked severe combined immunodeficiency. And also because of an expected intrafamilial recurrence of a disease, of an autosomal dominant disease. So it was suggested gonadal mosaicism in one of the parents. So mosaicism, it was talked with in this period. In 2004, it was described somatic mutations in FAS gene, and it was like an inflection point, and for this new mechanism for causing the diseases. And for autoinflammatory disease, 2005 was reported the first somatic NLRP3 mosaicism, and after that, and during the last years, many genes have been related to autoinflammatory disease. One of the very important one that makes like a new era of autoinflammation, it's UVA1 that causes Bex's syndrome, for example. And most of the variants have a gain of function mechanism, but we found like FAS or UVA1 that have a loss of function mechanism. So it's variable. So which are the consequences of somatic mosaicism? Patients that carry somatic mosaicism may have the same phenotype as germline variants, for example, in FAS, in ALPS patient, in CAPS, in Blow syndrome, TRAPS, and SAVI. They are patients that somatic variants cause the same phenotype of germline. They can associate with a milder disease phenotype. Here is an example, for example, the cryopyrin-associated periodic syndromes that have 3 independent diseases. The milder one is the familial cold autoinflammatory disease, the intermediate is the Michael-Wells syndrome, and the severe one is the thinkonomid. So for the same variant, if it's present in germline, it causes thinkonomid syndrome, but if it's present in somatic state, then we can find the intermediate state of disease that it's Michael-Wells. So it's really nice when we have the same mutation but different mutated allele. It has a relevant role of causing a late onset disease presentation, more than 50 years. And usually this type of mosaicism is restricted to a cell type. Some of examples we can found in NLRP3 causing CAPS, or NLRC4, or recently in UVA1 causing the BEXAs. And we also find different phenotypes. For example, mutations in NLRC4 that are germline cause a severe autoinflammatory disease that is associated with enterocolitis and macrophage activation syndrome. But when it's in somatic state, then the phenotype changes and it's like a Thinkanomid syndrome. In UVA1, the germline variants are related to spinal muscular atrophy, but when we found somatic, then it's BEXA syndrome. So we have a very variable phenotypes that mosaicism take a relevant role. And also, there are some genes only exist as a somatic form and not exist with the germline form, and that's because maybe this variant or this gene is incompatible with life if present in the germline. Here is an example. I want to illustrate you a gonosomal mosaicism example. It is a family with two children affected with severe congenital neutropenia, and the mother has a cyclic neutropenia but didn't have any type of infections. Just in the routine controls, they found that the cyclic neutropenia. So we study both of the siblings, and we saw Sanger sequencing an heterozygous pathogenic variant that when we quantify by next generation sequencing, it's a germline, compatible with a germline variant. But our surprise, when we did Sanger sequencing of the mother, we also see an heterozygous variant, and we say, okay, she's a carrier. But when we did the NGS, we found that the percentage of the mutated allele was about 30%. So we re-examined the Sanger chromatograms, and we could see that the mutated allele that corresponds, it's a cytosine in the siblings, are higher than the wild type. But when we see the mother, the mutated allele is lower. So this is when you go quickly in the routine diagnostics, maybe you see, okay, the mother is heterozygous, but not. It's a post-zygotic variant, and this explains the milder disease found in the mother. So sometimes it's good to perform NGNs for that, because you can quantify and see if it's germline or if it's post-zygotic. Here is another example of gonadal, this case, Gonadal Mosaicism. It's a family with two children affected of a primary immunodeficiency associated with PIK3CD. Both are carriers of the same mutation. So they send the parents, but none of them in the peripheral blood carry the mutation. This is an autosomal dominant disease. So because there are two children affected from the same disease, sure that one of the parents have the variant. So we ask for sperm cells of the parent, of the father, but we didn't find the mutation. So we suggest that it is a maternal gonadal mosaicism in this case. So to conclude, from clinical point of view, patients with gene mosaicism have expanded the clinical diversity of some diseases towards milder, atypical, or late onset disease. From therapeutic point of view, it's important gene mosaicism because patients can start treatments under labeled indication as soon as possible. From genetic concealing, it's also important because patients with gonadal and gonosomal mosaicism have a risk of transmitting the mutated allele to future generation and may provoke unexpected recurrence of dominantly inherited diseases in a healthy couple with no mutations detected previous with standard genetic analysis. And from methodological point of view, the detection of mosaicism is challenging, and we strongly recommended using the next generation sequencing methods at high coverage. This is very important for detecting and quantifying gene mosaicism. And Thank you for your attention. If you have questions, I'll be happy.
Thank you very much, Anna. Are there any questions from the audience?
Thank you very much for your nice talk. When one is faced with a family where it looks as if it's probably a de novo mutation of an autosomal dominant condition, for example, STAT3 gain of function in the child, and there is another sibling who is apparently healthy, what are the risks? What are the risks of the kind of gonosomal mosaicism? Is it high enough to mandate screening of those healthy siblings where parents themselves lack the variant?
Yeah. If the genetic study is carried in the parents, in both parents, you don't detect the variant, yeah, there is a possibility. We don't know which possibility, but there is a possibility that one of the parents carry a gonadal mosaicism. That's true, and I think families have to know that. But we don't know the percentage of the risk. Yeah, it's for each gene must be different, I think.
Thank you. Hi, thank you for the presentation. So I have a question. If you have a patient, an adult patient with a mosaic detected, for example, of excess, if you want to give him counseling for the children, what can you say? So it's not about the parents and children with the disease. He is the first person with the disease, and he wants children. So what can you say for counseling?
If the disease appears in an adult individual, then it's, in our experience, it's a very low risk that that person can transmit the disease to the offspring. We usually test other tissues, like, for example, nails. Or nails is a very good tissue to test because it's different from different tissue origin than hematopoietic. So if it's present in the nails, then you can suspect that maybe the mutation arises early in the development, and there is a possibility to transmit. If it's a male, then we can study the sperm cells to be sure that there is not there. But if it's a woman, that's not possible. So if the disease starts later in the life, it's very low probability that there is a risk recurrence of this disease, but we are not sure. So we recommend to study other tissues in the body to see if it's extended.
You can offer prenatal testing if they want to.
Yeah, exactly.
For the next generation, you can offer prenatal testing. Yeah.
Sorry?
You can offer prenatal testing if there's a pregnancy.
Yeah, the prenatal testing. It depends on the disease, but usually we are not performing prenatal testing for somatic. Yeah, if there is a high risk of that, and it depends on the disease. I mean, there are some rules on that, but if the disease appears early and it's very obvious that it's extended mosaicism, then we have to treat the disease like a germline, more or less, because it's somatic, but it's present, and maybe in the gonadal tissues also is present, so we have to be careful.
Okay, thank you.
Really nice. So we now perform sort of hundreds and thousands of patients with next generation sequencing, and we apply quality standards, and we filter out sort of variants that are sort of below these standards. Now, for certain variants like haploinsufficiency or X-linked genes or gain of function, this might even filter out some real mosaicism. From your reading, what do you expect? What do you guess? Sort of how much do we miss just by setting sort of our thresholds to 10%, 15%?
Yeah, for the normal filtering for germline mutations, I'm sure that we lost all the somatic variants. So we need to perform, I think, another analysis specific for looking for somatic mosaicism. In our lab, for example, we employ two analyses. The first one that it's for germlines, and we applied 30% of a mutated allele and everything. But then we have another analysis that is specifically for looking for somatic, very low somatic mosaics, and the filters are 2% of mutated allele. So we need high coverages to really think that this is the true pozygotic variant, and then we have to validate and see that it's true with another sample. But I am sure that with the routine analysis of performing of exomes, or we lost the variants. We have to know the clinical suspicion and look for that. If you suspect a STAT3 gain of function, then we have to be sure that the STAT3 it's really well covered. And I'm sure that the paper of NLRC4, for example, they looked position by position in the IGV to see if there is something. And maybe the candidates will be like hundreds. And then to validate with another technique to see which one is true and which one is not.
There are some questions in the chat, and then we continue
One
with the next
question on the chat. So, Anna? So, fantastic presentation, so thank you. One question, would you recommend studying for gonadal mosaicism every patient where somatic mosaicism is detected?
For men, yes, we can study, but for women, we can't study because every month the woman has one ovule, so it's impossible to test each one. So for that cases, we can only study males. We can study sperm cells, but maybe I don't know if one it's good, or we need maybe 2 or 3 times to see different points of that to really discard gonadal mosaicism. Yeah.
Okay, thank you very much, Anna, for a very nice presentation. And we would like to continue. We would like to continue with Professor Mrna Sequencing from the Molecular Medicine Institute in Helsinki, but she's also, are you still listening? Yeah. She's also affiliated with the University of Oslo, where she also partly works. And she will talk about today as well about ClinGen, and where she's an expert in reviewing genes in this entity as well. Thank you very much, Mrna.
Thank you, and thanks for the invitation to talk. I need to start with a disclaimer. My research group is mostly studying those cases where we don't find a clinical diagnosis and trying to find new genes. So I'm playing a devil's advocate here, in a sense that I'm trying to bring the difference between what we can do in the research setting compared to what we need to do in the clinical setting and the standards that are put in. But let's go ahead with this. And so what I was asked to talk about was the ClinGen resources. So ClinGen is defining the clinical relevance of genes and variants. And ClinGen is a international collaboration effort. It was founded in 2013 by the National Human Genome Research Institute. And it's a growing collaborative effort, so currently involving over 1,800 contributors from 45 different countries. And what it aims to look into is indeed the clinical relevance of genes and variants. And do that by using standardized semi-quantitative evaluation of the strength of evidence for gene-disease relationship or disease variant relationship. for gene-disease relationship, there's a classification between definite and no reported evidence, or there might be conflicting evidence, as we heard earlier. The variant classification is using the ACMG AMP guidelines. And both of these classifications are made by the curators who work for ClinGen, looking into either genes or variants. And then they are confirmed or adjusted by a focused clinical expert panel. And I'm part of the gene disease panel for severe immune deficiencies and combined immune deficiencies. So for the PIDs, there are two expert panels for evaluating the genes, the SCID-SID, and then the antibody deficiencies expert group. And there are also two variant curation expert panels who have started their work but have not started, have agreed on kind of the guidelines and standards, how they work, but not yet started qualifying the variants. So I'm a bit early here talking about variants in primary immune deficiencies in that sense. But as said, it is based on the standard and guidelines by American College of Medical Genetics and Genomics and the Association for Molecular Pathology, and I'm sure that this paper is very familiar to all of you, but I will still go through some of the points that they are making there. And a good reminder here is that these guidelines are actually intended to determine whether a variant in a gene with a definite role in Mendelian disorder may be pathogenic for that disorder. And we are currently all the time using that for candidate genes, which is not for which they are meant to. And another reminder is that determining the pathogenicity should not be dependent on the particular patient, but it should be actually independent in interpretation of the disease of a given patient. So in order for us to make a standardized evaluation, we should have a clear definition that a gene-disease pair is valid and is definite. And just to point out how that is done, I use a very familiar example. You all know ADA2 and the deficiency of ADA2 causing SCID. So it was the first immune deficiency identified already in 1970s. And there are over 80 disease variants in very many different categories. We know the model of inheritance. We know the disease mechanism. And there is a plethora of functional evidence that tells us that this is really the gene that is causing SCID. There are both human and non-human models, and also rescue experiments in non-human models, but also in humans when we have been given enzyme or gene therapy to rescue these patients. Even there, one can think of is it that there is this original phenotype that is T minus B minus NK cell minus severe combined immune deficiency, starting early on, very early on, and usually resulting in early death if not if untreated. But there are also variant phenotypes with the delayed onset or even late onset, or there are partial ADA deficiency, and then whether we should consider these as the same disease or a different disease. And the ClinGen gives guidelines to consider that, and whether you lump all the different phenotypes into the same disease or whether you want to split them. And what you should consider is whether the gene-disease pair is asserted in the existing databases like Mondo or OMIN as two separate or more separate phenotypes, or whether it's an entity, whether there are molecular mechanisms that are different between the different phenotypes. And so if the phenotypic characteristics are something that are clearly different between the two different phenotypes, or whether they are overlapping, and also whether the inheritance pattern of the disease is different or the same. So one example of the gene that we curated as split into two is that 3, with the loss of function and gain of function mutations causing a different disease. The one that you want to also consider is model of inheritance, so whether it's autosomal dominant, X-linked, or semi-dominant. So semi-dominant meaning that either one allele or two alleles can cause a disease. Usually there's a difference in severity. And the framework for classification looks into genetic evidence that may be on case level or case control level. And just looking at if there is enough evidence for the described variants that give you enough points to make it definite. So it's a non-exhaustive analysis of the existing literature. You look into whether you can find during the years that the gene is known enough points, and the maximum is 12 points. Then also looking into the functional evidence that can be in the gene function or protein function, if there's functional alterations, if there are models, either human or non-human, and if there has been done a rescue experiment or more of them. And there again, you just need to find enough proof to get to the maximum point of 6. So the curations that summarize all don't have all the evidence in it, but it tries to pull together kind of good evidence of what the disease is, how it is linked, what's the kind of disease model, and also whether it has been demonstrated in the research and clinical setting over a certain period of time. And here again we go to the problems with the recently described genes, because obviously there has not been time to do that over years. But if we go back to the variant interpretation, so the data that we evaluate is the variant frequency, as we heard well from the previous talks. Then the variant consequence, whether there is functional data, whether there is segregation in the family, whether the variant is de novo or not, and whether the variant is in cis or trans with another variant, if there is information in the other databases, and if there is other data that we should consider. And there are practically two sets of criteria. One is whether the variant is pathogenic or likely pathogenic, or whether the variant is benign or likely benign. And as there is different levels of evidence, or the strength of evidence, they are tagged with very strong evidence for pathogenicity, strong evidence, moderate, supporting, and similarly for the benign variants, standalone, strong, or supporting for the variant being benign. The kind of the very strong evidence for the variant being pathogenic or likely pathogenic is when the variant is assumed to disrupt gene function by leading to complete absence of the gene product. So these are typically nonsense, frame shift, canonical splice sites, plus minus 1 or 2, loss of initiation codon, and then single exon or multi-exon deletions. What is good to keep in mind is that not all null variants are pathogenic, and particularly if the known disease mechanism of that gene is not a loss of function of that particular protein, we should not consider those null variants pathogenic. And there it is also a challenge that if we only have one patient, or if we don't know well the mechanism of the disease, it is difficult to decide whether we should be excited about the null variant or not. But in most cases they are. One also needs to be careful when interpreting truncating variants close to the end of the gene that might not actually cause nonsense variated decay, and there might be a shorter form of the protein. And if that shorter form of the protein contains all the functional domains that the protein needs, it might not actually cause a disease. Or it might cause a dominant negative disease, which you don't know by just looking into the variant. And it needs then some functional evidence. Also the splice variants, whether they are causing a frame shift, which is more likely to be a null, or whether it's an in-frame deletion or insertion of one or more exons. And again, if that's not taking out a functional domain, it might actually be not pathogenic. Also, the truncating or null variants can be in alternatively spliced exons, or alternative gene transcripts. And if that particular transcript is only expressed in one tissue, that is not relevant for the disease that you are looking into, so it's unlikely to cause the disease. A very simple variant type, but still a lot of to consider. Then there are the strong evidence cases for the different variants. And strong evidence is when one misses variant is known to be pathogenic, but what you see is not the original nucleotide change, but the different nucleotide change that is causing the same amino acid to be changed. There it is, that's considered strong evidence. Particularly if the mechanism for pathogenicity is that the missense variant alters the protein function. For some diseases, we all know that the variant may actually act on DNA level, causing splicing differences, and not on the protein function level, so that's good to keep in mind. Second, strong evidence for being pathogenic is de novo variants, and we heard a good presentation today about how to identify the de novo variants. And in case you consider a variant to be de novo, you need to make sure that you've tested that the parents are actually biological parents. I think everybody has good examples on that not being the case. And in a case where you can't confirm the paternity of the assumed parents, it's only moderate evidence for pathogenicity. And I don't need to go to the next point. We just heard of a kind of variant that looks de novo, but is actually a germline mosaicism in the parents. And also in case of de novo variants, it is good to consider that the type of disease that the patient has actually has been described as being caused by a de novo variation. Then the third one that is providing strong evidence for pathogenicity is functional studies. And they are powerful in either supporting or actually against the pathogenicity. But there again, a functional test is not a functional test in a sense. You need to consider how close the functional assay reflects the biology. Are we actually testing and measuring something that is causing the disease, or are we testing something that just causes a difference in the protein function, but may be totally disease independent. So a functional deviation in a rare disease is not the proof of pathogenicity. I think you need to remember that. And kind of usually a very good functional test is if you are assaying an enzyme. Variant is making a kind of a stop gain or a missense mutation in an enzyme with the enzyme being a known disease mechanism for a particular disease, and you see that the enzyme activity is disrupted, it is good functional evidence. Then you can look into whether the assay is done in tissues from patients or animal models, or if that is created in the cell line, where it comes a bit further. It may be a totally good functional assay, but you need to be careful when evaluating that. Also, looking into the functional assays that they are well validated, reproduced, and robust, and so that the samples that have been sent for the functional assay are still in good condition. So if the blood samples sent for Treg analysis have been long on their way, you may see nothing with a Tregs or in their function, no problems, or you can see severe problems, but it's not valid if the cells are not healthy and in good shape, so that needs to be considered as well. And for variants that impact on RNA level, like splice junctions or within untranslated regions, RNA-seq, or looking actually to the RNA transcript, whether you do that with the more traditional Sanger sequencing or by using a long-read sequencing, is a valid way to look into variant effect. Then the variant frequency and how frequent it is in the population is also can give kind of strong evidence for pathogenicity, but it can also give evidence for the variant being benign. So as I said, as we have the kind of rare variants in a population level are not rare, so there are plenty of rare variants in our genomes, and if we look at the population level, there's a huge number of them. We can look into if they've changed function at all. Many of them probably do a bit. 5% reduction, 10% reduction. Does it really mean anything? But then if on top of that you can show reliably that the variant is more frequent, statistically significantly more frequent in the case population than in your controls, or if the odds ratio or relative risk ratio in the case control analysis is high and your confidence interval is either clearly below 1 or clearly above 1, that gives you strong evidence. So variant is not proven pathogenic if you don't see a difference between patients and controls. And here again is a caveat: if we only have one patient, how can we tell that it's more frequent in cases than in controls? Then kind of moderate evidence can be brought in by if the variant is absent or below expected carrier frequency in case of recessive disease in ethnically matched large general population or control cohort. And then we talk about the cohort that is over 1,000 individuals. If possible, the more is better. On the other hand, if the allele frequency in control population is greater than expected for a rare Mendelian disorder, that provides strong evidence for variant not being pathogenic, and it is suggested that if it's 5% in the normal population, it can not be a pathogenic variant in Mendelian disorder. Here we do need to remember the penetrance. If the penetrance is not full, we need to adjust the levels of the frequency accordingly. Also, highly or strongly suggesting that the variant is benign is when it's observed in a healthy individual, adult individual, and the disease is fully penetrant early onset disease. Then we have quite a few moderate supporting evidence for the variant being pathogenic. These are, for example, mutational hotspot or the variant being in critical and well-established functional domain. So if all the previously reported missense variants in a certain domain are pathogenic, it is supporting or it's moderate evidence for the new variant also being pathogenic. And particularly if no benign missense mutations have been reported within that domain. Or if there is a cluster of mutational hotspot or cluster of variants in a certain region that is conserved, a region between species. Then one can look into other kind of moderate evidence, is whether the variant occurs in KISS or trans of the known pathogenic variant. So KISS means that it's in the same allele, and trans means that it's in a different allele, or a different copy of the gene. And so if a variant in a gene for a recessive disorder occurs in trans with a known pathogenic variant, it gives you moderate support. And if it actually occurs in patients with many different pathogenic variants, that's even more stronger support. But then on the other hand, there's a kind of support or suggestive evidence for variant being benign for recessive disease detection of a second variant that is in KISS with the known pathogenic variant. Yes. Almost finished. Just a reminder that then kind of in-frame deletions and insertions and stop losses are in general considered moderate. If they are large and are happening in conserved amino acids, you can use a stronger evidence. If they are small and in repetitive regions, it's less likely that they are pathogenic. The segregation analysis, I'm just saying that segregation of the variant in a family, even that's a good way to start finding new candidate genes, is not the proof for that variant to be pathogenic. It is actually proof of linkage within this family. So one needs to consider that. And then on the other way around, the lack of segregation in the phenotype is kind of against the pathogenicity, but then you need to consider whether you've done the phenotyping of the family members kind of carefully enough, because there might be late onset of the disease, or it might be that there are also only immunological phenotypes, and you don't see that on a clinical picture. Then there's plenty of other supporting evidence that I don't go through. And when you put everything together, you get this matrix of supporting and strong evidence for being pathogenic or benign variant, and with kind of a classification that tells what type of evidence and how much of it you need to make a pathogenic call, or likely pathogenic, or when you can consider the variant to be benign or likely benign. And everything else is WUS, and most of them are WUS in that case. Just going back to the ClinGen resource, as I said, we are in a very early steps for classifying variants for primary immune deficiencies. But what is important and highly useful is the ClinGen variant curation interface. That is a global resource, and it has a good tutoring material, and that you can use to do this in a standardized way, and also record the supporting evidence why you made a call. And of the 3,615 curated variants across 67 genes, none of them are yet in PID genes. And I just want to thank my colleagues in the gene curation expert panels that are meeting every second week. Thank you.
Thank you very much, Jana, for this excellent and very clear talk. Are there any questions?
That was wonderful. Thank you so much. So I think one feature that always keeps me busy is that certain diseases may be caused by completely different mutational mechanisms. So we know that neurodevelopmental diseases very often are caused by haploinsufficiency. So clearly loss of functions of one allele can be sufficient. While for the immune diseases, probably gain of function mutations for dominant diseases may be much more important. So how can we set the same rules for all the rare diseases and still find some kind of fine grading on the disease type and the expected mutational mechanism? Have you given that some thought? So it could be that in some circumstances a loss of function may not be important at all, while a gain of function may be much more important. That's the question.
Yes, and I think even for primary immune deficiencies, definitely there are good examples of haploinsufficiency causing the disease rather than a gain of function. But I think the most important part of it is that in order for you to classify the variants, you would need to know the disease mechanism. And then you can see whether you give the points to that particular variant if it fits the disease mechanism. Here we go again to the challenge of the new genes that we are so eager to use in our clinical setting. And when we don't actually know these basic facts, how do we then interpret and classify the variants? I fully agree that we should do that. And maybe there is room, as I said, these rules are for known disease genes when we actually know the disease mechanism and how it affects. But for clinical use, we do need some other rules that we can use for candidates and a good discussion between the clinicians and the lab in order to understand how how strong the evidence is and whether we can actually use that evidence in the patient treatment. I fully agree with you there.
Okay, so if there are no more questions, maybe we move on to the next speaker. So it's my pleasure to welcome Esteban Ballesta, head of the Lab of Epigenetics and Immune Diseases from Barcelona. Esteban, please.
Thank you for the invitation and to speak here at the workshop. It's a pleasure. And well, first, maybe after the previous talks and this audience, maybe I don't feel so brave to say that. But the truth is that at least for genetically complex diseases and for many for many monogenic syndromes, it's not always, genetics alone cannot always explain the development of the disease. And in fact, sometimes genetic mutations or variants in the context of complex disease don't always associate with the development of the disease. And in other cases, there's a clinical phenotype diagnosis that occurs in the absence of mutations or a particularly susceptible phenotype. So I think that epigenetic changes can perhaps be associated sometimes in a better way with the clinical phenotype, that alterations, epigenetic alterations can be used as biomarkers or classifiers of patients, and also because they are pharmacologically reversible, open also potential for, I'm going to take this, no, I guess, for pharmacological treatments. So just as a reminder, we have to think that our genes are going to be, in the context of the genome, are going to be in regions that are active, euchromatin, and others that are going to be associated with heterochromatic regions that are silent, and epigenetic modifications are going to be associated with these specific profiles. So in fact, we can imagine like a normal situation where some genes will have a particular epigenomic profile. We can see it on the top in gene X. And so these epigenetic modifications are going to be characteristic with this gene in its transcriptionally active status, whereas other genes will be in a silent status, and this will particularly associate with another set of epigenetic modifications. So we can ideally imagine of an epigenetic lesion that is going to silence the gene that needs to be active, or it's going to activate a gene that should be silenced. So in this sense, with the tools that we currently have, we can obtain at the genome-wide level profiles of epigenetic modifications. In my group, we particularly work mainly on DNA methylation, which we can see there. So it can be acquired de novo through the activity or the catalytic activity of DNA methyltransferase 3-NA. So these enzymes are able to introduce in a de novo way and methyl groups into the DNA. And of course, there are passive ways of demethylation or methylation maintenance, but there's also active mechanisms of demethylation that are able to remove methyl groups and are going to have an effect on the transcriptional status of the genes. So today I'm going to sort of divide my presentation in two types of stories. So I will present an example of the in vitro models that we use to investigate how different factors are able to influence the DNA methylation status. So these are in vitro models that we use in the lab, but provide a perspective on how different factors can act and influence on the DNA methylation. And the second, I will present two stories, but the second group of stories will be focusing on analysis of DNA methylation that we have performed in samples from patients with different conditions, and trying to also understand the underlying mechanisms or the relevance in the clinics. So for the first sort of stories, basically the bottom line is that extracellular stimuli can influence through the activation of, through the binding to receptors and the activation of signaling pathways, can influence ultimately the transcriptional status of the genes. And the models that we use in the laboratory are based on the use and analysis of peripheral blood monocytes that we are able to differentiate in different cell types, in dendritic cells, using the standard method that Salusto and Lazzavecchia developed in 1994, using GM-CSF and IL-4. So we can generate dendritic cells, Langerhans cells, M1 and M2 macrophages, so more pro-inflammatory or anti-inflammatory, myeloid-derived suppressor cells, or we can also, for instance, study the influence of factors like vitamin D or corticosteroids in the acquisition of tolerogenic properties of dendritic cells. So these are the models that we've been using in the lab for some years. I will briefly present today an example of this, how these factors can influence DNA methylation. So for instance, in this model where we compare the differentiation from monocytes to dendritic cells on the top, using the method that I mentioned before, and we can generate tolerogenic dendritic cells with dexamethasone, and on the right panel you can see the different properties to stimulate T-cell proliferation. You can see in dendritic cells in orange that they stimulate T-cell proliferation, and in the presence of corticosteroids they block such proliferation in green. So when we perform DNA methylation profiling, we can see that the addition of corticosteroid is able to not only to generate these two different phenotypes, that has a direct effect on DNA methylation. So we can see that dendritic cells have a cluster, C1, of genes that demethylate specifically in the absence of the corticosteroid, and there's another cluster, C2, that demethylates specifically in these tolerogenic dendritic cells that are generated with the corticosteroids. So these changes in DNA methylation correlate inversely with transcriptional expression. We can see it on this panel. So genes that are demethylated in general are more expressed. So you can see that correlation between the DNA methylation data and transcriptional data. And when we perform this type of analysis, we can study the features of the clusters of genes that become demethylated in one or the other group. And in this case, for the genes that are demethylated in the presence of corticosteroids, we saw that there's an enrichment of transcription factors that not only there are motifs in these genes that are demethylated for the glucocorticoid receptor that you can see on the top left of that last graph, but you can also see MAFB and other members of this transcription factor family as enriched on the genes that become demethylated, specifically in tolerogenic dendritic cells, as a result of this corticosteroid stimulation. So in this study, we observed that, in fact, not only the glucocorticoid receptor translocate to the nucleus when we add dexamethasone, but also there's an overexpression of MAFB. MAFB, in fact, when we perform ChIP-seq analysis, we see that MAFB has a much higher genomic binding sites that the glucocorticoid receptor. We see it there, although there's a very good overlap when we consider the binding sites for the glucocorticoid receptor. And when we overlap this information with the DNA methylation changes, we can see that actually MAFB has a much higher correspondence with the sites that demethylate in tolerogenic dendritic cells, the glucocorticoid receptor. So actually, MAFB seems to have a more relevant role on the demethylation of those sites. So this is just also an example of the binding site of the glucocorticoid receptor, where we see that actually the glucocorticoid receptor binds MAFB. So it seems that sort of subcontracts MAFB to do the role of performing demethylation in these genes. This is just, well, in fact, when we downregulate, we reduce the levels of MAFB, we can see that we can restore the immunogenic properties. We can see this in these T-cell proliferation assays, and we see a reversion also of the demethylation that is occurring. Finally, in this study, we also obtained cells from the synovial fluid of patients, rheumatoid arthritis patients, treated or not with corticosteroids. And by doing single-cell RNA sequencing, we were able to identify 3 myeloid populations that could relate to the phenotypes that I have described. In green, this M2 population corresponds to the cluster of cells that are closer to the tolerogenic phenotype that we had described in the in vitro study. So when we compare the profiles of the patients non-treated versus those that are treated with corticosteroids, we can see that there's an increase in this tolerogenic population on the bottom on those patients that are infused with corticosteroids. And that cluster that is increased is associated, again, with the peaks and the expression of MAFB, sort of reinforcing the idea that MAFB has a main role in the generation of this tolerogenic phenotype, also in vivo. So this is just to illustrate sort of the type of information that we can get in these in vitro studies by correlating the effect with the factors that could be potentially involved in the acquisition of these DNA methylation changes. So for instance, we have a similar study where we studied the effect of vitamin D in the acquisition of tolerogenic properties. And these type of models, in fact, are used also for, and they are in clinical trials for the treatment of several inflammatory diseases. And in fact, the treatment with corticosteroids and the generation of tolerogenic dendritic cells are used to treat patients with rheumatoid arthritis or with vitamin D in the case of multiple sclerosis. So basically, this is the type of the information that we get from these studies. So in vitro immune cell differentiation models help us dissect regulatory elements in the acquisition of these epigenetic changes, in this case DNA methylation, and allow us to identify the impact of treatments and different factors such as cytokines, hormones, vitamins, etc., on DNA methylation. So, and finally, they may help develop novel tools for cellular-based therapies. So I'm going to move to the second sort of type of studies that we do in our laboratory, which is the analysis of DNA methylation samples from patients, and we try to associate with the relevance in the clinics. In this sense, we have performed several studies on rheumatoid arthritis, and as you know, it's a disease that is mainly characterized by inflammation of the joints, but this can also affect other organs and also in the end affects at the systemic level. So I'm going to present briefly two studies that we have performed in collaboration with Juan Cañete in the Hospital Clinic in Barcelona, where we obtain DNA methylation profiles of patients for rheumatoid arthritis patients. With different, it's a small cohort, it's around 35 patients, where they were treated with different, I mean, it's a small cohort, but we had the clinical information, including the activity of the disease activity of the patients, and also the treatments that they had, etc. So when we compare the DNA methylation profiles from peripheral blood monocytes of these patients in comparison with healthy donors, we observed that they had a differential methylome. You can see the rheumatoid arthritis patients on the bottom and then healthy donors on the top. So we obtain a differential methylation profile in the monocytes from the blood. The interesting thing in this study is that we saw more sort of variability in the DNA methylomes in the patients than in the controls. And by testing different potential elements that could be associated with that variability, one of the features that could be involved is the activity, which in the case of rheumatoid arthritis is measured as the DAS28, which in very briefly consists of counting the joints of the patients and then obtaining also their erythrocyte sedimentation rate and make a global assessment of the health. So when we looked at the DNA methylation profile of these patients in relation to the disease activity, we observed that patients with high activity had a differential profile in relation with those in remission. And in fact, the patients in remission had a virtually identical methylome that healthy donors. So activity seemed to be highly associated with the methylome of the monocytes in the peripheral blood. And these are just examples from the methylome from the very same patient at different moments. For instance, if you look at the healthy donors are on the top, but if you look at, for instance, at the first patient, we have the methylome at high activity. You can see at 6.2 of DAS28, and the methylome of the very same patient when it's in the remission. So it seems to be sort of sensing perhaps the inflammatory status during the activity flares. So these are for the genes that are more methylated in high activity, and those correspond to the genes that are less methylated in high activity. So we can see the opposite behavior. So with that, we were able, well, we first attested the possibility that the cytokines that are elevated, have elevated their levels during the activity flares, were influencing the methylome. And we tested several that we had measured to be elevated, like TNF-alpha, interferon alpha, or interferon gamma. And when we, in vitro, incubated peripheral blood monocytes and then isolated monocytes after those treatments, we were able to see that we could influence the methylome of these monocytes by treating them with those inflammatory cytokines. We can see there the overlap of the influence of the different cytokines. But moreover, when using this approach, we were able to calibrate the system and also use the DNA methylation profile of the monocytes as a way to estimate the DAS28 of the patients. So basically, the DNA methylation of the patients is able to provide an accurate measurement of the activity of the patient. Of course, in the case of the rheumatoid arthritis, there's no need to do that because DAS28 is already a very, very good marker. But there are other inflammatory conditions that have no such good markers, and we think that methylation could provide not only a specific marker for activity for those conditions, but also to serve as a meta-marker of activity in overlapping entities. I would like to explain a little bit also our work on immunodeficiencies, but before, briefly, I will mention that we have used also in the context of arthritis, we have also used methylation to profile patients that are enter the study when they have undifferentiated arthritis. So we did a study, this study also in collaboration with Juan Cañete in Hospital Clinic. So patients that had an undifferentiated form of arthritis, and we followed them over time, and after 18 months, some of them had good prognosis and had a spontaneous remission, whereas others had poor prognosis and had, well, they needed DMARD therapy, and they had a joint erosion and a persisting disease. So when we profile these patients over time, well, first we observe that they have a differential methylome against healthy donors, but the first interesting result in relation to their outcome in the future is that at the first visit, they already have a differential methylome that you can see in the middle and the right-hand panel. So you can see that these patients already in the first visit have a differential methylome in relation to their future outcome. And you can see it also here with the results from the first visit on the left. So you can see that the methylome of the patients with bad prognosis is more distant from the healthy donors. But also we can see that after these 18 months, they both recover the methylation profile of the healthy donors, which is on the very left. You can also see an example of a methylation site there, where you can see that the methylome of the bad prognosis patients is more different, but the treatment or their spontaneous remissions eventually lead to similar recovery of the methylation profile. We can see also this at the expression levels. So this sort of highlights the possibilities that these DNA methylation measurements allow in relation to the activity or even the outcome of the patients over time. So I'm going to briefly explain you a second story in relation to methylation in the context of immune disease, but in this case it will be in primary immunodeficiencies, and this is a study. As you know, there are many different, but we've been focusing on primary antibody deficiencies, and in particular, I will be talking today about common variable immunodeficiency. All these primary antibody deficiencies have similar clinical features in relation to bacterial infections, and common variable immunodeficiency, as you may know, is the most frequent symptomatic primary antibody deficiency, with a severe deficiency of switch memory B cells as the most common clinical phenotype. However, only 20% of the CVID cases are diagnosed with monogenic gene mutations in different genes. The most common are shown here, and the clinicians face usually that they cannot provide a molecular diagnosis to around 80% of their patients. So one of the questions is if there's a common umbrella for this condition, or if there are differences between those patients with mutations and without. So some years ago, we started a project in collaboration with Eduardo Lopez in Hospital La Paz in Madrid, first studying a pair of monozygotic twins discordant for CVID without pathogenic gene variants, with all the, but without, they had been, exome analysis had been performed on them, and we isolated CD19 positive cells, total B cells, and look at the methylation status using bead arrays, with the idea of validating potential results on a cohort. In that study, and using total B cells, we already found differences in methylation between the two identical twins. And we can see on the right the genes that were hypermethylated in red for the twin that had CVID. And, well, the genes that we observed in this set of genes hypermethylated were potentially relevant for B cell biology. And also when we looked in a cohort looking at naive and switch memory B cells, we saw that the alterations were occurring mainly on the memory B cell compartment, as you can see there on the right. You can see that the CVID, which is always the right bar, was more methylated for the memory B cell subpopulations. So that study, we followed up on that study, and we recently reported a more comprehensive study where we performed single-cell methylomics and transcriptomics, also ATAC-seq, to investigate again this pair of twins and then a cohort. So in this study, again, in the same patients, you can see here the healthy individual on the left panel. We can see that the switch memory B cells demethylate on the healthy sibling, and on the CVID sibling, there's a hypermethylation. You can see the outer ring that is more methylated than in the healthy control, and this is affecting almost 4,000 regions in their genome. So there's around 4,000 differentially methylated regions that are more methylated in the CVID sibling. And we can see several examples of genes that demethylate in memory B cells and do not undergo demethylation in the CVID sibling. So in that study, which is more, we were able to obtain more comprehensive data, we identified many regions and genes that are relevant for B cell biology. And however, 3 minutes. We couldn't find initially transcriptome changes in the peripheral blood, which was a little bit disappointing. But at the same time, we saw that the regions that become hypermethylated in memory B cells associate with chromatin regions and changes in the transcriptome that seem to associate with the germinal center. So, and perhaps looking at peripheral blood, we were not looking at the right material. So we needed to look at B cells from lymph nodes, which are not easy to obtain from a pair of twins that you are studying for several years. You need to have a good reason to obtain lymph nodes. So since we couldn't do it, we did activation in vitro using a direct or indirect approach, and obtained and generated single-cell transcriptomics. And by doing that, we were able to obtain, you can see on the right in blue and in orange, the different stimulations, and that they mimic the transcriptome of the germinal center, more or less. So by doing that, we were able to find in the CVID sibling lots of genes that were dysregulated, around 3,000 genes more expressed and 300 less expressed, which correlated partially with the DNA methylation data, but also with this approach, we had obtained not only the transcriptome of B cells, but the single cell analysis allowed us to generate the transcriptome of all cell types. So there are tools to use this data and infer potential alterations between ligand and receptors of different cell types. This approach that we used is called the cell phone DB, which infers potential alterations in the communication between B cells, if we are focusing on that, and other cell types. With that approach, we were able to see that in activated memory B cells, that you can see on the right, there's a lot of defects in interaction that perhaps are responsible for the changes in DNA methylation and perhaps other epigenetic modifications that are affecting the behavior of these cells. And in fact, with this approach, we were able to reconstruct many of the pathways that are altered in relation of the communication between B cells and other cell types. So I'm going to just leave it here, but basically these results were obtained for patients. We also validated this data with a cohort of 10 CVID patients and control, but of course it is important to consider whether those alterations that we are able to identify also occur in patients with mutations, or if the range of alterations is different. So we are now expanding these studies to larger cohorts. We are also making an effort to analyze relevant tissues like bone marrow, which is very important for those patients with mutations, and lymph nodes, so we could explore these alterations in the germinal center. And of course, we are trying in the end to use this information to identify novel biomarkers and druggable pathways. And with that, I will just thank my team and our collaborators. Thank you.
Thank you very much. Very interesting talk, Esteban. Any questions from the audience?
Thank you, Esteban, for this nice talk, very nice results. So I'm wondering, especially seeing this last study about CVID, these CVID twins, so it seems clear that the epigenetics play a role in that case. But my question is, these differences in the epigenetic signature between the healthy twin and the CVID affected twin may be genetically determined. So, in other words, why the affected patient do this different methylation than the other unaffected? Yeah. I think maybe it's a difficult question, but...
Yeah, it's difficult. I mean, I think, well, we sequenced the two patients in the end, so we performed whole genome sequencing. So, well, I mean, it's the first time that we do that because we are not a genetics laboratory and we get some guidance to analyze the results. So it seems that some of the two individuals potentially have a predisposition to develop CVID. So the genetics, of course, is playing a role. Then, so we were trying to find potentially differential variants between one individual and the other one. So we couldn't find any pathogenic variant at the level of that us and our collaborators could maybe, it would be interesting to see that. But we couldn't see differential pathogenic variants, but we also saw that both have variants that potentially could predispose both individuals to develop. So of course the genetics is there, but maybe there are some events that occur initially to one of the individuals and not to the other that generate a sort of loop of accumulation of epigenetic alterations. We don't say that these epigenetic alterations necessarily play a role in the development of the disease, but definitely are there. And they have transcriptional consequences in many genes. So it could also be that changes at other levels. So it seems that so this in vitro analysis that we did with the activation looks like the place where the dysregulation is generating is during activation. So this is why it's relevant to obtain lymph nodes and see what is characteristic of these patients, no? That would impact the epigenomics.
Thank you. I don't know. I have time for a second question, very brief. Yeah. So my second question, very brief, is you show very nice studies comparing cases with controls, or in this case, a healthy twin versus a CBI twin. How do you think that these epigenetic tools can be applied for a single study patient? So when you have a patient in which you don't find a genetic cause, how do you address the possible role of epigenetics in single patients?
Yeah, in single patient, I think in contrast to maybe the information, I mean, you have a similar case maybe with some genetic mutations that are inactivating, so they inactivate the enzyme, or it's a black or white, no? In this case, ideally you would like to have a 100% zero, and that, for instance, in cancer, is something that you can approach. So there's, for instance, for the treatment of glioblastoma, so one of the tests that is performed now as a routine is the methylation status of the MGMT gene. So if you get that the MGMT gene is 100% methylated, so you know that the patient is going to be a poor responder of Temodal. In the case of immune conditions, it's more complex because usually you don't have a black or white situation. You don't have you have something in between. But definitely it's very important to perform studies on cohorts, on individuals, and then on specific cell types to see whether we can identify our MGMT for a specific condition.
Thank you.
Thank you. So while we're trying to connect Yuval, I would like, because I forgot to say it this morning, so I really would like to sincerely thank all the people who have been here from yesterday to this morning very early to install the video. So thank you so much. I would like to thank also deeply, I don't think she's in the room, Jo Vanken has been of tremendous help to do this workshop. Lazaro, Lorenzo is helping a lot. Melanie Migo and Maya Schrabier also. And now we're going, hopefully, yes, Yuval is here. And my fault, I didn't say that we had people online. And thank you so much for the effort and trying to have this presentation. So Yuval is going to talk about Silinco program as tool for identifying diseases causing variant. Yuval has in his own lab of human disease genomics and computational biology at Mount Sinai in New York. And it's a great pleasure. I'm sad that Yuval is not here today, didn't join in person, but at least we'll have the pleasure to listen to him by Zoom. So Yuval, hopefully, can you hear us? Yuval, can you hear us?
Sorry, I think I have some sound issue. Anne, can you hear me?
Yes, we can hear you. Can you hear us?
Good. Thank you, everyone. So thanks, Anne, for the introduction and the invitation. It's great to be here. Sadly, it's remotely, but hopefully next time I will see you all in person. So I will talk today about two new methods that we developed relatively recently to detect new pathogenic genes in small cohorts of patients, and also to detect the functional consequence of mutations, if they are gain or loss of function mutations. So this is a slide that I was showing for a long time, and the reason that I keep showing it is that it's still, after probably 10 years, it's still relevant in the same way. We have a single patient or a group of patients, and then after trying to filter out for the best candidate genes, we are left with a long list of genes. Only one or very few of them are disease-causing in the specific patients. And it's a classical needle in the haystack problem. Which of all these genes is the one that is containing the variants causing disease in the specific patient? And it is still in many cases very intuitive the way people are going to really identify what is the causative gene, and it's usually by trying to relate the candidate genes to genes that are known to be disease causing, because usually the pathogenic genes are more or less in the same biological pathway for the same disease. But to do it manually is difficult, and in most cases, at least in genetically heterogeneous disease, the known disease-causing genes for the specific disease would not contain interesting variants. So we need to apply computational methods to try and resolve it. So just as an introduction to the new method, I will show you a method that we developed a few years ago called the Human Gene Connectome, where just briefly, it's taking the same approach like navigating between two positions on the map, but we do it between any two given human genes. So you can see at the bottom a very big network of all human protein-protein interaction based on empirical biological data. And what we did was that on top of this network, we applied the shortest distance algorithm to find the shortest distance between any two given human genes. And by that, we have something like a roadmap between any two given human genes, not only the directly connected, but any two of them. And by utilizing this data, what we can do is when we have a patient with a list of genes that we want to make sense of, we can prioritize them by ranking by their biological distance to the genes that are known to cause the same disease. And over the years, we have shown that it's a very effective method to use for single patients. But then what if we have a group of patients, especially in small cohorts, that we want to try and identify the pathogenic genes in this cohort of patients? So there are methods that are trying to handle it, like SCAT, and many others that are doing gene associations like GWAS and gene burden like SCAT. But these methods are requiring a very big number of cases and controls to be able to be genome-wide significant. And also they are relying on genetic homogeneity. Basically, these methods are looking for specific genes or specific variants that are containing many more cases compared to controls based on basically frequency. So this method would not work if, for example, we have a cohort of a very rare disease of 100 patients, and let's say 10 of them have a mutation in a specific gene. That would almost never be detected in the traditional methods. But what we know that I've shown you before in the human gene connector method is that in the case of a genetic heterogeneity, we can assume that in many cases the pathogenic genes would be functionally, biologically closely related to each other. So what we've done was to build on this hypothesis and then to build the network-based heterogeneity clustering, which is shortly called the NHC. And this method is thriving on genetic heterogeneity, basically on different genes that belong to the same pathway in the same group of cases. So the base strategy is basically like the human gene connectome, which is the biological relatedness between two given human genes. And based on that, what we do technically is in this demonstration of 5 patients, each one contains a few genes with candidate variants. We start with gene 1 in patient 1. And then we ask the question, which of all the other genes is most closely related by biological distance to this gene A in patient 1? And then we find that it's gene A, it's the same gene A in patient 3. And then we continue the process. Which of the remaining genes in all the other patients is closest to gene A? We found that it's gene B in patient 2, and then we continue until we find the shortest, the smallest, densest cluster of genes around gene A. And then we repeat the process for gene X in patient 1, and we build a cluster of genes around the gene X. And we repeat the same process for each gene in each patient until we have this number of small biological clusters. And then we rank the clusters by their size and by their p-value related to specific enriched biological pathways. Sorry. Yep. So one way to demonstrate this method was to study herpes simplex encephalitis. So we had a cohort of 120 patients, and of them, 13 patients had known pathogenic genes. So we wanted to see first if we can detect these 13 pathogenic genes, and second, if we can detect novel genes, and the answer was yes in both. That was done, I should say that this was developed mainly by Peng Zhang from the Casanova lab, and that was a great collaboration. And the HSE study was done with Shenning Zhang. And what we've done was to detect firstly that indeed the 13 HSE known genes in the patients were detected in the first most significant cluster. And then when we removed these patients from the analysis, the same first cluster appeared, which was very reassuring. And we also showed by poly-IC that 4 novel genes are likely to be HSE causing also in the first cluster. So that was demonstrating the utility also in a small cohort of patients where traditional methods cannot work. And then we moved on, and these are just preliminary results showing analysis in severe susceptibility to COVID-19, which is very well studied. It was shown already that genetics play a very major role in the pathogenesis of severe COVID-19. And what we've done here was to take 188 severe COVID-19 cases and 1,000 controls from Mount Sinai Hospital's BioMe biobank. And we tried to compare what are the results when we use a SCAT analysis, the more traditional one, and the NHC, which is the method I've shown you. So when doing SCAT, we get the usual suspects, the interferon pathway genes, which are the ones that are currently known to be related to COVID-19 susceptibility. But when using NHC, we see a completely different complementary pathway. So actually the most significant pathway was enriched in single nuclear RNA, SNOR RNA, where very interestingly, the Casanova lab has shown a few years ago the first genetic etiology of SNOR31, which is a gene that is shown to have to confer susceptibility to herpes simplex encephalitis, which is another severe viral susceptibility. So that is a nice way to show that we really see complementary results by using the different approaches and not seeing only the most like the usual suspects. And then we demonstrated it in inflammatory bowel disease, IBD, which consists of Crohn's disease and ulcerative colitis. So firstly, we use this only for IBD, then in Crohn's disease, and then in ulcerative colitis. So we've shown that the IBD and the Crohn's disease are having the same first pathway enriched, where interestingly the CD was having a much better p-value because most probably the IBD main cluster was driven mostly by CD and not by UC. And it's also biologically relevant. It's shown to that NADPH is closely related to Crohn's disease. And also in ulcerative colitis, we see a completely different pathway, which is the mitochondrial complex. And it's known to be suppressed in ulcerative colitis patients. And it's also causing increased permeability in the intestinal epithelium, which is very relevant to ulcerative colitis. And this analysis were done by Echikars, who is my postdoc. And then we moved on to do the same analysis shown here for IBD, but to do it in a population-specific manner, only for African Americans. Sorry, that's a mistake here. So on the left, it's African Americans, and on the right, it's Hispanic patients. So we wanted to see if there are specific signals that are specific for African Americans and for Hispanics. So what we've done first was to do PC adjustments of cases and controls to remove outliers by different covariates. And then we see that, firstly, the most significant cluster of genes enriched in African Americans is upregulated in collagenous colitis, which is a disease very closely related to IBD, so it's likely to be relevant. And in the Hispanic specific, basically the cluster shown only in Hispanics, the most significant cluster was upregulated in IBD patients in the colonic glands, which again is suggesting that it's very relevant. And for the shared most significant cluster that is showing up in both African Americans and Hispanics, it is a cluster that is also shared with the Ashkenazi Jewish ulcerative colitis cluster that I've shown you before of the mitochondrial complex. So again, it's showing that some genetic causes here are universal and some are population specific, which is again showing why it's important to sometimes focus also on specific populations and not not taking all patients that we have from the specific disease. And now a relatively sharp change into another method that I want to show you, which is attempting to generate the first computational method to detect if a mutation is causing gain of function or loss of function. So currently it can be done only in the lab to make this distinction. And shown here in an image generated by Stephanie Boisson-Dupuy is the gene STAT1. And in red, we see the gain of function mutations clustered on specific domains in the protein, and the others are loss of function clustered in other domains of the protein. And importantly, gain of function and loss of function mutations are causing different types of disease. So it's very important to try and estimate what they are. So what we first did was to generate the first large-scale database of gain of function and loss of function mutations. And we've done it by machine learning. And then by using many different variant level, gene level, and protein level features, we try to find which of the features are best to separate gain from loss of function mutations. So I will just show you a few of these features that are specific to gain and to loss of function mutations. So for example, we see on the left here that gain of function mutations tend to be much more of a frame shift, while the gain of function mutations tend to have a relatively more moderate impact. And one of the major features that were shown to separate gain from loss of function were the number of paralogs. Basically, how many gene copies the gene has that is containing the specific mutation. So the loss of function mutations tend to be in genes that have no copies, basically in single genes, which makes sense because if this specific gene would lose its function, then it would basically not have any functional copies. But for gain of function mutations, they tend to be in genes with many paralogs, more than one. And that also makes sense because losing a functional copy here might enable the other one to develop a new function and to cause this gain of function. And it was also shown in evolutionary studies. And also, like we know, there are different modes of inheritance, like autosomal dominant that is related to gain of function and recessive that is related to loss of function. And there are also different amino acid substitutions that are specific to gain and to loss of function variants. And if you would like to access this database of all currently known gain and loss of function mutations, you can do it sorry, I think I was disconnected. So let me just quickly wrap it up. Can you hear me? Okay, good. Right, so let me just wrap it up by the new work that was done by David Stein, which is actually creating this classifier, and that is the part that is unpublished yet. So what he did was to first use all the abstracts from the human gene mutation database, and by that extracting additional gain and loss of function mutations, and then also with neutral variants to train the data using LightGBM, which is the machine learning algorithm that we found to be most effective. It was more effective than random forest and deep learning for this specific task. And the output is basically the probability for any mutation to be gain of function, loss of function, and neutral. And we also pre-calculated all possible human missense mutations for estimating if they're GOF, or neutral. And this is showing by LDA clustering that it can very nicely separate GOF from LOF and neutral. And this is a raw curve also showing that it is by far the best performing method to separate gain of function from loss of function. And it's, to be fair, it's the only method so far that was attempting to do it seriously. And lastly, very quickly, we're currently working on a new method to detect pathogenic mutations based on the disease they're causing. So what we do here is to train the pathogenic mutations based on groups of the mutations of the specific disease group. So it can be immune system, cardiovascular, and others. And we are showing in very new results that are, again, we show very nice separation of mutations by disease group. So I think this is very exciting, but I don't have time now to talk more about it. I'll be happy to take questions if you have any. So thank you for listening.
Can you hear us?
Yeah, sounds good. Thank you.
Okay. One question?
So are there any question? I have no, because I was running a little. Okay, thank you.
That was fantastic work, Yuval. Congratulations, very nice. I also have a question on the gain of function. So if you train your set, I think we still are completely underpowered there. So you took the best available set globally of known gain of function mutations in genes where the gain of function is usually also well described and usually functionally even proven. But it could be that there is a second class of genes for which the loss of function is already very well established, but they may look slightly different from the already established gain of function mutation causing genes, if you know what I mean. So how much does your gain of function set bias towards the discovery of completely novel gain of function, or is it at all possible?
Yeah, it's a great point. So what we try to do to address this problem is to try and validate this algorithm on new mutations of gain and loss of function. And the performance is getting worse when we use new genes, but it's still very high, and it's still much better than other methods doing it. So basically, when you have let's say a completely new gain of function in a gene that is not yet described, it would just try to compare it by the machine learning method to the patterns that it sees in other gain of function mutations in other genes. So it might be incorrect, but it would just look for basically the similar the most similar related gain of function. And it's also the case if it's in a gene that is already well described. So if you have, for example, a gain of function, a true gain of function mutation in a gene that was trained only in loss of function mutations, then we might be, and we're actually likely to be still able to detect it, because it would look, here it would look on the characteristics of the gene and the protein level, for example, in which domain it is, and what is the functional impact of the mutation, and what are the tissues where it's expressed, and so on. So there is a good chance it would still be able to detect it, although the performance would likely be somewhat reduced here.
Yeah, fantastic. So, I mean, that's what we all need. So keep on pushing your tools. I think that's extremely useful. Just one additional question. Have you considered to take haploinsufficiency scores or susceptibility for haploinsufficiency? Oh. Okay, great. Yes, perfect.
Okay.
There are questions.
Yeah. Yeah, I'm here.
One more related question, so thanks so much for this. To be even more precise on the loss of function, let's say, sensitivity of a given gene, do you also take deletions into account, like Matthew Holtz does for this haploinsufficiency scores? Because different from loss of function point mutations, for full gene deletions, we are much more sure that it's really a dosage effect than for point mutations.
So I'm not sure if I completely heard, but if you referred to a full gene deletions or large scale, large scale variants, we didn't use it here, but we want to integrate it in the later version of that.
Oh yeah, that's a terrific idea. Great.
Thank you.
So is there any other question? So Yuval, thank you so much. I know that it's a day off in US, that you had to, it's 6, you started at 6:15, so we really appreciate. Thank you so much and see you soon. Bye-bye.
No problem. Thank you. Bye.
and we're just going to have a brief talk from, is it now? BioNano? Yes? No? No. So we'll do it later, but I just want to tell you, go and visit, would you? Late. So do you prefer now, or everyone wants to have a break and just come back 15 minutes before to listen to Dana? Okay, so thank you so much, and again, don't hesitate to visit the stand by with BioNano and PacBio, and have a nice lunch. Thank you. Fungin, which is not the good pronunciation, who's going to talk about the human phenotype ontology in immune-mediated disorders. Marielle is from the Department of Genetics and the University Medical Center from Groningen. And Marielle, you have 20 minutes.
Okay. Thank you, Anne. Well, let's go then. So for the human phenotype ontology, for the people who are not aware what human phenotype ontology is, HPO, I will call it like that from now on. It's a way to describe phenotypes bioinformatically, more or less. So it's an ontology. That means there is a structure there's a relation between the phenotypic descriptions. So you can see it for height over here, and below the height is a short stature or tall stature, or another way how to explain it, I use sometimes the leg. The leg is higher in hierarchy than the knee or the foot. So you would say the higher in the hierarchy is the leg, and that part of the leg is the knee. So then you describe your leg in an ontology-based way, more or less. So HPO is invented by Peter Robinson. It has more than 80,000 terms so far. It's a hierarchical structure, like I already mentioned, and it's also often used nowadays to link to disease databases, and it's actually nomenclature used in many genetic and database descriptions. So anyway, this is a translation of language of your phenotype, and because it's bioinformatic, you can use this one in bioinformatic tools, and you are not disturbed by any language differences, so you can translate it into different languages, and well, that's all an advantage of using HPO in the description of your phenotype. So what you can do, what are the benefits of HPO next to the regular descriptions of the phenotype we have? Well, it's a unifying nomenclature of the patient, so you can use it for data exchange more easily, so that you are aware that you describe the same phenotype the same way to a patient and to a doctor at the other side of the ocean, more or less. It's also used in databases and publications, so you can retrieve which phenotypes are being used. You can use it to develop bioinformatic algorithms to find the similarity of your patient, the phenotype of your patients to the disease, which I have some examples for afterwards. And what I focus most on is you can use it on variant pathogenicity prediction, or more or less a gene discovery. So you can use it to say which gene or which variant is most likely causal for your patient. So there are, we've heard this morning already lots of talks about variant interpretation, what the guidelines say, how you can use algorithms to interpret variants. But what comes down to mostly is that you have a patient, and what we are used to is to sequence the patient, and then you have a lot of bioinformatic algorithms and new tools being developed to predict if a variant in your sequence is pathogenic or benign. And there are more and more also AI-based, machine learning-based algorithms developed to prioritize actually your variant candidates. But and to the other side, you have the symptoms, and the symptoms now can be described with HPO. It's more often being used for other rare disorders, like intellectual disability, for instance. And then also tools can be developed from your description of your patient, your phenotypic description in HPO, to see if based on that, you can come to a disease candidate of your patients. And then in a multidisciplinary, usually, meeting, you can then describe which mutation, which gene might be causal for your patient, and then come to a diagnosis. So how does it actually work? How would you like it to work? Such an algorithm is that you prioritize genes using HPO. You have a phenotypic description of your patient. The relevant phenotypic features, you look up an HPO and you make the HPO terms from them. So this is a patient with CVRD. And then, so it has chronic sinusitis, all these different descriptions. You can search the HPO terms for that, put it in an algorithm, and then just ask the computer, just find all matching disease genes from all literature and databases, and could you also please prioritize the genes? So you don't have to do a lot of work anymore, the computer does it for you. But although it's very easy to put it on a slide, it's very difficult in daily practice. And that's why there are actually several HPO-based tools. And we compare them. We compared 9 of those HPO-based tools to see how well they performed. And this is not specifically for immune-mediated disorders, but these were general patients, so 305 clinical patients. We used 305 clinical exomes from patients with a wide phenotypic spectrum, and we knew there were 308 causal genes in there. We compared, so those, all the different colors, those different bioinformatic tools, and then, of course, what you would like a tool like that to do is you would like to find the gene. That's the most important thing. I think if the tool doesn't find the correct gene, that's something to be worried about. But also the total number of candidate genes is important, and the ranking of the genes is important in that way. So we compared them all in this cohort of patients, and that you can see. I can see here. Is that? You don't see my mouse, do you? No. At the X, Y, S, you see the total number of candidate genes returned. Yes, how do I? Okay. Thanks.
Dan werkt hij niet meer klikken, dan moet je nu die arrows gebruiken voor de volgende slide.
Yeah, top. Thank you. Thanks for your help, Alex. So what you see is they all, the different tools, first of all, some of them miss genes, and not that many. Some tools Gado didn't miss anything, but some missed quite some genes, so you don't want to use that in that case, but maybe for a specific subset, it's not that hard. And you see that they have different amount of genes they come back with. I mean, even 40,000 genes that your correct gene is in there, well, luckily, but that's not, it's still a search to perform to find your candidate gene. And if you then look at the ranking of the genes, so the rank of your real gene should be 1 in the ideal world, then you see that some of the genes are indeed ranked as 1 by the different tools, but lots of genes are not ranked that good. But then you use only the HPO terms, and then if you, we try to compare it to the clinical practice more or less. So already based on frequency and other IDs, you filter out certain genes or certain variants, and then we simulated clinical practice by spanking in the mutation of the specific gene in 20 random genes from the exomes from these patients to see if from these 20 genes the genes were prioritized. And there you can see that there is indeed that most tools actually perform more or less the same, a bit more or less. There are 2 lousy tools, what you see here, but the rest is actually more or less the same. Even also the recently developed Lyrical, which I will, by Peter Robinson, which I will talk about later a bit, also is in this area. So in the end, they all perform more or less okay, is what you can say. Oh no, I have to do this. Yeah. So nice. Why don't we in the immune-mediated diseases use HPO so far? Because actually, the HPO terms for these disorders is really not so good. It is really bad. The terms are missing from the hierarchy, and so if you cannot describe a patient if you don't have the term to describe it. The structure is incorrect. So if your leg is linked to your hand, that doesn't result in the right genotype, of course, then. And many diseases are just not annotated yet, so you never come to the disease. Terms from the disease are missing, or terms might not be accurate. So there's one reason why the use of HPO in our disorders is not that frequently used. So that's why actually Eir and Rita and the ACID had the initiative of bringing together clinicians and geneticists, immunologists, computational biologists to start curating the HPO tree for immune-mediated disorders. And we did that in meetings, but we did also on online things. We started in 2018. We were all invited to the lab of Kahn Bustuk in Austria, and we had 2 meetings there, one year later, and then COVID started, so we started to work online as well. So what we did actually is, so we had working groups because, I mean, we heard about 240 genes. Diseases are very difficult to tangle, so we based ourselves on the WES classification, and we started out with 4 working groups: antibody deficiencies, autoinflammatory disorders, mixed immune-mediated disorders, and well, I can't read it from here, but I hope you can read it. So what we did was, so we had these 4 groups, so the clinicians had it all put in 4 groups, specifically also for the antibody deficiency, which was really necessary to restructure the tree. So that's what they mainly focused on. So see if the tree is correct, really hands-on, as you can see here. And next to that, all the working groups also performed re-annotations of the immune-mediated disorders, also by machine learning, by collecting the right literature, getting the terms from there. And then, based on machine learning, collecting more and putting the terms correctly. After that process, we got judged by 2 independent specialists, so the personnel from 2 different centers as well, to have accuracy probable. And then, if they didn't agree on if it was correct or not correct, the terms, or should go out or not, we had some meetings again to discuss those. If the terms were correct or not. So that's what we did, and we published that already, and we restructured then 18 branches of the tree. We completed it. We requested, because it's a website, it's the JAX lab who does that. We requested those changes. We added missing term, and we created over 82 diseases. And then you see that indeed what we did indeed, you have now many more terms per disease than before, and also we restructured the tree, as you can see here. So, but the most important thing is it was a lot of work. Is it worthwhile to do so, because it doesn't result in any change? And luckily it did. If you then look at disease similarity, so if diseases match together, if you go to your left-hand side, and you can focus on the easiest part, is this one, the most diseases we had. It was from autoinflammatory disorders and immune dysregulation. And you can see here they're scattered all around, if there is some immune-mediated disorder. But after the curation step, they neatly almost, of course you need some discrepancies, but they matched neatly together again. So in really, the diseases matched again to each other, so it comes closer. So if you have some disorder there, then you have a higher chance of finding indeed the right gene in there. So we did the work, and it improved the disease similarities, but did it improve performance of matching patient phenotype to disease? So we gladly we could use a cohort of Sophie. And we added the HPO terms to it. We used Lyrical as a bioinformatic tool. So we used old HPO disease annotations, so before we started the curation and afterwards. And what we indeed found was that afterwards, more and more, you then get a list of candidates per patient. And what you see is before, well, almost 10% matched the disease ranking of 10. So the first 10 genes which the tool said this could be the gene was only in 10% of those cases. And after the curation step, you go to 75% of the patients then go to the first ranking of the genes. So it did improve, and I think we made it worthwhile to use it more often. We are currently doing some more work on autoinflammatory disorders. So the subgroup for autoinflammatory disorders went on. They went on to curate also the last 10 to describe the last 10 diseases from the recent WES classification and the 2 new disorders. And we also, curated that, that we had to first described, of course. And then what Lyrical does is if the likelihood ratio increased after our annotation. So what Lyrical does is it predicts the likelihood that this gene is disease causing for this patient. And so what we did for our, now it's not finished yet, but from the 82 cases we just did, was for that indeed the likelihood ratio increased for 37% of the patients, so it did have effect. And more importantly, it didn't decrease the likelihood ratio either. So if you can also imagine that if you put in more terms, that it dilutes the right disease, so that the likelihood ratio also dilutes as it gets, but it didn't, so it only gets better or it stays the same. So that was a good thing. And we, from these patients, we also tried to do some WES analysis and see how that performs, because to get all these data together in one spot between the different hospitals is difficult as well. And for verified patients, it did increase. And if you then now see the autoinflammatory patients, which are rather difficult, because the phenotype is so broadly, so heterogeneous, and many genes are involved. You can see that now with Lyrical, after the curation step, most of the patients, so you see the different, the other 2, FMF, Merkel-Wells syndrome, TRAPS, and VEXAS syndrome is also in there, that you see that most patients now end up within the top 10 of the ranking of the genes in this exome-based gene list. So that it's not exome-based. Based on the phenotype, it now ends up at the top 10 of these diseases. And of course, what you can see is that some Merkel-Wells patients, some patients do not match the phenotype either. So it's not the holy grail yet. It just doesn't match there. And also here for other diseases, you see FMF is still difficult, that many patients still are difficult to based on the clinical symptoms, rank the disease properly. And just the last thing to show is that there is an added value, because what I showed you were just HPO terms, and based on HPO terms go to the disease-causing gene. But if you combine the HPO terms with your genetic data, then it is a lot better. Here we described, I think you know, does anybody know which disease this patient has? It's a male of 60 with those symptoms. Must be very clear, because we already it was already mentioned. The disease was already been described previously and mentioned before. But then Lyrical performed without the genetic information we had. It already performed rather okay. It mentioned that the rank first was VEXAS syndrome, which this patient indeed had. But what you can see here is that 21 genes came up, 21 diseases came up. So then you still have to decide which one is the best one. And here I only showed 2, but there were 6 genes which matched for full 100% to this syndrome, and they only the likelihood ratios only very small. So you still, without genetic information, it is still difficult to prioritize. But already it's done a good job. If you then add the whole exome sequence of this patient, again, to combine it, then you can see it only ends up with 3 genes, 3 disease-causing diseases, and it matches for the full 100% with VEXAS syndrome, and the other not so much, even a negative score. And also the likelihood ratios really differ between them. So I think what I hope to have told you, that using HPO and a curated HPO in your genetic analysis might add a lot in these disorders. But then we need a good HPO, of course. So the curation of HPO, which we've performed so far, is done really well. You can identify diseases from these symptoms. It improves genetic analysis. So your genetic analysis might go faster, which is important for diagnostics. But also, what Isabel already mentioned, what she didn't continue in, is the incidental finding, because sticking to the phenotype of your patient might also avoid incidental findings in this way. And so now we curated the immunodeficiencies partly. We only did 4 subtypes of the WES classification. And it did work, but the question here is how would we continue? Because that's rather hard. You ask a lot of people's free time, and you really need expert, of course. You need expert opinions, and the expert are usually the people not with a lot of time to perform these issues. And so we would like to link it to an official organization and find people with more time and expertise in the specific diseases to help us with this endeavor. It's really an endeavor, but I think it's worthwhile. And this is my last slide. I would like to acknowledge all the HPO consortium. Matthias Heimel, Julia Pasmandi, and Kaan Boestok were most prominent in the first paper we published, but of course with all the help of all the friends. Peter Robinson is there, and some people continue to work on the autoinflammatory disorders I mentioned there. And I also would like to thank my colleagues at the Department of Genetics at the UMCG. And I would like to thank you very much for your attention.
Thank you, Marielle. We have time for one short question.
I have a very quick question. Is there a way of automating the extraction of relevant terms from clinical notes or letters?
Yeah. We didn't use them, and there must be. I think there are. Kimberly wants to answer that. So there must be. There are tools for that, but none of them our hospital is using, but they are being developed.
Yeah. So there's for people who have Epic, which I think you have, Kumar. There's something called Fire, and it's like an app that can go over the Epic notes and pull them out and turn them into HPO terms. It's not as slick as it could be, but it's better than having to do it all by hand. Exactly.
Yeah. Any other question? Yes.
Thank you. Very nice talk. Quick question. I'm just wondering, I'm here. In this system, whether it's possible to counter-indicate some kind of abnormality, like let's say with normal monocyte or something like this.
Yeah, I think the HPO Lyrical does allow for that. I agree. So yeah, you could use it. So, but by describing a patient, we describe patients with HPO. We describe diseases with HPO, and there you don't want to. It's not a classification system. So for your patients, you could do that, but for describing the disease in which we did, you don't need to do that, because then you might be wrong. Yeah. Okay.
Are there any interaction with ClinGene or not at all?
No, we're looking for some interaction, not with ClinGene, I think, with the WES people, with the expertise there. We're looking for interaction, but it did not result in concrete things then. And it's for free for everybody. I mean, already now you can do it yourself if you want it. HPO and Jack's lab accept everything from not everybody, I don't know that, but so everybody in small communities, you could do the same thing. So you don't need to wait for other people or for us to do it. So and to improve things.
Okay, thank you, Marielle. So our next speaker is going to be Jérémy Rozan, who is going to talk about the identification and study of copy number variation. So Jérémy is working in the lab of human genetics of infectious disease. And so Jérémy, you have 20 minutes. I'm sorry, I told you it's 30 minutes, but it's 20 minutes to present. Thank you.
Hi everyone, I'm a clinical immunologist from a clinical lab. I'm not an bioinformatician, and I thanks a lot the organizer to give me the opportunity to share with you what I've learned about copy number variation while investigating patients with suspected inborn error of immunity, either in clinical lab or in research lab. So copy number variation encompasses both large deletion and large duplication, which are duplication or deletion of above at least 20 base pair. This copy number variation belongs to structural variation, and these structural variation are favored by homologous region in the genome. And a population genetics of this structural variation was conducted 2 years ago by the Genome ID group, and they found that at least 200 predicted loss of function structural variation are present per individual. And they also found that at least 20% of predicted loss of function variant in a single individual are caused by structural variation, which are mainly composed of copy number variation. And this result is quite striking, because if we have a look at all Mendelian disorders that are registered in IGMD database, copy number variation only represent around 10% of reported mutation, and it's roughly the same for gene of inborn error of immunity. So for some gene of inborn error of immunity, it's known that it's much higher. This copy number variation were, for instance, frequently reported in patients with Artemis, Docate, Fanconi anemia, or EGHM immunodeficiency, but an obvious statement that we can get from this population genetics data is that CNV are probably underdiagnosed in patients with suspected inborn error of immunity. So there are different methods to evidence CNV. The oldest one are PCR-based method. The first one is a targeted PCR. So targeted PCR, to conduct a targeted PCR, you have to know approximately already the breakpoint of the copy number variation that you are looking for. So it's mainly for a confirmation technique or to screen for recurrent copy number variation. You have to be careful while doing your PCR, because your PCR may be allelic and selectively, for instance, amplify the wild type allele. And in that case, you may wrongly think that the patient is homozygous wild type, despite, for instance, as in this case, carrying a heterozygous deletion. Another assay which is commercially available also for clinical lab is MLPA. It's a PCR-based assay that gives you a score. When it's close to 1, it means that there is no deletion or duplication. And when it's below or upper 1, it can suggest a duplication or a deletion. So there are few commercially available probes with also quality control available, which is interesting for clinical lab. And there is also an option to make homemade probes. Also, another method is qPCR-based method, perform in-house qPCR-based method performed directly on genomic DNA. This method is quite interesting because it's quite cheap and it's sensitive enough to screen for heterozygous or homozygous copy number variation. So you can also screen for copy number variation through high throughput sequencing. There are different methods. The first one is either a targeted sequencing of a whole locus if you have a strong candidate gene, or the sequencing of a defined set of genes. This is what we call a panel, or whole exome sequencing, or whole genome sequencing, either a short read or long read method. And you have to do, with this assay, a specific calling to call for copy number variation, usually with the usual pipeline. The usual pipeline call for short mutation or single nucleotide variation. You have also to keep in mind, as Isabel Mates mentioned earlier this morning, that with this high throughput sequencing, except maybe for long-read whole genome sequencing, some region will have a lower or incomplete coverage. For instance, GC-rich region, which are, for instance, usually frequent in first exon of genes, Also, stretch of bases or repeated element will be less covered, as well as alignment of pseudogene is challenging. And besides this high throughput assay, you have also cytogenetic studies that you can perform, either targeted cytogenetic study, such as FITCH, to screen for Digeorge syndrome, and various other methods with various resolution, either karyotyping, CGHRA, or bionano, that I think will be presented later, and that will display a very high resolution. So that being said, I will now share with you some examples from my experience and also from the literature about how to screen for this copy number variation. And so this first example starts with 5 different kindreds that were referred from all across the world, and patients from this kindred all display mycobacterial disease, and they had a very strong phenotype of Mendelian susceptibility to mycobacterial disease. And we investigate this patient by assessing their expression for IL-12 R-beta-1 on cells of individual of patients of the various kindred, and this expression was abolished, suggesting, of course, a deficiency, an autosomal recessive IL-12 R-beta-1 deficiency. However, by performing classical Sanger sequencing, we could not have a clear explanation of their underlying mutation. We thus decided to conduct, in collaboration with the genomic platform here in Imagine, a targeted high-throughput sequencing of the IL-12 locus and on its flanking region. So it's like a mini exome or a mini genome, but specifically on around one locus. So it's quite cheap, and it will give you two kinds of information. The first one is the number of reads per base. And as you can see in kindred A, patient 1 didn't add any read in the first 7 exons of IL-12 R-beta-1, and it was confirmed that this patient displayed a homozygous deletion, whereas the coverage compared to the control in his parents was half reduced. Similarly, in kindred B, C, and D, we found half decrease coverage of different exons, and we could confirm in this patient through targeted Sanger sequencing heterozygous deletion. And in the last kindred, there was a 25% increase in the coverage of the patient, as well as from its father, in 3 different exons, and in this patient we could confirm from an heterozygous duplication. And so, when you do a screen for CNV on high throughput sequencing, you can screen them either on data from coverage, but also on data from alignment. And all bioinformatics will screen for this CNV either on data inferred from coverage or data from alignment. And with this targeted high-throughput sequencing of a whole locus, we get data of alignment also in intronic area where the breakpoints were. And if we have, for instance, if we visualize in this patient who carry an heterozygous deletion specifically for the read where the breakpoint is suspected, we can see that, first of all, in terms of coverage, there is a drop of coverage here, but also there are reads that there are bases that does not meet match, and it's actually reads that overlap the breakpoint. So we found several overall copy number variation in IL-12 R-beta-1 locus. A few others were previously reported, and since then we have found many more. And interestingly, this IL-12 R-beta-1 locus is full of ALU repeat elements, which are displayed here in gray. And these ALU elements are 30 base pair highly homologous sequence. And this makes the locus prone to ALU-mediated rearrangement. And interestingly, a parallel study conducted by James Lubsky, in which they address which are the locus of the genome that are the most prone to ALU-mediated rearrangement. They found among the top genes a few genes for inborn error of immunity, including IL-12, R-beta-1. So another example of screening for CNV through targeted high-throughput sequencing is displayed here. It was reported by the group of Sylvain Latour in Imagine. So it's a bit a similar story. They had 3 unrelated kindred in which they had strong suspicion of XEAP deficiency, either because of their clinical phenotype and also through preliminary biochemical investigation. So they perform similarly to us a targeted high-throughput sequencing of XEAP locus. And interestingly, they found in all 3 patients no read in 3 different deletions that all encompass the non-translated exon of XEAP. And this non-translated exon actually contain a promoter that is important for XEAP expression. So I found this finding is interesting. It describes a deleterious deletion in a non-translated region. Another example is about this patient. So this patient is a 50-year-old patient that were referred to the clinical lab, and it was explored through panel sequencing, and we found biallelic mutation in DOC8. And this was really surprising because DOC8 deficiency is a very severe deficiency, and usually patients are not alive if they die before the age of 30 years old in the absence of hematopoietic stem cell transplantation. So this patient really had an unusual phenotype of DOC8 deficiency. At first glance, we did not believe he could have the DOC8 deficiency. However, further immunological studies performed by the group of Sylvain Latour and of Stuart Tanguy found that there was a subset of lymphocytes that express in this patient DOC8, suggesting there was either a reversion or a mosaicism. And of course, they first thought that it could be the single nucleotide variation that lead to nonsense that could have reverted, but actually it was not the case. It was the duplication that had reverted itself. And this is evidence with targeted high-throughput sequencing of DOC8 locus. In bulk PBMC, you can see the deletion, but in memory T cells, there is no such deletion. So this targeted high-throughput sequencing of a locus is interesting, but of course, usually is interesting when we have first biochemical data or immunological data or very strong phenotype, but in most of cases, when we investigate a patient with suspected inborn error of immunity, we have several candidate genes. So another way to investigate such patient is, of course, panel. That usually contains all non-PID gene. And the main advantage of panel is that you usually can get a very nice coverage rate. And from that, you can with a good sensitivity and specificity, screen for copy number variation in a specific patient by comparing, by normalizing to the mean coverage of the patient in all other locus and 2 other individuals in the same batch. And for instance, in this study with the PID panel of Necker, we study more than 100 individuals and could find in some of them deletion through this scoring. For instance, here this patient display heterozygous deletion in IgHM, and they were all confirmed through other means. However, now patients are more often explored through whole exome sequencing. So screening of copy number variation or whole exome sequencing is very challenging because the coverage is highly heterogeneous all across the genome. Also, there is a batch effect, a kit effect. And we cannot rely at all on data from alignment to screen for this CNV because most of these CNV have breakpoints located in intronic region where that are not covered by definition by whole exome sequencing. So one of the best tools to screen for this CNV is HMZ DEL Finder that the bioinformatics lab in the research lab here improve. And it gives you a score of putative deletion across all sites. And the higher is the score, it's negatively inverted here, the higher there is a chance to have a true deletion. And this is effective to screen for homozygous or hemizygous deletion. And so while doing this study, my colleague Paul Bastar was investigating a patient with herpes simplex encephalitis that died of this infection. She also had siblings who died from viral disease. And at first glance, by looking at variant calling file of WES, he didn't identify short mutation. However, using the tool I previously showed you, he found a high score of putative deletion in IFNAR1 gene, and this was indeed confirmed. So as you can see, there is strictly no read in the patient, whereas in a control there are some read. And he subsequently characterized this patient with need display complete IFNAR1 deficiency. It's the first copy number variation reported in this gene. So really the best tool so far to screen for copy number variation besides cytogenetic study is really, as mentioned earlier, whole genome sequencing. First of all, because as there are two main advantages of whole genome sequencing compared to whole exome sequencing. The first one is that the distribution of reads in whole genome sequencing is much more homogeneous on a whole genome sequencing than on a whole exome sequencing. And the second one is that you can use data of alignment to screen for copy number variation. And even with short read copy number variation, in these studies, they could evidence a lot of copy number variation. So one important thing to keep in mind also is even if we find a copy number variation, we have to ensure that it's not a frequent, it's not a polymorphism. So there are some population genetics data available, especially from the Genome ID project. There are more than 10,000 individuals of different ethnicity. And regarding the specific base for copy number variation that can be useful, there is DGV, Decipher, or IGMD Professional. So this was for copy number variation, but there are many more of structural variations that exist. And here is an example. It's concern an MSMD patient that display very severe susceptibility to mycobacterial disease. He was originating from a highly consanguineous family. And while doing whole exome sequencing, we found weird read in interferon gamma R1 locus. Read display in gray here are bases that did not align to the wild type sequences, and it was this read were flanking a 30 base pair motif. So at first glance, we didn't really get what was going on. However, the patient looked to have a complete gamma R1 deficiency because he had no expression of gamma R1. So we performed targeted sequencing of this exon and found that the patient actually had an insertion of around 30 base pair, and while aligning this base pair, we found that it was an ALU element, and with duplication flanking, with a duplication of a 13 base pair flanking this insertion, and this is a hallmark of line 1 mediated ALU retro insertion. So this was interesting because so far I think there are less than 600 line 1 mediated ALU insertion reported across all genetic disease. And for this specific example, I think we were lucky to catch some misaligned read because probably the insertion was exactly where was the capture probe of WES. But of course, this kind of structural variation is much more better screened by whole genome sequencing. So we can screen for copy number variation on panel on WES on data only from coverage. On targeted high throughput sequencing and on whole genome sequencing, we can screen for them through data from alignment and coverage. Also, it was reminded earlier across the day, we have to keep in mind that there are complementary studies that can be performed, such as cytogenetic studies or RNA-seq also that can help to increase a bit the diagnostic yield. One thing that I think is still challenging to do is screen for mosaicism in case of copy number variation. Of course, all these variants need to be validated through functional studies. And so with that, I would like to thank you for your attention and thank the colleague. I've been discussing a lot about this problem of copy number variation across the last year. Thanks a lot.
Is there any question?
So I have one. So you were talking about this ALU sequence, and you were talking about some genes with more ALU sequence. Do we have, so is there any patient who have been reported with copy number variant? I think there was IL-12 beta 1, there was also TIC2, or so among the gene reported with...
Yes, indeed. For instance, their deletion reported in TIC2 patient and also in FUNC-A gene, and which deletion are around that half of reported mutation indeed.
And the other thing is, okay, so you recommend to do whole genome sequencing.
Yes, long read if possible, but it's not affordable really at the moment, but for sure it will be the gold standard as mentioned earlier in the day.
So it's something that people should think of, this copy number variant, because I'm not sure that that many people are looking at this in their gene.
Very nice work. Just on your deep sequencing of your custom panels, where you have obviously more sensitivity, maybe I missed it, but next to coverage, do you also consider the B allele frequency of common SNPs?
No, we did not. It was sufficient enough, the sensitivity with just coverage and also alignment, but it's
because for some other CNV tools, we now start seeing that if you use not only coverage but also the B allele frequency, obviously deletions can give you a loss of heterozygosity, so that gets more sensitivity, but also for duplications or gains, I think it may help some of our both exome and genome tools for CNV calling. So something to consider for the future tools.
So for the sake of time, maybe we move to the, thank you, Jeremy. It was a great presentation. We move to our next speaker, Roger Crowell-O'Brien, who's going to talk about somatic revertant. So Roger is from the Lab of Translational Immunology Research Group and Immunology Division, and is from Barcelona, Spain. So Roger, thank you so much.
Thank you, Anne and Marielle, for organizing this nice meeting. Do you know how it works, the pointer? Because I know Alex.
At least I have one task here.
Yes.
Useful. Come on.
You are our expert.
It's over there.
Okay,
Then
perfect.
Use the arrows for the next slide.
Okay, perfect.
The mouse doesn't work.
Okay, thank you. I have a disclaimer, which is, I think there are no experts in somatic reversion unless you have a case of somatic reversion. So this is my case. So today I want to talk you about some general concepts of somatic reversion, especially in primary immunodeficiencies, and then I will tell you a story. So I will not spend time in the introduction because Anna excellently covered what is a somatic variation or genetic mosaicism. So I will save 1 minute of my presentation. And these are two highly related concepts, which are somatic reversion, which is typically the reversion of the mutated allele to the wild type. This phenomenon is also known as back mutation. And then another highly similar concept, which is somatic genetic rescue, which is a more wide concept, including any type of mutation that affects either the gene mutated or even occasionally modifying another gene that indirectly affects the pathway that is altered by the Germline mutation. This somatic genetic rescue concept is very well covered in this review by Patrick Révy and Alain Fischer, who are in this institute. And at the end, the idea is that somatically modified clones with a fitness advantage generate a somatic mosaicism and can be detected if they expand, differentiate, or survive better than the unmodified cells, which is clearly shown in this basic scheme. Obviously, if a mutation is deleterious, the lineage do not survive. If it's neutral, it may survive, but it's difficult to detect because it will be with a low frequency. And only when there is an advantage, the cell line may expand because this somatic genetic rescue. So since we need that this population expand and survive, somatic genetic rescue is mostly detected in easy accessible, long-lived, and rapidly expanding cells, like in the hematopoietic system, which populates the blood and the immune cells. So this figure is to illustrate the different mechanisms of somatic genetic rescue that have been described in Mendelian hematopoietic diseases, and according the different types of heredity. So most of the examples include point mutations that can be back mutations, reverting to the wild type allele directly, or substitutions that may cause a less deleterious change, for example, from a stop codon to a missense variant. Then we have second site mutations, usually in the same gene, in the other locations, that sometimes are compensatory. For example, imagine an autosomal dominant disease with a gain of function mutation. You can find a somatic reversion introducing a stop codon later in the gene. So by this way, abrogating the gain of function mechanism. And even there is some unusual cases in which we can find somatic reversions in non-codifying regions. This was this case, for example. It has been described, for example, in telomere-related diseases by mutations in gen TERT. There are a couple of papers reporting patients with haploinsufficiency due to loss of function mutations in one allele. And in these papers, they find point mutations in the promoter of the wild-type allele, not in the mutated allele, of the wild-type allele, increasing the expression of the wild-type allele and thereby compensating a little bit this haploinsufficiency. Then we have also loss of heterozygosity mechanisms that may be intragenic recombination, and that is possible only when we have a wild-type copy of the allele. For example, in a compound heterozygosity, you can have an intragenic recombination, and then in one allele you will have the two mutations, and the other allele will be wild-type. Or other forms of copy-neutral loss of heterozygosity, like segmental uniparental disomy and other mechanisms. And finally, large chromosomal rearrangements, large deletions, entire chromosomal loss, or chromotripsis, which is a rare event. So moving to primary immunodeficiencies, the somatic genetic rescue was identified in a case of autosomal recessive ADA deficiency almost 30 years ago. And then later reports expand these findings in X-linked severe combined immunodeficiency. You can see here different cases reported with different back mutations or second point mutations affecting different cell lineage, but mostly T cells, and some of the patients displaying mild phenotypes according with this reversion. So at the beginning, this somatic genetic rescue was believed to be a rare event and restricted to few specific gen defects. But as you know, over the last decade and using these sensitive genetic tools like NGS technologies, this has changed this assumption. And detecting growing number of somatic genetic reversion events have been described. And in primary immunodeficiencies, for example, Wiskott-Aldrich syndrome is the primary immunodeficiency in which somatic genetic rescue occurs more frequently. In fact, there are almost 40 cases published, and there is an exit survey that indicate that approximately 11% of the patients with Wiskott-Aldrich syndrome may have this somatic reversion, which is a remarkable percentage. And in these patients, since there are a lot of patients described, some interesting data may be characterized. For example, revertant cells have been detected in a wide age range from a few months to up to the 4th decade of life. T cells are the most common subset containing this somatic reversion. And the presence of revertant cells not always confer a clinical benefit, because depend in what time the revertant appear, the cell type that in which is present, and the proportion of cells containing this reversion, okay? So, for example, the question is why in Wiskott-Aldrich syndrome this somatic genetic rescue is so prevalent? So the answer is not known exactly, but it seems that may be related with the induced genomic instability produced by WES deficiency. Moreover, WES gene has some CPG regions that may be more prone to be mutated. And besides WES syndrome, there are a lot of, I don't know if it's a lot, but several other primary immunodeficiencies in which somatic revertants have been described. The frequency is not always the same. For example, there are genes more prone to be mutated and accordingly more prone to be reverted, like DOC8 that Jeremy showed us, because DOC8 has some repetitive subtelomeric sequences. For example, in ADA deficiency, it is thought that the accumulation of toxic metabolites may be mutagenic and may provoke a high frequency of these somatic reversions. And for example, there are rare events like in WHIM syndrome, which is caused by mutations in the CXCR4 chemokine receptor. There is one case of a patient that by the mechanism of chromotripsis, which is a complex genetic explosion, as Alex told us before, in which there are fragmentation, scattering, rearrangement of chromosomal parts, usually with a loss of genetic material. And in the patient, in this patient, that patient lost several megabases, including the CXCR4 mutated gene. And in the paper is interesting because the patient is for 20 years completely healthy because he lost the copy with the gain of function mutation in the CXCR4 gene. So now I will tell you the story. And I will tell you the story as occurred in the real life, because these are unpublished results, and I want to tell you what happened. So this is the patient. I will only highlight some interesting points. The patient was a girl who, at the age of 12 years, presented with HLH, hemophagocytic lymphohistiocytosis, in the context of highly active Epstein-Barr virus infection. At that time, the patient was treated with the HLH 2004 protocol, and no mutations were found in the 4 genes typically causing familial HLH. The patient received allogeneic hematopoietic stem cell transplantation from an HLA identical brother. In the next years, they received several infusions until she reached a stable chimera. But in the next years, the patient will not be able to control the Epstein virus. You can see here a high number of EBV viral load. She developed chronically active Epstein virus infection and even a smooth muscle tumor related with this virus. She was treated with several approaches. And at the end, in 2020, the patient experienced a spontaneous decrease in EBV viral load, and currently she's clinically asymptomatic in a good general condition, taking into account the complex clinical history. So in 2019, this patient was included in a project in our lab that aimed to find genetic causes of patients with non-familial HLH. So in this project, we include patients with suspected primary HLH or secondary HLH, but in which we excluded mutations in the familial HLH genes. Using whole exome sequencing, we identified a probably pathogenic variant in TNF superfamily member 9, which is most known as CD137 or 41BB. 41BB is a positive costimulatory molecule for T cells, and is probably most known because it has been used for engineering chimeric antigen receptors, or CART, for the cancer treatment, okay, for the immunotherapy against cancer. So our variant was a stop-gain variant with a very, very low frequency. And when we saw this variant, we realized that just 1 year ago, the deficiency of this gene was described as a new inborn error of immunity that causing immune dysregulation and chronic Epstein-Barr virus infection, so perfectly fitting with the patient phenotype. Our variant was quite close to the end of the protein, and as Jana told us before, this may be quite challenging because the stop codon makes that the mutant protein lacks only 12, the last 12 amino acids. But reviewing the functional study in this gene, we found a paper related with the use of this intracellular motif in CAR T cells. And this is the last fragment of the 41BB. And this last fragment, intracellular fragment of 41BB, has 2 traf binding domains, which are very important. And when the authors mutated the first, the second, or both motif, in the 3 cases, there is no signaling through an FKB pathway. So we realized, or we thought, that this mutation was probably loss of function. So at that point, we considered very important the familial study, as you may know. But welcome to the real life. The family completely refused the familial study. It was a complex family. The relation with the family was pretty difficult since the beginning. So we have a picture in which we have a patient with a homozygous mutation, probably pathogenic, and which received hematopoietic stem cell transplantation from a donor, from a brother that we do not know the phenotype, the genotype. Okay, the patient at the time has a mixed chimera. You can see the mixed chimera in granulocytes and B cells, but the T cells, which are important for the Epstein-Barr virus control, are almost all from the donor. Okay, so at that point we decided to study the status of the mutation in the peripheral blood of the patient to infer the genotype of the donor. And here you can see the Sanger sequencing. This is the stop codon originated by the mutation. This is the reverse sequence, so you have to read it the other way. And we found two surprises. The first one is that in the mutation position, there were no signs of the wild-type allele. So we thought that maybe the brother was also homozygous for the mutation. But the brother was completely asymptomatic at that time. And the second surprise, thank you, Anne. The second surprise that was this little peak adjacent to the mutation position, and we ask ourselves if this can be a somatic revertant, because they convert the stop codon to a missense mutation. So finally, we explained the family the importance of the genetic study, and the brother agreed to do the study, and indeed, the brother was homozygous for the mutation. So the patient received a transplant from a homozygous brother, was homozygous in blood and in saliva. And interesting that there was no traces of this little peak. So the next step was to try to confirm the presence of this revertant in the patient. Oh, before that, you may think that the patient was homozygous but clinically asymptomatic. And this is a case of incomplete penetrance. And in fact, in one of the papers published 1 year ago, they reported 4 families. And in 3 of the families, there was 1 homozygous affected and 1 homozygous asymptomatic patient. So it seems an uncommon case of incomplete penetrance in an autosomal recessive disease with a quite severe clinical phenotype. But in these 3 families, there is a homozygous asymptomatic individual. I have to say that this brother has slightly increased levels of EBV copies in blood, but it can control the virus. So we studied the mutation in the saliva DNA of patient, which was not present, this little peak, so confirming that it was restricted to the hematopoietic system. And we went back to the DNA used for the exome, that obviously was DNA before the transplant, and before the transplant, obviously, the little peak was not there. And then we tried to see how this revertant was located, and you can see here that we isolated T cells and B cells, and the reversion was located only in T cells. And another surprise is that we detected a second revertant. You can see here the first revertant converting the stop codon to a missense mutation, and the second revertant in the original mutation position, that is a back mutations reverting to the wild type allele. So at that point, we have several questions. Which is the frequency of these revertants? And are the revertant 1 and revertant 2 independent revertants? Or maybe the revertant 2, which is a smaller, has been developed in the revertant 1 cells. So at that time, we moved to deep Amplicon sequencing to solve all these questions. You can see here with a coverage of 100,000, we confirmed that the revertant 2 was specifically in T cells at 14% of frequency, whereas the revertant 2, which is the back mutation, was present in 8% of the reads in T cells. The second question was if these 2 reversions were independent, and using the NGS data, we confirmed that there are 2 independent revertants. You can see here that the reads containing the revertant 2 do not contain the revertant 1. This is the revertant 2 is a back mutation to the wild type situation, and the revertant 1 has the original mutation and the reversion event. Okay. So the other question is in which time these reversions arouse. Okay, because we're not present at the moment of the transplant and are present now. So we took advantage of the DNA stored for the chimerism studies at the blood and tissue bank, and we were able to obtain DNA from 2018, not before, but from 2018. And you can see here the evolution of the revertant 1, which increased the frequency over time, but then it seems that it's slightly reducing. And the revertant 2 is also increasing the frequency up to 10% in 2021. So the last steps, and the project is ongoing, we isolated CD4 and CD8 T cells, and interestingly, the revertant is specifically present in CD8 T cells, which are the cells crucial for the control of Epstein-Barr virus. The two revertants are in the CD8 T cells. Now we are trying to do some functional studies, which are complicated because we have no samples of the healthy brother, which was a good control because he carried a mutation in homozygous but is clinically symptomatic, and the patient has the revertant, so it's difficult to discriminate the effect of the revertants versus the non-revertant cells. And we are doing also CD8 single-cell RNA-seq to see if we can infer where these revertants have been produced intrathymically, studying the TCR rearrangement, etc. Okay, so, but interestingly, the patient experienced a spontaneous decrease in EBV viral load at 2020, which is more or less when there is highest frequency of these two revertants. As you can see here, that the revertant 2 is increasing its frequency, and the revertant 1 is decreasing. We don't know what will happen over time, but the revertant 2, which is a true back mutation restoring the wild-type allele, maybe will continuously growing. We don't know. So if you want a hero in your life, this is the revert man. And this project is ongoing, and it's the first revertant described in this very recently described inborn error of immunity, and like all these genetic events is very fascinating. So just to acknowledge Laura Valle, who did most of this work, and she will present now a case report of other project, and all the team in Vall d'Hebron and in other centers located in Barcelona, in Catalonia. Thank you very much.
Thank you. Thank you. Beautiful, beautiful story.
Beautiful presentation. Very interesting. Do you think that defective DNA damage response could affect the capacity to revert mutations?
Sorry, can you repeat, please?
Do you think that a defective DNA damage response, as in Wiskott-Aldrich patients, could affect the capacity to revert a mutation?
It's possible. It's possible. You are referring to the Wiskott-Aldrich patients or to our patient?
Wiskott-Aldrich.
Yeah, it's possible. I'm not an expert in Wiskott-Aldrich, but as I told you, the genomic instability produced in Wiskott-Aldrich, these CPG islands that are in the gene are probably influencing this high rate of somatic reversion that have been described.
And the second question is if the second patient that you described with this last case encountered the EBV. The second yes, patient, yes. The two homozygous patients, the first patient was a BV positive, it didn't resolve the infection. The brother,
yeah,
what about the brother?
Yeah, the brother was clinically asymptomatic, but when we look at EBV in blood, we found that they have some viremia in blood, indicating that the EBV is there, and the viremia is higher than that you can found in control population, but we don't know why, but the patient can control the viremia, for example, in few thousand copies, which is not normal. You don't have to, healthy people don't have thousands of copies of EBV in blood, but they may control the virus without clinical involvement.
One last question.
Yeah. Sorry, it's not really a question. It's just a learning point, which is that where you do not have a genetic diagnosis, it is hazardous to use a matched sibling as a donor for allogeneic transplant. Obviously, this experience emphasizes that.
That's a good point. So at that time, this was 2012. At that time, the clinicians preferred to transplant the patient with an HLA completely matched brother, although the risk was that the brother may have also the disease. But at that time, the brother was asymptomatic. Now he's also symptomatic, so it was a tricky case, but I agree with this a good point.
Okay, thank you very much, José. And I would like to introduce the next speaker, which is Dr. Andres Caballero Garcia de Oteiza. He will replace Michele because he couldn't make it, and I'm very happy. He's from the Center of Chronic Immunodeficiency of the University of Freiburg. And we will discuss the GEMMA database.
How's with the pointer? Let's see. Okay, so thank you, Marielle, and thank you for the invitation. I'm sorry that I'm not Michele, and I'm sorry that I'm not going to speak about GEMMA either, but I hope that I can engage you during the talk anyway. So I'm going to talk today about Genia. Let's say it's GEMMA, Dima baby sister, and I'll explain you why now. So I'll start the talk with these basic questions. So do we need this database? So I'll take this analogy of generalist and specialist in nature. So we've seen in nature that both strategies are possible. They both are compatible. They don't have to compete to each other. And this could also be applied to our real-life problem, which is different type of databases or resources that we can use for our genetic analysis. So here, as generalists, we have databases like OMIM that, CleanVar, Orphanet, or VARS. If you can consider as a database as well. You all know them. They are great resources. I'm not here to say anything against them. I use them, and we all benefit from them. But we also have some other specialists, and we'd like to include ourselves into this category. So like there are others, like the Infuverse database, the CFDR France initiative, ClinGen could also fit in here. And I think both are needed, and I'll show you why this could be beneficial. So basically the idea to come up with this resource or database is that oftentimes when we look at our patients, at the genetic data, we ask ourselves these questions, right? So is this gene associated with human disease? Normally we look at the genes that have been linked to disease. Is this gene associated with an immune-mediated disorders? As the variant been described, how many cases or families have been reported if the gene is recent? What's the clinical spectrum of these patients? And does the variant fit my patient's phenotype? Has the variant been tested? It's only been published as a big whole genome study. Or also the last question, like this case that Toyera presented, am I missing something that is not in the textbook? So what are diagnostic paradigms of this gene? So we hope that with a specialized database, we could more accurately respond to these questions. So we wanted to become a specialist, and what did we do about it? So 3 years ago, we started with this project called GEMMA. We presented this at the last ACID meeting. So I will not talk about GEMMA, just to introduce where this came from. So basically, this is our internal database. We have incorporated different resources and data sets for from different ontologies, like the HPO, the gene ontology, OMIM ensemble. We have various different data sets, so clinical data, genetic data, lab data, literature data, functional data. And this is all in one place, all, let's say, using these ontological terms, so these common standard dictionaries. So it is, we created a simple web application with a login page and user-restricted access and different roles. And this is currently limited to our hospital in Freiburg. Here just some numbers, general numbers of the data that we have inside. But of course, this is not open for the public. So just as an overview, so we have a mix of, for many patients, we have a complete picture of genetic lab values, clinical data, microbiome data as well. But we hope to do better analysis with this type of data sets. Just here as an example of regarding the gene or variant creation. So this is, for instance, the number of gene and variants that we have annotated or classified, manually classified or looked at. And also, as an example, CTLA4 variants, how many have been published or not, if we consider them pathogenic or not, what are the functional data, evidence about them. But as I said, I will not go deeper into this right now. So I'll just highlight the two main issues with GEMA. So as I said, it's tailored to ENIS or our CCI genetics unit or the AG-Grimbacher group, and it contains sensitive data. So of course, this cannot be publicly accessible. That's why we decided to create GENIA. So as I said, GENIA is probably her GEMA's baby sister. And we designed this with the goal that this could be open to everyone. So no sensitive data inside. The only way you could relate back to the patient or connect the patient is if you are the author of that study. Of course, you should know what patient is, but I will not be able to know what patient is that. So just here is the homepage. I mean, the link is there in the slides. Just with a summary of the numbers that we have. So about 1,200 subjects from 700 families. We have almost 400 or more than 450 published variants linked, curated genes, and these are the number of inspected papers. So by inspected, we mean that we have manually make sure that all the information we could extract from that paper has been imported in the database in regards to patients, lab data, cell values, everything. So we've done this for just as a proof of concept to show this. We did it with a few genes we are more familiar with. And of course, we start with the cases that are a few cases reported. We will not start with stat 301, but I will discuss this later. So here, just to navigate you through GENIA, just very quickly, I don't want to spend too much time on this. So if you can enter through one of these different modules. So if you decide to type a gene name, here you would come up with a short list of your matching your search. You click on the gene of interest, and then you go inside and see details of the gene. So in the upper pane, you have just basic details from the gene that you can extract from these public resources, linked to HGNC, Ensemble, Omim, etc. Just below a normal function field where we could also get this automatically, but these fields are editable. So that means that if you are the expert in the field on this gene, or well, you think you know enough about this gene, you can tell us, and you can update this content or write it yourself. The same about the dysfunction on disease. So what is known about the disease? So when this gene is not functioning properly, what disease is linked to that? So if you scroll down, then you have a list of associated conditions. So right now with RPC1B, there's just RPC1B deficiency, an autosomal recessive, very severe disease. Again, with the mode of action linked to Omim, and a summary of the number of cases and families. So this is basically now what has been imported into GENIA. So if you continue to scroll down, you go to transcripts of RPC1B. This is just information that you could also get from directly in Ensemble. And this is a link of published variants. So just here a small table with a summary of the variants. You can go and click on one of those variants and look for more information. So if you scroll down below, just here as a warning, this is not RPC1B because there are no diagnostic paradigms described for this gene yet. But if you check, this is an example for ADA. Here's where we say, well, we let the physician, clinician know, or geneticist know what are the things to take into account when analyzing this gene, or when you don't find the answer in your patient. So there could be a case of uniparental disomy, somatic mosaicism, reversion as has been described here, cryptic splicing. So we highlight this for this gene and link to the reference and do a small comment. So another example with IL2RG, just the same. So we could link more than one paper for the same type of diagnostic paradigm. So if you go back to the variants just very quickly, and we zoom in into one of these variants, Again, just basic information here above that you can get from the normal annotation pipelines. And below, just a comment also editable that you can edit with important information regarding these variants. So in this case, it's just that is mentioned differently or referred differently into different publications, consecutive publications. So something to take into account. Just what is the effect of this variant on different transcripts. And here the list of reported subjects. So this is probably the most interesting part, which is, as I said, subject ID is just a random code. But you have also information about the family, the origin, if they mention in the paper, if the variant has been homozygous, heterozygous, or wild type, which is also important to know if the father, mother, or relative was tested, and just the link to the code of that patient in that publication. Of course, patients are used in several publications, so this is also important because sometimes we think that we have more patients than we actually have, and so here you can see that one patient has been published in 3 different publications. So, and below just a functional characterization. So in that study, was there some functional work done on that variant? What are the assays used to come to that conclusion that let's say the protein expression was reduced or not? And just the effect, if it's an amorphic variant, hypomorphic variant, or isomorphic, or gain of function. So you could tag these different effects. And as I said, for each paper, I mean, you can add this data for different papers. So if we go back within the gene to the associated conditions, here again, you could also arrive to this place by this the genetic disorders. So module, you just enter research and you will come up with what is the disease itself as an entity. So here, if we look at the disease again, in the upper pane, just general information about the disease, number of cases in the database, just description. Again, we can also edit this field as you would do in like in Orphanet or OMIM. And here again, patients reported with this phenotype, so with this genetic condition, with similar information that we were looking before with the variant. And below, this is I guess the probably most important thing to highlight here is that we have a summary of the clinical findings in this 28 manually created subjects. So we can know if for those patients all classified as ERBC1B deficient, how many of them had increased IgA levels, how many of those did not have increased IgA levels, which is also an important information that is generally not mentioned or not captured. Again, also eosinophilia, eczema. So here you can see what are the most prevalent terms in that. So this is something that you will not find in OMIM or other resources. You'll find the HPO terms, but you don't find the frequency of those. So if you go to the individual subject, again, just the only thing you could know is just the sex, family code, origin, who's the father, who's the mother in the database. Whether it's affected or not, the age at death, if that patient was deceased, and again here the link to the different codes for the different references. You could also see the age at when was diagnosed and the age at first manifestations, if you can get this information from the paper. And you could also have access to, if you have the family completed, you can generate these family trees automatically as well. And again, if you go down, you see the list of genetic variants reported for that patient in that publication and the list of clinical manifestations, all based on HPO terminology. Scrolling down below again, so you can see cell counts, immunoglobulin values, or we also added now lab values. So if you want to add CRP or hemoglobin or whatever other lab value you want to add. And if you click on one of those terms, is basically, again, if you could also enter through the clinical science module and click what is reduced proportion of CD4 T cells. This is just a way of seeing what you would see in HPO when you go to the HPO browser. Basically the basic information and how this term is classified hierarchically. So what are the parent terms, what are the child terms, and you can go up and down and navigate. And again, if you go down, you see the list of patients that are reported in the database with that manifestations. So You could repeat this step again, going through or entering through any of these modules. So if you go to the paper, you look for the PubMed ID or for the title or whatever, or the clinical sign if you type thrombocytopenia. So you could, again, everything is interconnected, and you can arrive to the patients or the variants. So why it's important to collect all this information? It's a lot of work, as you might imagine, but we want to have good quality data, because this will, of course, have an influence in how we do our analysis and interpret our findings. Also for the variant filtering and prioritization steps, this would also allow us to do better predictions, to do better computational algorithms, probably better research overall, and maybe train future machine learning algorithms, as we've been mentioning before. So I just want to point out that here, I know that many groups and companies are using HPO, and I mean, a great initiative, and they're using this to filter and prioritize their exomes and genomes. So I guess this is why it's important, as Marielle was pointing out, that the creation of HPO and the work on this, and this is why, in this way, we want to contribute to that project. So here I'll just show you 3 examples of how we could maybe contribute to this end. So with RPC1 deficiency, as I said, autosomal recessive conditions, if we compare the number of patients, papers, mutations, or families reported in OMIN or ClinVar, of course, if you do it manually, if you do this manual work, you will have a better overview of what's the reality of this condition. That if you go to these databases, as I said, I mean, they're great, nothing against them, and we benefit from them, but we want to go deeper. And if we compare the clinical presentation of RPC1 deficiency so what you find in HPO, if you go to the HPO browser and download the terms that are linked to RPC1B as a gene, there are, I think, less than around 10 terms linked to this gene. After the creation of these papers and patients of these families, we could find 130 clinical terms that we could assign to this family, to these subjects. In OMIM, if you go and read the clinical synopsis, you would also have almost 50 different clin terms. I mean, they don't use HPO, but it's very similar. And so here's just an overview of what would be RPC1 deficiency, and again, what is the percentage of that presentation, in how many patients it is, and how many patients it is not, and so on. And just to show here, these are the HPO terms downloaded from HPO for this gene, and what we think is important is that 10 of our most relevant or frequent manifestations are not even present in HPO. Again, I mean, nothing against HPO, needs to work to be done, and this is why we want to collaborate and make this resource hopefully better. Here just to show you an example of collecting all this data in this detailed way, we could also do this type of analysis. So for this disease, where's the age at first manifestation? So this is very severe disease, very early onset, and how many years it takes for these patients to diagnose. So many of these patients spend many years to be diagnosed, but hopefully most of them were diagnosed within the first 10 years. Second example is SEC61A1. In OMIM, I have 17 minutes. So it's 17 minutes now. I have 4 minutes more. Yeah, so SEC61, OMIM only recognizes one condition. However, there are 3 different conditions published now in the literature. Again, with the number of cases here reported. Doing the same comparison, of course, we count more patients that you can find in these other resources. And you could also compare the different clinical manifestations per condition, so not directly linked to the gene, but to the different conditions of this gene. And finally, the last example. So this for OIS1. This is also a recent gene. Recently published. There are two papers reporting cases. So here, of course, with two papers, the numbers are more similar. But again, there are, let's say, 10 patients overall described so far, where if you go to OMIN, you will only find 6. what is interesting about this disorder is that well, all these patients occurred de novo, and even in one family, 3 cases. So the parents were not carriers, but the 3 children had a de novo mutation. This is probably, as Anna Mensa was talking before, probably something that happened, I don't know, during the gonadal stage. That's interesting to highlight. And again, here we could also plot the age at which they start presenting symptoms, age at which receive hematopoietic stem cell transplantation, age at which they died or they were diagnosed. And finally, yeah, so for OIS1, since it's a recent gene, of course, it's understandable that HPO is not linked to any term yet for this gene, which I guess is important because you would probably miss these variants. So all the variants that have been published so far are missense variants that if you go to CleanVar, there are, I think, some of them, not all, even just tag as variants of uncertain significance. And maybe you would probably miss them if you use this approach. So I think it's important that we collaborate maybe with HPO and share this data. So just as a summary, GENIA is a public created resource, more detailed than other databases. Of course, this is not fit for all genes, but we hope that slowly over time we could get to the 400, 500 genes. It's free to query. We maybe have to implement some tools that you can have easy access if you want to get information. If you want to write a review or something, we could also help you with the curation of the cases described. And the clinical terms, the HPO terms, can be linked at 3 different levels. So you can link it to the subject, but from that you can link to the gene or to the condition of this gene if the gene has 2 or 3 associated conditions, or to the gene itself. So you can have 3 different levels. Also taking into account if that HPO term is present or is not present, which could also be important. As I said, as an outlook, what we plan to do is to do this maybe gene by gene, start with the cases that there are only few cases reported, paper by paper, and slowly getting there. Of course, we want help, or we would need help if someone wants to contribute. And as I said, this could be a way, if you want to write a review, this could be a win-win situation. And we want to collaborate with the HPO team and get more experts involved. And finally, probably we need to create a login system that we can, so if people want to, or experts want to contribute, collaborate, that they can edit content and so on. So finally, I'd like to acknowledge Michele and Xiao, who were with whom we developed this project together, and of course Bodo Grimbacher and his group, who helped a lot and collaborated, and you for your attention.
Well, thank you. It was a very nice talk, very beautiful work. I take the first 2 questions already. The first, so you did not, because you recruited the HPO terms and you added HPO. Yeah. So you added HPO terms and you noticed they were not complete. Did you add the HPO terms you detected to the database? Did you submit them to the HPO general database so everybody could use that?
No. I mean, so this is also why we decided to present this. This is very recent work.
Future work, yeah.
And this is our plan to now talk to HPO and see how we can do this. I mean, maybe in a faster way or real time updating. So, yeah.
Yeah, no, it's very nice. No, very promising. And the next question is, could you link it to other databases? Because or many institutes have their own database. Is there a way you could link databases together to get all the information together instead of re-upload it to another database? Is that a possibility?
Yes, we could discuss this. Yes, I mean, as I said, I would love to give people access, free access to query the database if they want to have a look how the data structure, how to. I mean. Yeah, we have to decide. Maybe we need to design an API so to interact with different databases. Yeah. The thing is that, of course, there's no. You need to keep track of what are your codes, and so we don't want to have any sensitive data inside because this will be open. So yeah, if as long as you send us a random code, and we can enter the patient as well.
Oh, nice. Very promising. Any other questions?
Sorry. So I'm asking, is access free?
Yeah, yes, it's open. You can enter now.
And so I have a question.
Yeah.
So let's say I would be very curious for a gene, but so if I'm asking, let's say, I don't know, let's say STAT1, for example. Who is going to, so if you don't have it, how do you choose the gene you are prioritizing?
Yeah, so as I said, I mean, we could choose now ourselves to make our lives easier if we start with the genes that we know that there are few cases reported, to slowly feed the database to become more attractive to people to go and look. But of course, this also the other way. If someone like you is interested in STAT1, let's say because you want to write a paper or because you're just interested, you can tell us and we say, okay, let's work on this, and we prioritize this gene and we do it first. So there is no clear rule, but we would love to do this type of collaboration.
Okay, thank you.
Hi, I was just saying, I mean, and you mentioned databases before. We have the ESID database, which is literally thousands of patients now, many of whom have genetics in there and their clinical phenotype. Is there any way of being really, and it should be all in there anonymously, is there any way of being really slick in using a lot of that data? Because there's thousands of patients in there, most with full phenotypes, a number with genetics, that would be really useful to get in. So you don't have necessarily the publications, but you have a massive data resource.
Yeah. So this is what we've been thinking about. So to use with patient registries, right? This is also a very rich source of information. What you could do is to create the ESID registry as a publication, and you link all the patients to that if you send us the codes. So as long as you keep track of what code you send us, so it has to be completely anonymized, then it would be perfect. I mean, you give us a list and send us the data, and we can upload everything. So what we would need to take into account is that the patients are not duplicated. So this is also a very important point that I did with the paper being the same patient being published in two publications. So because otherwise we will be doing miscalculations of the penetrance or of prevalence of this disease. And
thank you. A very useful tool. In practice, how is it curated and how are the data downloaded in the system? Is it dedicated staff, informatician or medical staff? Is it curated?
No,
how does it work?
Sorry. Yeah, so this is something that we've been doing because, I mean, we also do genetic analysis for our patients. So whenever we encounter a variant or a gene that was of our interest, then we use that time to include this patient into the database. So many of these patients, as I said, we have like 1,200 subjects inserted there. Most of them come from GEMA. And I said that we curated almost 20 papers right now. So there's a lot of information, but not all of it has been deeply curated. But there are many cases that have been published and they are already in there. It's just that we just now decided to go gene by gene, paper by paper, to make sure that everything is completely curated. But it's something that we've been doing over these last years.
Okay, sake of time, thank you very much, Andres. And before we have the coffee break, we still have Bionano, because she was presenting at lunchtime, but couldn't make it because there was no. Do you understand me? Yeah. There was no online possibilities. So the coming 15 minutes, Bionana is going to present what it is all about.
Okay. So hello everyone. Thank you for attending today. My name is Dana, and I'm going to be presenting to you Bionano's next generation cytogenomics technique. It's called optical genome mapping, or OGM. So in the morning, you assisted to the talk of Alex Hoyschen, where he presented some of his great results using our technique. And I'm going to be talking a little bit more in details about the technique and some other great results as well. So optical genome mapping, or OGM, is a technique that allows the identification of structural variants and copy number variations throughout the whole genome. In its principle, it's comparable to karyotyping, but at a much higher resolution. The way our technique works is by direct visualization of long-labeled DNA molecules. So the DNA molecules are labeled on an average of every 5 kb genome-wide. This way, you can distinguish a unique pattern of bands throughout the whole genome. And labeled DNA molecules are scanned and analyzed automatically using our Sapphire system. Similar to karyotyping, every abnormality in the labeling pattern will indicate the presence of a structural variation. So this is how it works. On the left side, you can see the labeled DNA molecules being linearized on a flow cell of about 120,000 nanochannels. Electrophoretic cycles are being applied to the DNA, and therefore the DNA molecules are being scanned and then flushed, and new DNA molecules are scanned again and flushed again, and so on. So this allows the acquisition of huge amount of data in parallel. Can everyone hear me? Okay. And so by running the instrument longer, and thanks to those electrophoretic cycles, you can increase the coverage to very high levels that outrange the coverage with other sequencing techniques. And this you can do with no extra cost. Another major difference is the size of the molecules we image. They are above 200 kb. So we even have the luxury to disregard any molecules smaller than 150 kb for our analysis. So we combine all you need for to address constitutional and cancer samples, including high coverage and long read information, and for a few $100s per sample. All this without the need for culturing, no need for probe design, library prep, or amplification, or denaturation. And actually, our workflow is extremely simple. So first you start by extracting the DNA using our DNA isolation kit to obtain ultra-high molecular weight DNA. Then the DNA is labeled using our DLE1 enzyme that will insert a fluorophore on a 6 base pair motif that is present every 5 kb genome-wide. So this is where the specific labeling pattern will be applied. Following this, the stained DNA will be loaded on a chip, and from here the rest is automated. So the molecules are then imaged and aligned together, and this alignment is based on their labeling pattern. This will allow to construct a consensus map that we call optical map. So the optical map of the reference is always shown, of the sample is always shown in blue, and is compared to a reference shown in green. So this reference can be the genome reference of the human, of the genome of humans. It could also be control tissue or the sample's parent in case of trio analysis. And by this comparison, this is the step where any deviation in the labeling pattern will indicate the presence of structural variants in the sample. And in parallel, we have the copy number variation tool that will do a dosage of the molecules genome-wide and identify any large gains or losses. So the results come in a very user-friendly report on our BioNano Access software. This is called a circus plot, and analyzing data does not require previous knowledge in bioinformatics. For SV filtering, one of our most used features is the control database. So this is a control database built on 300 controls. So you can filter your SVs against this database to only retain rare and likely deleterious SVs. So you can also use BET files in case you are interested in looking at structural variants in particular genes of interest or particular loci of interest. And eventually, your filtered SVs can be exported as a VCF file or a PDF report. So for data analysis, we have two pipelines. Both can identify all these classes of SVs. And the difference between the two pipelines is the following. So our first pipeline, the de novo, is used for highly homogeneous samples. So it detects germline SVs down to 500 base pairs. And our RVA pipeline detects slightly bigger events, so above 5 kb, but events that are present at very low allele frequency. So it can go down to 5% for 400x coverage, and even below that when increasing the coverage. So this is the pipeline that you usually use for highly complex or mosaic samples. And as I've mentioned, the copy number variation tool will detect large gains and losses, even those that are present at 10% allele frequency. So optical genome mapping overcomes the limitations of other techniques, and in one experiment allows the identification of all these classes of SVs. So we detect whole chromosome changes, such as monosomies, trisomies, and triploidy. We detect deletions, whether they are terminal or interstitial. Duplications, for which we give you the exact location and orientation of the duplicated fragment. Translocations, whether they are balanced or imbalanced. Inversions, whether they are peri or paracentric. And we also detect absence of heterozygosy, and we are currently working on the detection of loss of heterozygosy if it is somatic. We also detect repeat expansions and contractions. And it is important to note that OGM is not a sequencing technique, so therefore it does not detect single nucleotide variations. So the application of OGM in constitutional diseases is very wide. I'm going to show you some examples to give you a better idea about the power of detection of this technique. OGM is a great technique to identify large SVs, and it allows the refinement of the information that is given by standard of care. This is an example of the George syndrome case, for which chromosomal microarray identified a 2.8 deletion. When using OGM, of course, our technique confirmed this deletion of 2.8 megabases, but also showed an inversion on the second allele. OGM is also a powerful tool in identifying small SVs, even those that are present in regions rich in GC. And a classic example for this is the Fragile X syndrome, in which the CGG repeat expansion in the FMR1 gene can be detected and measured automatically using our BioNano Access software. So OGM allows the identification of repeat expansions or contractions, even those that are extremely large, or even those that are present at mosaic state. And this is an example where OGM identified 5 different haplotypes with different repeat expansions in the C9orf72 gene. This was in a single postmortem brain sample of a ALS patient. Additionally, OGM allows the identification of structural variants in highly repetitive regions. And this is the case in the CR1 gene, where OGM allows the measurement of the number of binding domains in this gene. So those are repetitive domains. And in this case of an Alzheimer's sample, it identified a deletion in those domains. And another great example of how OGM can solve cases that other techniques cannot is this paper that Alex presented this morning on ATRT and SMARC-B1. And Alex also showed you this morning how OGM shows 100% concordance with standard of care in constitutional diseases. So now we're going to talk about OGM and its application in solid tumors and heme-onc to give you an idea of how this technique resolves complex rearrangements and even in heterogeneous samples. Here is a group from Augusta University that was led by Dr. Ravinda Kole that was interested in comparing their standard of care consisting of OncoScan and FISH to compare it to OGM. So this was done on 30 adult brain tumors. And first, they were very pleased to see 100% concordance between their SNP array and OGM CNV tool, so in the detection of large gains and losses. And interestingly, they identified many additional translocations affecting oncogenes and tumor suppressors as well. Those translocations were not previously identified. And this is because, conversely to FISH and arrays, OGM brings the great added value to detect balanced and unbalanced events genome-wide. OGM has the capacity to zoom out and give a comprehensive understanding of what is happening genome-wide. And in this work of James Broach, OGM was used to characterize head and neck tumor patients based on their HPV integration status. And just by looking at the circus plot, they were able to segregate between samples or patients that have episomal form of HPV, so no genome instability, and integrated form of HPV, so a lot of rearrangements due to instability. And when it comes to OGM and its application to Hem-Onc, in this study that was also shown by Alex this morning, OGM showed 100% concordance to the standard of care in a cohort of 52 myeloid and lymphoid neoplasms. And in a larger cohort of 100 AML cases, OGM showed 100% concordance as well to the standard of care using karyotyping FISH and chromosomal microarray. And additionally, OGM allowed the increase of the diagnostic yield by 6% for cases with a normal karyotype. And overall, in 40% of the cases, OGM brings significantly new information by refining the breakpoints and by clarifying complex rearrangements such as marker and derivative chromosomes. And now I'm going to talk briefly about the use of OGM in cell line QC. We are seeing more and more interest in implementing OGM for the quality control of cell lines after gene editing. So whether the gene editing is targeted and followed by clonal selection and expansion, or if the gene editing is random and a heterogeneous cell population is expected, OGM is a great technique for the detection of unwanted rearrangements genome-wide. This allows to monitor maintenance of edited cells in culture at different time points, or to compare two cell lines in parallel, for example, edited versus unedited cell line. And when the gene editing is targeted, you can also confirm, in addition to the quality control, you can also confirm or validate your gene editing if it is above 500 base pairs. And a great example of the use of OGM in iPSC QC was presented by BitBio in our annual symposium. So I invite you all to scan this code if you are interested in watching their presentation. And in summary, they used OGM to see if they can replace their standard of care QC methods using OGM, and they were extremely happy with the results. And they showed that OGM is uniquely suited for characterization and authentication of cell lines. And in conclusion, I would like to mention that OGM is a genome-wide technique that can provide you with a true structure of the genome. It allows the identification of small and large structural variants, even those that are present at very low allele frequency or present in complex regions of the genome. All of this with a smooth workflow, automatical data generation, and user-friendly data analysis. And just to mention that OGM is used for research use only for now. Thank you for your attention.
So thank you very much for this very clear presentation about a very promising technology. Are there any questions?
Thank you very much. I wanted to ask you about that research only label, because clearly your talk is demonstrating the diagnostic potential of the technology, and we would probably be quite keen to use that. So what is the legal position? Do we currently have to confirm any findings made using your technology by an alternative method?
So currently, this depends a little bit on the regulations in different countries. Our users for now have all validated the technique, so doing studies, retrospective studies in comparison, so comparing the samples to standard of care, as I've mentioned, either karyotyping, FISH, or chromosomal microarray, and showing those this concordance allowed the validation of this technique. So it depends between departments, between countries, but a lot of our users still use, for example, OGM, and in parallel they do the karyotyping, maybe on a smaller number of events, smaller number of cells, metaphases. They always have one standard of care technique in parallel, meaning the events that are identified with OGM are confirmed in parallel. And if you want to use, for example, to compare it to FISH, how our users would do is start with OGM, identify their translocation, for example, of interest, and after that confirm it with a FISH, replacing then starting with many FISH panels to find their event of interest. Yeah, and we are of course currently working on this research use only. Yeah.
If there are no further questions, I would like to thank you very much for this nice presentation. And we think you need at least a break. So if we can meet again at 4:15 maybe, then that would be nice. We just wait for Anne. There is. Okay, let's continue the next session, which is a very interesting one, I think. It will be the last session of today, and we have young fellows presenting cases, difficult cases, so we expect all the public to think with them and to solve each of those cases. We have 4 presentations. We start with Jeanne Bourchier from Montpellier.
Thank you very much for this opportunity to present this case that I hope will illustrate the workshop. So this is the case of a young girl, a 10-year-old girl who was referred to our lab for diagnostic, for genetic diagnostic. So I'm a molecular geneticist in a reference laboratory of autoinflammatory disorder in France. And her personal history was livedo racemosa and low IgM since infancy, ischemic stroke with ocular motor palsy. The test revealed absence of malformation, occlusions, or aneurysms. Hannah were positive. She has no history of recurrent fever or inflammatory syndrome, biological inflammatory syndrome, and no history of recurrent infection, and no family history. So what is the suspected diagnosis? DADA2, donc the deficiency of ADA2, was hypothesized by the prescribers, the clinicians. And you can see on the right that even it's an autoinflammatory disorder, some patients do not have any biological inflammatory syndrome. It's 16% in the cohort of Barman et al. Published this year. So when this diagnosis was hypothesized, we performed genetic testing of targeted ADA2 by Sanger sequencing. And in parallel, we tested the serum activity of the protein ADA2. And we identified 2 likely pathogenic variants. The both variants are rare, with in silico prediction deleterious. And each variant is already published once, and you can have all this information in our international autoinflammatory disorder database called InFevers. And the enzymatic serum activity of protein ADA2 was very low. It was 5.8% compared to a pediatric pool of healthy control. So this genotype is consistent with the DADA2 diagnosis according to our current guidelines. And genetic counseling is recommended. So for genetic counseling, we ask for testing of the parents and pre-symptomatic diagnosis of our young sister who is asymptomatic to prevent an eventual stroke if positive. And you can see that the sister is heterozygous carrier, asymptomatic, and the mother is carrier of one of the 2 mutations, and each daughter had inherited from the mutation from the mother, but the father was non-mutated, wild type on both allele. So what are your hypotheses now? Yeah, it was the or. So we analyzed this 24 microsatellites among 12 were informative. And in fact, not shown on the slide, we perform NGS panel, including ADA2 on both father and daughters, and we evidence rare variants shared by the father and the daughters, very rare variant absent from GNOMAD in other genes of the panel. And as you can see, the SRA analysis reveal that father seems to be the biological father because he has transmitted all the informative allele as good in this analysis. So what other hypotheses could we explore? Yeah, so we try to explore the existence of a low mosaicism on the blood of the fathers. And we have our technology and pipelines are validated to identify mosaicism below 2% only. We have 1,000x coverage for ADA2 genes, but we don't identify any low mosaicism. As you can see, there are 4 reads mutated here. This one has a very bad mapping quality. And these 3, you can see that the quality score of the sequencing, the Fred score, was very bad for the 2T on the reverse and on the forward read, and the A2. So we cannot distinguish this read as a low mosaicism from artifact sequence. And you can see here that on the same read with the T at our position of interest, there is another T here, and here there is another T here and here. So for us, we cannot, of course, exclude a risk of recurrence because of gonadal mosaicism, but with the limits of the techniques used here, we cannot say that there is a low mosaicism in the father. So for us, with the limit of our techniques, the family study shows one of the 2 variants is a priori de novo. The remaining question is what the phase of these variants. So we have the enzymatic test that says that the phase is probably trans, but this was not genetically confirmed. So at this time, we have 2 possibilities, or the de novo variant is on the same chromosome, or the de novo variant is a new chromosome. And the 2 possibilities are open. So what can we do to phase this variant? In our routine lab, we don't have all the techniques and all the technology possible. So first, we try to know if the allele that was inherited by the asymptomatic daughter was the same for ADA2 than the symptomatic proband. So we designed 2 STR flanking ADA2 gene, and between the 2 STR, there was 55,000 pair de base, so the risk of recombination is low. And we sequence the STR, and unfortunately, you can see that, of course, the mother has transmitted the same chromosome to both daughters, but the father obviously transmitted a different chromosome for each daughter, so we cannot conclude with this analysis. So another technique was tried. It's a long read sequencing. So we amplify and sequence a long fragment incorporating both variants and sequence it with a MinION platform. And as you can see here, we could evidence that the variants are not on the same read, and variants are biallelic. So this was possible in our lab because we have a MinION platform, and because the distance between the 2 variants was only of 3,000 per BP. So to conclude this case, on a genetic counseling point of view, we found 2 likely pathogenic variants on the proband. The long read sequence shows that RB allelic, so the patient is compound heterozygote, and the serum enzymatic activity of ADA2 and the clinical description is consistent with this result. So this genotype confirms a clinical diagnosis of DADA2. The familial study showed, with the limit of the technique studied, one of the two variants a priori de novo. We can exclude a risk of recurrence due to mosaicism in the father's gamete. So as a result of this atypical inheritance, the risk of recurrence with each pregnancy is lower than in the case of classical recessive inheritance. And as we discussed it this morning, in France, we usually suggest prenatal testing for ADA2. And the risk of recurrence for mosaicism, gonadal mosaicism, is estimated by convention at 1%. So for this family, the risk of recurrence would be of 12.5%. Or if the gonadal mosaicism is true and confirmed by another deep sequencing techniques, it will be between 12.5 and 25, but not precisely determined. Only if we have exactly the amount of gametes in the sperm that are mutated, but I think these are very complicated techniques that cannot be performed in routine diagnosis. So this case has only been possible after an extensive analysis of the proband and their fathers. And I hope it illustrates the need to carry on systematic families studies. Thank you. This is my team.
So, thank you very much. Any questions?
Did you try to clone the cDNA from this variant, the product of the cDNA from the father, and to see if cloning you can see the representation of your mutated sequence?
We didn't try cDNA. You mean to see if the mutation could be identified on the transcript, but not on the genomic DNA?
Yes, you can.
No, we didn't try
You can
this.
amplify the cDNA and then clone the product of this, the fragment, and then see if the second mutation is represented how many times so in your PCR product.
Yeah, no, we didn't try this. Maybe a good idea to explore.
Thank you for the presentation. Have you performed the AD2 activity to both parents?
Yes.
And it had been compatible with a carrier, the mother, and normal.
Yes,
the AD2 activity.
For the activity?
You perform the AD2 activity to the patient, but I don't know if you test parents for that, because the carriers have an intermediate AD2 activity. So just for conclude everything.
Yeah, you're right. In fact, our techniques is we do not conclude about heterozygote carrier, because you know the reference value for ADA2 are very variable and between individuals. I don't remember the results, but I'm not sure we cannot conclude that the father is heterozygous, or it could be 60% and on LC control. It depends on the mutation. Some mutation has like G25C is a pathogenic mutation with very high level of enzymatic activity of 100%, in fact, with this mutation. So I don't think we can conclude with this enzymatic activity in this family, but I don't remember the results. But maybe a good idea too. Thank you.
Yes, thank you very much for the talk. Since you just mentioned that this ADA2 activity can be really different between different people, so can I ask, when you were testing the activity of the patient, how many healthy donors you included?
Alors, for pediatric pool, it's very difficult because we don't have lots of healthy donor, because we called for blood donor at the French National Blood Center. So we have a 12 healthy control for pediatric pool, and I think it's 25 for adults. And it's very important to match pediatric and adult because the activity is not the same. It was a bit tricky to have this ADA2 activity in routine diagnosis. I think we made 1 year of performing tests before to put the activity in routine diagnosis, and lots of discussion with NIH.
Have you thought of sending the samples to a reference lab in France or the EU, which does ADA activity measurement? Because that way you might get a more definitive answer.
In fact, we are the reference laboratory. There is only one lab that performs ADA2. I think it's Necker or Cochin in Paris, and it's new. It's, I think, for 2021 or something like this. And it was a lab in the Canada or something like this that was the reference, but it was made on not on blood, but on patch, and it was on dry blood patch, and not the same techniques. And in our lab is serum activity. The lab in Canada is heparin activity, is plasma activity. The quantity of the inhibitor is not the same. So I think this functional test is not very reproducible between lab. It's very variable. So it's an indication to help us in the diagnosis, but not a real biochemical test as glucose or creatinine.
Okay. If there are no more questions, just one last one. So you do offer prenatal testing to this couple?
Yes.
Okay.
If the family is agree, yes. And after consultation of the committee, prenatal diagnostic committee in the hospital where the family is taken in charge, is the rule in France. But we offer prenatal diagnosis for lots of autoinflammatory disorder, CAPS, MVK, ADA2, yes. And we also have a prenatal testing for FMF this year. It was accepted by the local committee, so.
Okay, thank you very much. Another round of applause. And the next presentation will be a duo presentation by Laura Butter-Masso and Johanna Leetonen. And the clinical phenotype they are going to describe cases from, they will explain themselves, I think.
Yes. We can do the pointer thing. Okay, so thank you for this introduction, Marielle, and thank you for giving us the opportunity to present. And also thank you to Kimberly for bringing us together. So we are Laura and Johanna, and today we're going to present 2 genetic studies in autoimmune lymphoproliferative syndrome in pediatric patients. So first of all, I want to give a brief overview of autoimmune proliferative disorder. An ALPS is usually an early onset disorder characterized by 2 main criteria: chronic non-malignant, non-infections, lymphadenopathy, or splenomegaly, or both, and elevated CD3 double negative cells, which are the ones that doesn't express CD4 or CD8. These cells are usually less than 1.5% of the total lymphocytes, but in these patients, these numbers are quite elevated, as we will see later on. So there are other accessory criteria for ALPS, such as defective lymphocyte apoptosis, somatic or germline mutations in the candidate genes, and other molecular parameters. Just a word about the therapeutic options. Typically, ALPS patients are treated with corticoids, and then depending on the lymphoproliferation and the severe or not autoimmunity, they are given immunosuppressants. Also, in the most severe cases, they can undergo hematopoietic stem cell transplant. So now we're going to focus a bit more on these DNT cells, which we will see are really important. These cells are a very small part of the lymphocytes, and they will be super important in ALPS patients because they are usually the carriers of certain type of variants, which can only be detected on this population, but are really disseminated in the whole blood. So these cells can be isolated through different techniques. The gold standard, I will say, is flow cytometry and cell sorting, which enables us to identify these cells that do not express CD8 or CD4. But this is quite challenging and quite expensive and may not be available in all labs. So there are other strategies to enrich this population in our sample. For instance, CD3 selection through magnetic beads or rosette separation. And now Johanna is going to talk about the pathway.
Yes, so ALPS and ALPS-like disorders are caused if there are defects in the apoptotic pathways of lymphocytes. And if there are problems in those apoptotic pathways, then the lymphocyte homeostasis is not functioning properly. And then excessive immune reactions or lymphoproliferation can happen in the patients. And typically, ALPS can be divided in 3 different subtypes, and it can be caused by mutations in 3 different genes. So ALPS type 1a is caused by the variants in FAS gene, and that encodes FAS self-surface death receptor. And then FAS type 1b is caused by the mutations in FAS-LG, and it encodes FAS ligand. And then type 2 is caused by the mutations in the caspase-10. Heterozygous germline variants in FAS are the most common genetic cause for ALPS, but then somatic variants is the second most common reason for ALPS, and it's like up to 20% of the patient cases. And FAS variants, they can be located all over the gene, but then most commonly those FAS somatic variants are located in the intracellular domain, so in the exons from 7 to 9. And although FAS is typically dominant disease, it can be also inherited in the recessive manner. And then FAS-LG and caspase-10 variants are much rarer. And in 20% of the patient cases, then the molecular genetic diagnosis is not found. And then also there is for the previous slide, I forgot to mention that there is also over 20 genes that are associated to those ALPS-like disorders. So then next we can move on to our first clinical case. So we had, our patient was a 3-year-old boy, and he was an ALPS suspect manifesting the typical ALPS symptoms. So he had lymphadenopathy, large spleen, cytopenias, and increased vitamin B12 levels. And then he had also elevated number of those DNTs. So first we did exome sequencing for this patient, and we analyzed the exome data for PID genes, so primary immune deficiency disease genes, and our panel had approximately 400 different genes. then from this exome data, we could identify a perforin missense variant in the amino acid 252, changing asparagine to serine. And perforin is known to be an ALPS phenotype modifier gene, but it's not an ALPS disease-causing gene. And the same variant has also been described in another patient who had ALPS and who had also a variant in the FAS gene. But even though this variant is listed in ClinVar with conflicting interpretations of pathogenicity, it would probably not explain the phenotype of the patient on its own. Then our patient had two variants in the ATM gene. ATM causes recessive ataxia telangiectasia, and it's also associated to lymphomas. But then the other of these, like the phenotype also did not match that well, and then the other of these variants was pretty common, so we thought that maybe it's not disease-causing. And then intronic GATA2 variants are also known to cause a similar phenotype, but of course those we could not identify from the exome data. So then, in the end, we found out that this BID gene panel results, they were inconclusive, and next we would like to ask, what would you do next? So, A, just report this case as negative, or then P, check GATA2 haploinsufficiency with the method used in our laboratory, or then C, ask for an additional sample and try somatic sequencing with the targeted panel for cytopenia genes. So we also had this option in our lab. It has 200 genes for known cytopenias, and then better coverage as well. Okay. So just in case, we we decided to check GATA2 haploinsufficiency in this patient. And then on the left side, you can see the genomic DNA sample, Sanger chromatograms, and then on the right-hand side, there is the cDNA chromatograms. And this variant is just like a polymorphic SNP, and then from the cDNA sample, we can then conclude that actually the both alleles are expressed in the patient, so he does not have GATA2 haploinsufficiency. But then we also did the option C, so we asked for additional sample and got the lymph node biopsy sample from the patient, and then did this targeted cytopenia panel sequencing for that biopsy. And still, the findings remained inconclusive. And again, we would like to ask you, what would you do next? A, just report this case as negative, or then B, ask for a new sample and then try somatic sequencing using double negative T cells, or then C, do genome sequencing. Yes. So finally, sequencing of sorted double negative T cells revealed a somatic FAS variant in our patient, and this splicing variant is located in the intron 7, just like the most of the somatic FAS variants are located in those exons from 7 to 9. And this same patient has, or no, same variant has also been described in another patient as somatic earlier. And when we checked afterward, we could actually see that this splicing variant was found in the 4% of the reads of the exome sequence sample, and then 9% of the reads of this lymph node biopsy sample, with this targeted cytopenia panel. But then the reason why we did not identify this variant earlier was that we used a pipeline optimized for germline variants. And then that excludes all the variants seen in less than 20% of the reads. And then Laura will continue.
Hi again. So I'm going to present a second case. In this case, it is also a pediatric patient. In this case, our patient had splenomegaly, elevated DNT alpha beta lymphocytes, 7.4%, elevated vitamin B12, EL10, erythema, persistent cough, and unspecific fever. And for all of that, we had a very high suspect of ALPS. And my first question here will be, which would be the first genetic study to perform in this case, taking into account that this was 2017? A, Sanger sequencing of candidate genes, B, whole exome sequencing, or C, deep amplicon sequencing of candidate genes? You can vote. Okay, so at that time we did Sanger sequencing. Sorry to disappoint. And we sequenced the FAST gene, CASP8 and CASP9. And here I will show you the results for this sequencing in a blood sample with these elevated DNT cells. And unfortunately, we were not able to find any candidate variants. So we can see here just an example of the sequence. But then, as we knew about the importance of this DNT cell population, we decided to perform the same experiment, but using a sample enriched in CD3 cells using magnetic beads. And in this case, we performed the Sanger sequencing in the 3 genes. And in the FAST gene, in the exon 8, we found this variant, which is an insertion causing these frame shift patterns, leading to a premature stop of the protein. So we identified the candidate variant. And my question here is, do you think this approach could be used to routinely detect fast somatic variants? Yes, it seems reasonable. No, you were lucky. Or C, maybe, but it's not the best option. Okay, I'm glad to know that. You don't think we were lucky? I think we were a bit lucky because, of course, this was a frame shift variant, and you can see all the peaks that result from the insertion. But in case of a missense variant, this could have been very difficult to differentiate from the background noise. So yeah, I will say it's a mix between B and C. So we explored a bit more the variant, and we found out that it was not described at databases. And of course, it was located at a gene that was really concordant with the phenotype of our patient, and located at this exon 8, which, as Johan has explained, is hotspot for variants causing ALPS. So we considered this variant as causative, and our patient started immunosuppressive treatment, and he has been doing well since that. But we wanted to go a bit in deep in this study, and I want to ask you, which technology would you use to further study this variant? So to get to know the frequency of it, and which is the variant or the frequency of this variant at each time point. Whole exome sequencing, deep amplicon sequencing, plus a target search, so looking specifically to the variant that we already know is there, or deep amplicon sequencing plus a somatic caller, which will be more of a discovery mode. What do you think? Thank you, Marielle. Okay, so
still, you can still communicate.
People is taking a nap. In this case, we did option B, because we were pretty sure that this variant was likely pathogenic, and using IgV, it's quite easy to go and inspect. But we are also doing C right now in order to create and optimize a bit our somatic color pipeline, taking into account that we already know the candidate variant in this patient. So this was last year. We obtained a new blood sample, but in this case, the patient is under the treatment, so the percentage of DNT cells was quite low, 0.89%, completely in the healthy values. And we performed deep Amplicon sequencing at really high depth. Here you can see the total reads, which is a bit over 2,000. And then the reads here supporting our variant, which is the 4 base pair insertion. So if we just do a quick calculation of number of alternative reads into number of total reads, we can calculate the variant allele frequency, which is 0.68%, which is quite low. So again, as we had this CD3 enrichment being used in our lab, we decided to perform the same experiment, the deep Amplicon sequencing using cells enriched in CD3. And in this case, we can see that with the same number of reads, we have many reads supporting the insertion, and in this case, the frequency was doubled. So as expected, if we sequence a population which is enriched in our target population, we will have a higher frequency. And just to finish, we were studying this patient over 5 years, and we took advantage of having all that samples. We had blood samples in our lab from all these time points, and we decided to do a parallel analysis of the percentage of double negative T cells and the variant allele frequency of our somatic insertion. So here we can see that before starting the treatment, this was the initial time point when we performed Sanger sequencing, both frequencies were quite high. But then when starting the immunosuppressor treatment that we have here indicated with this line, obviously the frequencies were dropped dramatically because the number of DNT cells were reduced, and so it was the variant of the insertion. So now we are here, 2022, and we have these low values of both frequencies. And I want to finish with an open question, which would be, do you think it is possible to detect somatic variants from previously unsolved patients under treatment? I don't know if any of you have thoughts on that.
If you don't know the variant, it's difficult, no? Because you sequence, you have to cover the whole gene or multiple genes, so then it's quite difficult.
Yes, I completely agree that I will say it is possible, but you have to do a really target search. Thank you for participating, by the way. So if you have a concrete exon that you want to look at, or even a gene, it's a possible strategy. Or if you have a really good and optimized pipeline to do the somatic calling, it can be possible, but just doing it in the whole genome or even few genes could be difficult. But I definitely think, and we are demonstrating it now in our lab with some new samples, that this can be a nice approach. So yeah, with that, we jump to the conclusions.
Yep. So as a take-home message, we would like to conclude that DNT cell sorting and then CD3 enrichment increase the likelihood of identifying pathogenic somatic FOS variants. And then, as in our case, optimized sequencing and data analysis pipelines are needed to find somatic variants with low variant allele frequencies. And then investigation of somatic FAS variants can lead to molecular genetic diagnosis in previously unsolved AUTS cases. Okay, thank you. We want to thank our group members and Kimberly.
Well, thank you very much for this very nice and enthusiastic presentation. I apologize for the public, which was less active in this case, but very well done. Are there any questions?
Thank you for the nice presentation. I actually have more of a philosophical question. Is this academic medicine, or is this research, or is this clinical genetics? Is this done in the research labs, or is this part of the clinical genetic diagnostics?
Yeah. How to, maybe Anna can respond better, but
research, or you can say it should be a diagnostic collaboration. It should be diagnostic collaboration between immunology and a sequencing lab, because immunologists could help you with sorting the double negative T cells, and I think that's a requirement for this somatic analysis.
Yeah, in my case, it's clinical medicine, so this was a patient and we were working toward diagnosis, and now all these extra experiments that we have done with deep amplicon sequencing are more focused to research, but always with the aim to optimize the pipeline for diagnosis. So it's kind of a mix, but it's not only a research purpose in our case.
Yeah, and I definitely agree that it would be really helpful to collaborate to sort the cells.
Because in the end you notice a very low percentage of cells positive. In hematopoietic, how do you say that? In oncology, you look more at relapses. You know, when the mutation relapses again or the treatment stops. Is it in the case of ALPS? I'm not a clinician, so is it in the case of ALPS the same? So patients have to be treated for always, also when they have somatic mutations. Otherwise, it relapses again. Do you have any knowledge?
As far as I know, I'm not a clinician either, but I was talking with our clinicians, and apparently if they are doing great with the immunosuppressive treatment, they can go many years with that and then reevaluate the situation once they are not pediatric anymore. And obviously in these super severe cases, they can undergo the stem cell transplant. But I think it's, correct me if there is a clinician there, but I think it's not, that not happens that often in ALPS cases.
Any more questions? Yes.
I have a question for the first case. Which was the variant allele frequency in double negative sorted cells?
Yes, that we unfortunately don't know because the sequencing was then done in Germany.
Okay,
it was the clinician then decided to send the sample there, so we did not do it in our lab. Okay,
but so you or in the other lab, but they detected the mutation in the sorted T cells,
yes,
but they didn't give you the frequency of the mutation?
Yeah, probably then the clinician who sent the sample there got the report, but I didn't get it, like research side.
Okay.
So yeah, but I was thinking the same when I was doing this presentation, that it would be super interesting to know.
Just wondering that if you use a highly purified cell population, which are the double negative alpha beta T cells, you should expect a variant allele frequency higher than the Laura showed, obviously, in CD3 in rich cells.
Yeah.
So was I wondering that?
Yes, because it was in the lymph node biopsy sample, like 9%. So. Yes.
I also loved it. Very well done. One more technical question on your deep amplicon sequencing. Do you use technologies that have UMIs included, or is it without UMIs? So unique molecular identifiers to really trace molecules. Because if you use standard PCR, 1,000-fold coverage can either mean one molecule of the patient that is amplified and sequenced 1,000 times, but if you have these UMIs, we can pretty much trace that it's really 1,000 individual molecules of our sample. So whenever we talk about mosaicism below 5%, I would strongly recommend to use any kind of technology that has UMIs involved. Is that the case for your assays?
I completely agree. We are doing this in our genetics facility, so I'm not super sure about that. I don't know if you know that, but I will go for no.
I think it's something, it's becoming, there's like tens of technologies now using that, and there's so many features. It also allows you some consensus calling of over-amplified molecules with the same UMI, so we can sequence now better than Illumina ever offers because of this trick. So I think it's, I mean, especially in this field, I think it's just so important. I mean, there was already now 5 presentations on somatics and mosaics. I think it's super cool, but also very important to be very accurate on our technologies.
Yeah, completely agree. Thank you.
Okay. Well, thank you very much. It was a very nice presentation. Thank you. And for our last presentation of today, Jonathan Boland from the Institute Imagine himself is presenting a case, and I hope of a very active audience because the last 50 minutes, and then we're all free again.
Okay, so thanks very much for bearing with me. I tried to make this not longer than necessary, but I still hope to keep you entertained for this last talk. My name is Jonathan Bolen. I'm a postdoc here in the Casanova lab investigating susceptibility to infection. And today I want to present you a challenging case that taught me a lot about genetics. And I hope you can maybe also see something new. So I have a molecular biology background, and when I started here, I really had no idea about genetics or immunology. So I came with a little bit different perspective, and one of the problems that I was thinking about as I started was the idea that in each exome that we sequence, and you guys also do, we observe a lot of missense mutations, many of them also rare, but we have really a hard time telling whether they are functionally deleterious or completely neutral. And so this is just one small idea that I had as an additional analysis or investigation to predict more confidently whether a mutation could be deleterious. So I asked whether the missense site that we observe in our patients could be subject to post-translational modification, particularly when it's regulatory. And I hypothesized that in such a case, it would be more likely for such a variant to be deleterious. And so I used this, and maybe I can take the laser pointer. Yeah, I got it. So I used this phosphosite plus from Cell Signaling, which is an open database for post-translational modifications in different systems, including humans. And I overlapped it with our extensive WES and whole genome datasets from patients with severe infectious disease to find potentially deleterious missense mutations that we might have missed up to this point. And from this small query, I came out with 8 patients with homozygous and a little bit more than 600 patients with heterozygous variants in around 400 genes. And some of them confirmed my approach because they were already published disease-causing variants like STAT1 and STAT3. And today I want to present you with just one of them, which is homozygous CBL tyrosine 371 cysteine. So very briefly about CBL. CBL is a E3 ubiquitin ligase that negatively regulates and degrades activated tyrosine kinase receptors, including such receptors as EGFR, the T-cell receptor, the B-cell receptor, and FLT3. And you can already see that here 3 major lineages of the immune system are potentially affected by the activity of this protein. There are also monoallelic diseases already described for this gene. So people who harbor heterozygous mutations are predisposed to develop juvenile monomyelocytic leukemia, JMML, or Noonan syndrome. And the phosphorylation of this protein at the site 371 is documented in multiple publications to be essential for its activity because it causes like a restructuring, a steric rearrangement that facilitates the activity. And indeed, and here I just present this as a start just to take it out of the way, we confirmed that this variant is indeed loss of function for the ubiquitin ligase activity of EGFR here in an in vitro cell line-based system. So now I get into the genetics. So we have actually here the first big surprise of this project is that it's not one patient that we are studying in this pedigree, it's actually 3. We, to start with, we sequenced only one exome because they are monozygotic triplets. So I think at the time, and I was not there, it was reasoned that we can sequence just one because they're all going to have the same variants. So they are 3 French boys, and now they're 9 years old. And I studied the family segregation and confirmed that in DNA from whole blood, they are homozygous for this A2G substitution, resulting in the tyrosine to cysteine missense. But the second surprise was that the parents do kind of not fit with this homozygosity because it's only the father bearing this missense mutation. And here I think the mother being not the biological mother is not really an option. So this is the first question to the audience. What could be the mechanism of homozygosity in this pedigree? Sorry, I didn't hear it. Mosaicism could be, germline mosaicism, that's very hard to test, so. Uniparental disomy, very good, yeah. I think before I go to that, another option could be copy number variation, right? Could be deletion, but from our WES data, it didn't look like a deletion. The coverage was roughly stable, even though that's not the most robust readout. So we also hypothesized that it could be uniparental isodisomy. And so, just very briefly, that is a phenomenon where a child inherits 2 copies of one chromosome, either from only the mom or the dad and none from the other. Can also happen in a segmental way, where it's only a part of the chromosome that is affected. And this can cause disease in a couple of ways, including imprinting disease if imprinted genes are affected, or autosomal recessive disease if it leads to homozygosity. And when inspecting the WES data from patient 1, we indeed observe a large stretch of homozygosity here in chromosome 11, where the CBL gene is also present. And so this indeed looks like a uniparental segmental isodisomy of this chromosome 11q arm. And this is quite a large region, so it affects roughly more than 400 protein coding genes, including 2 that are potentially imprinted. And when querying the rare, potentially deleterious variants in this patient, or in the 3 patients, we nonetheless didn't come out with so many. So there's actually the CBL, which is a private variant. There are 2 missense on chromosome 11, which have a MAF of around 2%. So you could place the cutoff above or below as you, as you wish. And there's also an in-frame insertion in another gene. So at that point, we concluded that the most likely genetic cause of the disease is the homozygous CBL missense, particularly considering that it is loss of function, and loss of function mutations in this gene are really absent from the general population, and in monoallelic case already published to cause disease. I also want to mention that imprinting disease of chromosome 11 is already thoroughly studied, and it can cause Beckwith-Wiedemann or Silver-Russell syndrome, depending whether the isodisomy is paternal or maternal. And I just want to say that these patients do not display any signs of either of these 2 syndromes, so that also leads us to conclude that imprinting disease is unlikely in this case. Okay, another surprising observation was when we moved to sequence the additional 2 exomes from the 2 brothers of patient 1. At the time, we happened to have DNA from saliva available, and not thinking very thoroughly about that, we sequenced that, or maybe it was a serendipitous decision, because we observed then that as opposed to whole blood, where the homozygosity appears to be really strict to the degree that we can expect from WES data, which is error prone. In the saliva, there is a solid around 15% of the alternative, the maternal allele still present. And so here's another question: could be the cause of this intermediate allele ratio? It could be. So how would that happen? Somatic recombination, like a reversion, or?
This is reversion of the other allele, no?
Yeah. I think another. Actually, I didn't consider that, so that's a good point. For me, the mosaicism was the really obvious candidate because it's a different type of tissue, no? And I used also deep amplicon sequencing to then try to very accurately quantify the presence of this allele, and I used the CBL missense mutation here as a readout of the UPD. And indeed, I find that in some tissues it's completely heterozygous, like in the dead, so there is a complete absence of this UPD, whereas in other tissues it's variable, let's say. So yeah, so here, like this 15%, indeed are the mother's alleles. Yeah. In that case, yes. And so in summary, for the genetics part, these siblings have paternal, segmental, uniparental isodisomy mosaicism, causing CBL deficiency in some tissues. Incomplete CBL deficiency, let's say. Feel free to, yeah.
So this is a triplet?
Yeah.
Is the mosaicism identical in all three?
Yes, the 3 red points are the 3 distinct brothers. So they are strikingly uniform. And this is also, for me, really puzzling. So in Beckwith-Wiedemann syndrome, yeah.
So you can calculate back when the triplet split, I mean, that must be known anyhow, and when all the different lineages arrive.
Yeah, it's, this is something that I'm still processing. The splitting of the embryo can happen at different time points.
So this mosaicism must have occurred before the split then?
Yes, the UPD must have occurred first, for sure. Yeah.
No, in the residual cell, it must already been there before the split, otherwise it's impossible.
Yes. Yeah. So actually, segmental UPD is a mitotic event and happens in early embryogenesis. I think that's the trisomy rescue is in the case of whole chromosome isodisomy, whereas the segmental isodisomy is a mitotic recombination event, as far as I know.
But it also must be after differentiation between mesoderm and
Yeah.
because it's the same across tissue.
Exactly. It's a very distinct time point, so some cell lineages were already specified, apparently, when this UPD happened, right? And then the tripling must have occurred. And we also have the feeling that these 2 events must somehow make each other, influence each other and make each other somehow more likely, because they are 2 both very rare events, and just to coincide like that, maybe there's some causal connection, but that's speculative. Okay. With that in mind, now I want to present actually the clinical phenotype and the immunological description of these patients. And I invite you to briefly try to predict what the phenotype could be, keeping in mind that CBL mutation is connected to leukemia and developmental deformation. And I think it's a tough challenge because you won't be able to predict this. Generally, these boys are really healthy, but they have a disposition to develop pneumonia. And this is expressed to a different degree between the 3 boys. P1 is the most affected and has already had 2 or potentially 3 pneumonia, which have usually responded to antibiotic treatment. I have to say here that the infectious microbe has not been cultured, so we're not exactly sure, but we suspect it's bacterial infections. And at this point, at the age of almost 10, they all present some parenchymal lesions for which we are still debating whether they are emphysema or bronchiectasis or something like that. Here I actually present you with the CT scan, so this is the pneumonia, one of the pneumonia present in patient 1. And then afterwards, they develop this kind of lesions in the lung, and I'm actually, for any expert who is interested to give me some input on this, I'm very happy to take opinions. Additionally, we then carried out immunophenotyping using mass cytometry and find really a major disturbance of the immune system. So, and I'll try to summarize this briefly. So this, and the 3 dots are always the 3 distinct patients. So we find them to have a myeloproliferative phenotype where myeloid dendritic cells and monocytes are quite strongly elevated. They also have a really striking B-cell phenotype with B-cell lymphocytosis, roughly 10-fold elevated transitional B-cells, which then propagates on or also affects naive and memory cells, but not plasmablasts. The T-cell compartment is the most unaffected, but still slightly perturbed. So naive CD4, CD8 are strikingly low, but the memory compartment is essentially normal. And in the gamma delta, it's inverted. They are more naive and less memory. So this is really complex phenotype. And in addition, we also observe hyperglobulinemia for IgA and IgG. The last piece of data I want to present, and this is the one that we think is pointing us to the explanation of the clinical phenotype, but still preliminary. So this is a VIR-scan panel for pathogen-specific antibodies, a range of microbes, mostly viruses, but also some bacteria. You see here sex and age-matched pediatric controls, and so these are the normal levels of pathogen-specific antibodies that they present globally. Here are CARMEL-2 patients, which are CID patients that also present B-cell defect and really defect in forming specific antibodies, as you can see. And then here are the 3 CBL patients. And what I hope you can appreciate is that they present some kind of intermediate phenotype between these 2 controls. And the same is visible on a PCA analysis of this data. And so now we are working with the hypothesis that they have some kind of quantitative impairment in the humoral immunity, maybe related to a CVID, but somewhat less severe. With that, I want to thank everybody who contributed to this project, particularly the patients and their family, but also my colleagues and supervisors, and also all the labs that are collaborating with us on this work. And with that, I leave you with my summary and happy to take additional questions if there are any.
So thank you very much. Very nice presentation, Jonathan. And thanks to the audience for being interactive. Are there any additional questions? Yes, there are.
I have 2 commentaries. Looking at the lungs, it looked like emphysema, so I think it might be useful to make sure that they have normal alpha-1 antitrypsin phenotype.
We checked it.
That's normal. One. And secondly, it would be useful in planning the management to check their vaccine responses, say pneumococcal, tetanus, Haemophilus, and viruses, because, you know, arguably they could be candidates for immunoglobulin replacement.
Yes. So for the first question, sorry for interrupting, but yes, alpha antitrypsin is essentially normal, so it's not decreased. And I don't know for every vaccination from the top of my head, but generally the vaccine response in seroconversion is normal and okay. Also recently for SARS-CoV-2. Yeah, I'm not sure. I mean, for now, our hypothesis is not mature enough to warrant indication of treatment. Also, I'm not sure how IG replacement is compatible with hyperglobulinemia. Yeah, yeah. But the vaccine response seems okay. So I really think if the B-cell hypothesis is correct, it's really a quantitative defect. It's not a complete abrogation of B-cell function. Yeah.
With the monocytosis and also the gene being potentially reported in JML and things like that, do you worry that these kids are at strong risk of developing hematological malignancy?
Yeah, we were very worried about that. And actually the historic monocyte counts, which I don't present, have risen up to almost 4,000. So they were really high at the age of 3 or 4. Maybe that would even suffice for a JMML diagnosis. And then they are sloping down ever since. And for that worry, the clinicians agreed and have recently obtained bone marrow and profiled for blast and clonality, and it looks all okay. So there's no sign of leukemia at this point, but I still believe they could be susceptible for the whole of their life. Yeah.
Is the diversity of your B cells given this high number of B cells, did you look at the diversity of the B cells or for the repertoire?
Not yet, but we are doing the repertoire for naive, memory, and transitional B cells, and it's going to be very interesting. Yeah.
Thank you for the presentation. Just a clinical information. Do the patients show some degree of hepatosplenomegaly? And the second question, if it's the father that is heterozygous, shows some kind of phenotype?
Very good question. So the father is, as far as we know, healthy and has no history of leukemia. But also the clinical penetrance for monoallelic CBL mutation is not so high. It's probably below 25%. I don't know. And the physician has noticed that the spleen is slightly enlarged, but like at the upper end of normal range. So they have no severe hepatosplenomegaly.
Thank you.
Are there any more questions? If there are no more questions, and there are no... still one question.
Do you consider transplantation, allogenic transplantation for those patients?
Yeah, I think it's difficult. First of all, it's not my decision to make, and I have very little expertise on that. But I think also, given the fact that they are really essentially healthy at this point and haven't suffered from a pneumonia in years, I'm not sure if it's warranted, although the trajectory of the lung phenotype on the CT scans is worrying. So, but then that is really complicated to think about because it could still be a lung intrinsic phenomenon. We don't know the genotype of the lung, and bone marrow transplantation would not rescue a lung intrinsic phenotype. So it would also be speculative, I think. And I have thought about this, but it's also not really easy to genotype the lung at this point. So I don't know. Yeah.
Well, thank you, Jonathan.
Okay.
Thank you. Very nice. So then I would like to thank all the speakers of today for these marvelous sessions we had. I had very an interesting day today with very nice presentations. I would like to thank all the speakers for this. And the speakers of tomorrow can upload either now or tomorrow morning their presentation. And is there anything else I should say? No. So then I would like to thank you for the day. Enjoy Paris and see you tomorrow morning. Thank you.